Hello, 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 hello. Okay, everybody, Assalamu alaikum. Good morning, everybody. So everybody is ready today, inshallah. So today's my talk is little longer, okay? Because this I had to cover a lot of things in this. So new therapeutic modalities in drug discovery, as you know. Okay, let me before I start. What is that? The picture. Can Very good. Okay, so you are going to learn a lot about antibody today. So I will try to cover part of new modalities in drug discovery because as science is moving, there are a lot of new things are coming. So earlier it used to be back old days when there was no medicine, modern medicine, pills and all those things. We used to have the herbs and all those things. Now there are pills came, antibodies came, and proteins came, and then there are a lot new things are coming every day. You heard about vaccines. They are messenger RNA vaccines, only RNA strand. So I'll try to cover as much as possible. It is going to be a little bit longer, I hope. So let's move on. So these are the new therapeutic modalities. I have to stand here because he's making a movie on that. So in last 20 years, there are 17 new modalities has been discovered because of the new medicines, new diseases has come up. So earlier it used to be a small molecule. When I started my career in India and US, it was only a small molecule. I did not touch anything else. Then there was antibody drug conjugate that came, and I'll answer some question on that. Then targeted, this is a brand new technology which is being used, which is called targeted protein degradation. So disease are caused by proteins, and if instead of inhibiting that, can you close that door? So this is a brand new, so I will, because I am working right now, other than a small molecule, I am working on antibody drug conjugate and targeted protein degradation. So I will give more information about that. Other than that, there are RNA targeted small molecule. So a small molecule RNA vaccines, everybody has used that. There are many RNA vaccine. Just last week or so, President Putin from the Russia, he announced that he has a RNA vaccine for to treat the breast cancer. So it was in news, and they are going to give free throughout the world. RNA therapeutics are there. Macrocyclic peptides are there. Macrocyclic peptides means, if you know peptide, it is sometimes open chain. So if you make a ring of that, or if you connect one end to other end, it becomes cyclic. So that we call it macrocyclic peptide. Gene therapy and editing. This is a very, very well known modality, but it is very expensive. Gene therapy is very expensive because you are going to change your DNA, or you are going to change DNA ex vivo, means outside your body, and then inject that DNA into your body to cure the disease. And editing is CRISPR editing. We know everyone knows that CRISPR is an editing tool to edit DNA of a disease molecule. Engineered cytokines, I don't know, you remember, cytokine storm was a word used during COVID time. It means that inflammation. Cytokine is inflammatory proteins. And when people used to get sick, doctor used to say, oh, he has a cytokine storm. Means cytokine was generated by body and they were attacking your own body. So you have to contain that. So there is engineered cytokine which will not, attack your body. Cell therapy is another modality, and oncolytic virus is it is working on. People are working on for 10, 20 years. So these are the couple of new modalities which I will discuss. So black color one I will discuss briefly. Red color ones I will discuss a lot, because I cannot, if every modality need one lecture like an hour. So you can go ahead and read all those things, so you will get the slides. Okay, so antibody drug conjugate. So since I'm working on that, not only I am working, this has become very popular and it is very safe medicine also. So people started working on that, and I will give you the history how it starts. Then mechanism of action of disease cell, component of ADC and its assembly and means synthesis. I will give you a flavor of how it is synthesized. Actually, I have an approved project on this. And one of my, because in Saudi Arabia, we have to have high school student who will come and work with me. Because they have, this is called Mohiba program. I think we should start something like that here. So high school student right from the school, summer they will come and they will work in our lab. Usually they don't work in the lab because we don't allow them to touch the chemicals, but they will see or shadow somebody. So just I got that one. So this project I gave to one of the student, and he is in the next step. So there are steps where people go to from one place, they call it Talmida, IBDA, and then ISEF, which is International Olympiad. So this person has been selected, went to next step of the program. Then approved ADC drug, then use of ADC beyond cancer. I will not talk about that because I think I will cover only cancer part and then summary. So history of antibody drug conjugate, and you can see that in 1913, Paul Ehrlich, he describes the concept of magic bullet. What is magic bullet? Magic bullet means the pill which will treat everything. So you take one pill and you are cured from all diseases. So that was the proposal. It was by Paul Ehrlich. He won Nobel Prize also in 1910. Okay, and this was concept, and you can see from there nobody has done anything here to here. Means concept. He gave a concept. Who cares? Nobody wanted to do anything. Then in 1950, somebody started doing few things. And then slowly it started picking up. ADC proposal for was there on 67, and moving on, there was one person who asked about that which drug was withdrawn. So Pfizer made this drug called Mylotrag in 2000. Okay, and this was a ADC. With colchicine and another antibody with a linker. I will explain all those terms in next slide. So what happened that this drug was on market for 7 years. So from 2000 to 2007 it was on market, and in 2007 it was withdrawn. So he was asking that it is possible, can somebody withdraw that? Yes, this is a huge conglomerate, big company. They have to withdraw because there was some issue with dosing, and it was toxic also. Why it was toxic? There was a reason for that, because the drug colchicine was releasing before it reaches the target. So the concept for antibody drug conjugate, which will come in the next slide, that how it works. means you can use calcium like Taxol alone. You can antibody alone, and it will be cured like doxorubicin, etoposide, mitoxanthone. All are used alone. Also, they are toxic, but they treat the patient. Why to do antibody drug conjugate? So the next slide will explain to you. This is the mechanism of antibody drug conjugate. So what happened there on the cancer cell, there are antigens. So if you have an antibody specific to that antigen, it will bind only to cancer cell, okay? Now, okay, so what? It binds that, right? So what we do that we make a toxic drug which is called calcisiamycin, for example, or doxorubicin, for example, and we have to connect that drug to a protein which is antibody. You can see there. So this is antibody drug conjugate, and you'll see in next slide how it is being built. So you can see that this is the antibody drug conjugate. It has an antibody which is very specific to the antigen. This is antigen here, okay, and the drug is attached to antibody. So it is like a targeted targeted attack, I will call it, okay. This is a missile which is going to target timely only at that part of the cell, okay. So that's why we call it warhead, okay. Warhead is a word is being used on TV a lot. So this becomes a warhead for this cancer cell, okay. So the antigen is here. This antibody with this drug, which is linked to here, binds to antigen, and then the process called internalization. So the antibody drug conjugate, okay, I'll go back. It internalized, okay. So internalized means it gets into the cell, okay. Then mechanism starts. There are lysosome, endosomes. So now this drug has to be released from the antibody, then it will work only, right? So there is a process that the antibody will be destroyed by lysosomal enzymes. So because the antibody is a protein and there are protein enzymes there, they will destroy the antibody, then it will be released. The drug will be released inside. So what happened? I don't have to take calcisiamycin and doxorubicin directly IV. We have to bind it to the antibody. Antibody will go only to cancer cell. It will attach to the antigen which is on cancer cell, internalize. Once it is internalized, once it is inside the tumor cell, as I mentioned here, it is inside the tumor cell. Now other cells are safe. The healthy cells, they will not bind the antibody. So these antigens, one more point I want to make sure, I think you will ask me. These antigens are only developed on the cancer cells. They are specific. So researchers, they figure out how these antigens, which are the antigens which are on the surface of the cancer cell, not on the normal cells, okay? So it is clear and mechanism. Then what happened that it is by lysosomal, degradation. This is degraded. The drug is released. You can see the drug is released here, and some of the drug they are going to, and mostly cancer drugs, mostly cancer disease is due to DNA. So it will go to DNA, bind in different mechanism, whether it is intercalation, whether it is alkylation or whatever it is done, and then this will destroy the nucleus and the cell death, the cancer cell death happens, okay, everybody. But sometime these, we call it, they usually don't, they release early, okay. So we call it bystander effect, but it happens very rarely, okay. So no modality is 100% safe. That's the message I want to give you, okay. So ADC, this is the mechanism of ADC. When you get this slide, please study every part of that, and you will enjoy how it works, okay? Now, coming to the little bit of construction, okay? Yesterday I said that, and in my first lecture also, I think I said that molecules are built like brick by brick, like this building is built, carbon, hydrogen, nitrogen, you have to connect. Same here also, it will, we have to do that. So this is antibody. On antibody, there are few attachment sites. There are two kind of amino acid, lysine, which has NH2, means amine, and other is cysteine, which has sulfur. So both can be used. The chemists are smart. They know how to do that. So I will explain in few slides. So the attachment site is fixed now. Antibody, we know. Attachment site, we know. Now linker is very important, and this is the chemist, because if you put a strong, if you put a like a linker like a steel rod, it will not release the your drug. So you have to have a linker which can be, which can release this drug easily, and there are mechanism of that, and I will discuss a little bit more on that. And then the drug, and then you have to choose a nice drug. For every cancer, there are different drugs, so you have to choose a different drug, okay? For breast cancer, people use doxorubicin also. For colon cancer, there are other drugs, so you have to choose the different drug for that. Okay, so antibody, this is target a well-characterized antigen with high tumor expression. So I already explained that the antigen is expressed on tumor and it should be high so that the antibody can bind to that. Maintain all the properties upon conjugation to the drug. That's another. So antibody is a protein. When you do some chemistry on that, it can change. So you have to think about it, how you are going to do that. Maintain non-specific binding also. So there are amino acids. There are two amino acids usually we use, but there are other amino acids. So if you use a proper chemistry, it will bind only to cysteine and lysine. Okay, attachment sites through cysteine and lysine. I already explained that. Variable drug antibody ratio. This is an important part. It will be explained, and it should be site selective conjugation also. So it will be site selective means it should be on this part only, which is FC part, we call it, okay? So the conjugation should not be here and there, because this is the part which is going to bind to the antigen, okay? So that's important. Linker, this will be cleavable and non-cleavable. Those both kind of linker we use. Stable and selective release of the drug, this is very important. If you use a weak linker, as soon as you inject, it will release. You don't want that. You want a linker which will circulate, goes to the cancer cell, bind it, get inside, and then release. As simple as it is, okay? So, and drugs, this will be highly potent. So in this case, we use very, very toxic, highly potent drug which cannot be given IV, okay, for the cancer. So as bad drug as possible you use here. That's what I will say, okay? They should be non-immunogenic, because immunogenicity is a problem. If drug is already immunogenic, you will start getting, before even reaches to target, you see the side effects could be linked to and this drug should be linked to antibody and defined mechanism of action. So this should have a defined mechanism of action, means it should be doing on the nucleus, like you can see here, microtubulin, DNA alkylations. So it has to do something within the cell. Okay, so evolution of antibody, I already talked about it when it started. So the technology started in 1975, although Paul Ehrlich already told in 1910, but we started doing that. And the first approval was a very, very weak, very small doxorubicin antibody. It was not even antibody. I will not call it antibody drug conjugate. Then Marion et al., he started making some. So this was by Morrison in '86. Joan et al., he made a dadizumab, And then real antibody started in 1990 and then 1994 by these two gentlemen, okay? So the classification of antibody is also important. When you see drug, you will see Z-mab, Zumab, S-mab. So why it is like that? So I have already mentioned, when you see on something OMAB in literature, it means it is a mouse antibody, okay? Because we start working with mouse. We don't work directly to human. So we have to make a drug for mouse first and then make a drug for human. Okay, Zimab is like 35% mouse and 65% human. When Zumab, actually, these things I have to remember myself. I really forget sometime that what is the meaning of Zimab, Zumab. It looks like some Chinese language, but you have to remember that. In literature, you will see a lot that if you see something, they call it Zumab, it means this is 5% mouse and 90% human. And then UMAB is 100% human. So for human, most of the time, 90% time, we like to use humanized antibody. So there are production procedures, so we use only humanized antibody. But in certain cases, mouse antibody also can be used, so it is not off the hook. Okay, so when you do that, okay, yeah, that's important. I just, in previous slide, I talked about immunogenicity, because mouse has a different makeup of antibody with different amino acid, and human has a different, although they are same antibody. So immunogenicity, it is a problem. The mouse antibody will not be very good for human. It will be immunogenic, and there will be side effect. It will kill you. But as soon as you go to human side, human antibody, that works very well. So, but you have to start, for making antibody drug conjugate, you have to start with the mouse. So that's the point I want to make, that's why we do that. Okay, conjugation strategies, sometimes it is site specific, sometimes it is random. I already talked about almost half of that. So random is like lysine is on the surface of antibody, cysteine is on the surface of antibody, so we do that. So in case of lysine, it is all over, but cysteine is only 4 of 5. So we break the bond of cysteine and then try to bind that. So then we have control. How many drug per antibody is going? We don't want 20 drug per antibody. We need only maximum 4. So this is what we call is DAR, or drug antibody ratio. Okay, so in some cases there is no cysteine, means very little cysteine on antibody. So we engineer, which is called protein engineering. So we put more cysteine in the antibody, and it will not affect its work. Then there is enzymatic modification. We have to do that sometime, and we use an enzyme called transglutamase. Then conjugation to non-natural amino acid also. So there are 20 natural amino acids. There are many non-natural amino acids. So sometime we use that also, and by protein engineering, we put in the antibody just to make sure that our drug is attached to that. Sometimes, lysine and cysteine is not enough. So some other amino acids you have to use. Okay, so linker strategy, this is more chemistry part. So linker can be two kind, cleavable, non-cleavable. I said that it should cleave, but in 1 or 2 cases we have seen that if it is not cleaved, even then it works. So it is very strong binding antibody drug conjugate, and it does not need cleavage. So it just goes inside the cell and then bind to the endoplasmic reticulum or DNA or Golgi apparatus, and it works. And the whole molecule is destroyed by lysosomal enzymes. Enzymes are within this cell, okay? There are many enzymes are there. So, so we call it acid labile, which is hydrazone. Those who have little chemistry background, they can understand better. We have valine and cysteine, and that is what I am using in my research. Because I don't have big resources to do other. So I am using valine and cysteine, and we have synthesized. This is, we call it maleimide, and this part we have already synthesized, and and this cysteine formation is very easy on that. So we design that we can work in the lab properly. Okay, we don't have full resources like pharma company has. Then mercaptoacetyl caproyl antibody. This is just length of carbon. That is why it is called caproyl. Maleimidocaproyl. This is another one where the where you form the amide bond. So it depends on your drug. You should have a handle to bind to this linker. So we have to maneuver here and there and see which can be good for other. And then MCC, and there are many more. There are many new ones came. I wanted to just emphasize that I am using this one at present. Okay, I cannot use everything. Okay, so payload means the toxic drug like doxorubicin. I'm just mentioning few of them here so that you understand how diverse the anti-cancer compounds are. That's the point I'm trying to make. It is not like one compound, okay? It is like, look at the structure of the compound. And selection criteria is very important. As a team, we sit down and we see, okay, which one is we can use for this particular antibody. I worked on, in my previous life, I worked on prostate cancer. This, we call it mesothelin in Bayer, and that was a different story. Here I am doing for breast cancer. So you can see there is a auristatin, doxorubicin, nimorubicin, and PNU. They are very, very toxic drug. If it is given by injection IV alone, it will have a very bad side effect. So that's why we have to make very specific delivery part. You can call antibody drug conjugate as a delivery also, drug delivery, which is part of the course, okay? Because you are using a specific delivery vehicle, which is antibody, to deliver your compound to the cell, okay? So everybody get there. So this is CBI dimer, and you can see their potency, 0.0007 nanomole, means it is 7 maybe picomolar or femtomolar. It's so toxic. This much amount is more than enough to treat your cancer. So that's why I wanted to show these structures that you should know that how potency. Look at this auristatin, 0.07 nanomolar means it is 0.7 picomolar. You cannot imagine what is picomolar, okay? So it is very, very toxic drugs, and they are all used in the practice in many places. The other one is tibulicin, 0.01 to 0.8, very potent molecule. Everyone has a very different structure, very different structure. And this is statins, and this is catechiamycin. I talked about catechiamycin. This is very toxic drug, and this was the part of Milotrag, which was in market and then withdrawn. And then again it is back. Last year it is back because someone found out that the dose which was given early was very high, and it was given one time. So the clinician said that, okay, let's divide 2 milligram dose into 5 times in 24 hours. Still it works. So it is that this is a one drug in the history of FDA. It was there, it was withdrawn, again came back. So this is called Milotrag. So that is for one of the. So this is the compound. You can see the complexity of the structure that was bind to the antibody, and it was done by one of our Indian fellow actually in Pfizer. I know him. It is just we have to have the resolve and the courage to do that, and he did that. Okay, I just listed, I'm not going to read that. This is the payload. This is the very latest Nature review which came. So you can see earlier it was like 1 or 2. I used doxorubicin and auristatin in my work. But now there's a lot of choices you can use that. So it is not one, because research has grown, so it is a lot of choices now. So this is the list, and I'm sure that you get the slide and you can read this literature also. And this has given the payload classes and what they do actually, whether a mode of action is given there, like inhibition of tubulin. Inhibition of tubulin polymerization induces DNA double strand break. Similarly, DNA alkylation, DNA cross-linking, DNA intercalation. So these are all words. When I started working, my postdoc was on anti-cancer compounds, so I learned that alkylation is different than intercalation, and linking is different than so DNA is a prime target for the cancer, and then stabilization of topoisomerase inhibitors. This is an enzyme which is usually expressed in the cancer patient, so that also can be targeted. So these are all very new. You can read that. Okay, assembly of components in of all of now how we actually make. So we have the antibody, okay, and as I told you that I like personally, so that's why I put it there. I like personally that cysteine should be the target, okay, not the lysine. So we have 4 cysteine bonds in antibody. Usually we have 4 cysteine bonds. Why I target cysteine? Because I can control. That's all it is, okay. So there is a partial reduction by TCAP. This is a chemical compound. Easy thing to do. Once you have open sulfur, you use enough TCAP to reduce only one bond. You need little bit more to reduce another bond, okay. So this is again, I say control. You can control it. Then we conjugate with the maleimide here, like that. This is called a Michael addition in chemistry. So we do Michael addition here, and then after Michael addition, either you start making work here. There are things that you can make the whole drug from here and just do last step, or you can start making different way. Lysine is also there, so you can do through this. So there are many ways, so I just simplified the whole mechanism. It is not as simple as look here, but this is the way it is done. So in 2 cases, 2 antibody drug conjugate which is on market. One is Adcetris, which is heterogeneous ADC, and this is Kadcyla. I think it is already available in India also. I know that some of the companies, they are making already. So these are the 2 approved. I just wanted to show you the complexity of the structure. They are sold in like 10 to 15 billion per year in many places. In India, both of them are available. What they are, this is brentuximab vedotin. This was approved in 2011. The antibody is brentuximab directed to CD30 that is expressed on HL and LCL. Attachment site for me is thiomaleimide caproyl. Caproyl is the long length chain of the carbon. Spacer is PBAB, which is here, right here. This is your linker part, and this is the warhead or cytotoxic drug. Okay, so this is Excedris. This is made through sulfur conjugation to antibody. In case of Kadcyla, antibody drug conjugate, this was approved in 2013. The MAB is for breast cancer, HER2. Attachment is again through lysine, and we have to put another part into that, and then okay. So these are the linker. We have to increase the length so that it can internalize properly and go inside the cell. And the drug is mertansine, very potent, very cytotoxic drug. Okay, so these are the 2 I wanted to show you the enzyme example for that. Okay. How much is okay? So I have to rush a little bit. These are the approved antibody. You can read that there are 13 antibody drug conjugate used. Now, in like 2020. The first was made from 2020 to 2000. 2000, sorry, 2000 first was made, and 2007 nothing was made other than that. That was withdrawn. So from 2007 to 2025, 13 antibody drug conjugate made already, and you can see that how new it is. Okay, everything, and in last year there were 4 of them they approved. So this is a good area if you have resources to work on that. And so this is the 2025 data which I am giving you. The second is clinical trial landscape, and there are 200 clinical trials are going on right now. So using antibody drug conjugate. So this is about antibody drug conjugate. I will move on. I told you that I will talk about protein, targeted protein degradation, or PROTAC, what we call it. This is very important. This can be done very easily here also. So this is a new emerging drug target, okay? Outline of the presentation. So I'll give tribute to a pioneer of PROTAC, introduction, attributes, why PROTAC, mechanism of degradation, TAC universe, and conclusion, challenge, and recommendation. I was fortunate enough to work with this person, John Houston, in Bristol Myers Squibb. He was the one who started antibody drug conjugate, not antibody drug conjugate, the protein degradation in Bristol Myers Squibb, and then he left and he started first PROTAC company in the world, I will say. So he was visionary. He thought like our Dr. Azhar is a visionary. He came from US and Canada, and he said, I will set up this thing. Okay, he is also like that. He left a very lucrative job. He said, no, I want to do what I want to do, okay? And when we were talking to Mr. Syed, he also is like that. He said he went to very high and dropped to 0, and then again started. So he took the risk. Dr. John Houston, he also took the risk. His company is called Arvinas. It is valued at $3 billion right now. And so first compound which he made is already in phase 3 clinical trial in 2019 for prostate cancer, and there is another compound, Arvinas 471. This is also in phase 2 clinical trial. So this is the person who is a visionary, and as I said that I was very fortunate to work with him in Bristol Myers Squibb, and sometime I'm in touch with him. He is the chairman and chief executive officer of a company called Arvinas, okay? So who are these people who thought about this protein degradation in the body? And these are the 2 academic people. So this is the academic institution. This whole protein degradation came from these 2 people. You can see their paper. The year it was published in 2001 in Proceeding of National Academy of Sciences. Professor, so I want to give tribute to them because this is very, very new. This is like exploding in US, this technology, which is called protein degradation. Professor Sakamoto and Professor Crew, he thought about it that, okay, every disease is due to proteins. So what we develop a compound or a pill or a drug which will inhibit the protein, production of protein. Why not destroy the protein itself? And you have to be very selective. If you are going to destroy protein, you should not destroy all the proteins which is in your body. So they published the first paper, and so this was a PROTAC. In our paper was peptide degrader. Now we are using a small molecule. Peptide have problem with stability and delivery, and second peptide degrader were published in 2008. So now you can see that 2000 and 2001 and 2008, everyone thought, oh, they are useless people. Why they are destroying the protein in the body, okay? And doctors call us to take more protein, and they are calling to destroy the protein. No, but it happens that this became a very, very good way to treat people and do research also. So now just this morning, Dr. Azhar was talking about post-translational modification. So post-translation modification are, so I will explain more little bit about that because we just talked very little about that. So I will explain little bit more that PTM or enzymatic modification of protein after protein biosynthesis. These modification, so these modification have lot of things happening there, okay? So I will put the words and you can check later. That includes phosphorylation, acetylation, glycosylation, hydroxylation, methylation, sulfation, ubiquitination, amidation, sumylation, nadylation, isdylation, lipidation, acylation, and myristylation, palmitolation, and farnesylation. I cannot explain everything right now, but if you Google, you will find out and ask your biochemistry teacher especially that they can explain all those words. All of them are very, very important in protein degradation. The following event impact the protein of interest the following way. So the proteins are there and these PTM events, what they do. So structure, which is folding and conformation. I talked about alpha fold, beta fold, gamma fold by Google. That is one thing. Function, activity, interaction, and signaling. So these are the protein functions. So, sequestration, trapping, compartmentalization, and distribution, sorting, secretion, and stability. These are the function of that. So dysregulation of PTM of protein may result in aberrant or disease-causing protein. Aberrant post-transcription factor. Aberrant means the protein is bad, okay? So this is the word we use scientifically, okay? So dysregulation of PTM, post-translation modification, what they do, they produce aberrant protein because it is not normal now, it is abnormal. So they produce disease-causing protein. So aberrant post-transcription factor could lead to various diseases like cancer, neurodegenerative disease, diabetes, immuno-related disease, and there are many more. So I just, these are the diseases we are tackling right now. Most of these proteins are undruggable. So this word I'm going to explain a little bit. Only few has been targeted by conventional molecules. So I told there are 23,000 genes. They produce about 4,000 known proteins in the body, and out of 4,000, only 400 are being targeted to treat a disease. What about 3,600? So we call it undruggable because we cannot drug them. We cannot, drugging means nothing else but treating them. So that's the word we use for undruggable protein. So therefore, targeting undruggable protein is a new area of therapeutic intervention termed as targeted protein degradation, TDP, or in other words, we call it proteolysis targeting chimera and molecular glues. I will not go into molecular glue, I will just stick to PROTAC, okay? What are the attributes of targeted protein degradation? So this is already I mentioned, I will not repeat this, only 400 protein are successfully targeted right now. So why PROTAC? Why not anything else? Because some of the diseases are due to these proteins which are aberrant. So we have to find out another way to treat those diseases. So now we are using this technique. So a small molecule cannot target undruggable protein due to structural complexity and off-target effects. We know that side effects are there. Traditional inhibitor and antagonist only block part of protein function, occupancy-based PROTAC degrader system. Some enzymes inhibitor have drug resistance. This is happening all the time. You develop a drug, there is a resistance for that. In bacteria, there is a resistance for bacteria, okay? So we have to develop something that we avoid the resistance and the drug should survive for longer time. Degraders are active in catalytic manner, reducing the possibility of toxic side effects. So protein degrader, they can recycle themselves, okay? You give 5 milligram, this is enough maybe for your life or for a week. So that's another advantage of using protein, because protein degrader, they don't destroy easily themselves. They just destroy the protein, which is aberrant, which is bad, right? So they are still there. So this is another advantage. You don't have to take all the time pills. It may be one pill for 1 week. Protect can target both druggable and undruggable proteins, and they offer multiple advantages. And there are disadvantages also. The mechanism, how it works. Protein degradation process in the cell. So, so it works through a garbage cleaning system in the body. Now, what is garbage cleaning system in the body? So proteins are being generated in the body, but if you keep on accumulating, think about it, you have a kilogram of protein, right? All kind of thing, which you eat protein, it has to degrade somehow. So there is a in-body, nature has given us a system, we call it UBQ or UPS system, which is called ubiquitination proteasome system. This is the natural cleaning system of the body, okay? So this is what a God gifted. Otherwise, we will be, having a lot of protein in our body. So that's why we call it the protein can be degraded by 2 pathways. So E3 ligases, these are the enzymes. These are the target protein which we want to degrade. So E3 enzyme is part of this UPS system that binds to the target protein, and this is the protect. First, we have to bind this into ligase and then let it go into the body. This will bind this ligase or cleaning system, which will bind to target protein. There will be complex form and it will degrade only the protein, and then your ligase is still, your machinery is still there. So this is, we call it natural garbage disposal system or UPS system. So again, in this case also same thing happens. It has to internalize, but not through the antigen directly. And there are ways to do that, okay? We have to design the molecule which can directly go inside the cell. So proteasome eliminate the short-lived soluble misfolded protein by UPS system, and lysosome are responsible for long proteins, insoluble proteins, aggregate, and entire organelles. So this is inside the cell it happens. So this field has grown so much that I call it universe. So tech universe, protech, I call it, okay? There are many things. So there are trivalent protein, bioprotech, RNA protech, aptamer protech, glutech, peptech, nanotech, phetech, photech, ectech, riptech, aboprotech, traftech, phototech, and PC protech. All of them are looking for a bad protein in the body, okay? So I said that this field has grown out of proportion, and it's very successful. There are many companies who are working, all big companies are working on this technology. Chemistry-wise, it is very simple. Example of targeted protein degradation, I think it is very faint. You may not be able to see properly, but just tell you that there are these compounds. They are already in clinical trials, okay? And in this case, only the one thing which I will, maybe hopefully I will show that the UPS system has a binding pocket, okay? And that is a specific, and chemists, they are keep on improving that binding molecule, okay? That binding molecule has to be there. Okay, and there are these protects are all in clinical trial, and this is very latest data which I wanted to show you. Future of protect discovery, it is not going to be limited to cancer only. It will be going to many other disease areas, and I have already proposed a proposal about a cardiovascular problem. So you can see that earlier it was target protein was these, and there was only one system. Now you have cancer, immunoinflammation. Actually, I'm really interested in infectious disease because somebody was talking about AMR yesterday. That is, and then Mr. Sayed was talking about AMR also. There is not much there. Just only one nephromycin has been approved, and I'm sure, I hope not, that it will get resistance soon on certain bacteria. This has happened to all antibiotic, okay? It is well-known fact. So maybe this technology for AMR, and I will talk to Mr. Syed about that privately, that we think we should think about that, infectious disease. And we have in Saudi Arabia, we have a huge problem. Dr. Azhar knows that we isolate a bacteria from hospitals and they cannot be treated with any antibiotics, okay, even combination of antibiotic. And by WHO, I believe Dr. Azhar can correct me, that after cancer, AMR is the next, that's true. So next most killer disease. We don't think about it, but it is there already. And in neurodegenerative disease and many other more. So I'm looking into cardiovascular disease in this area. Okay. Rest of the modalities, I told you I'm not going to have time. I'm already over time, but I will take 5 more minutes, inshallah. Okay. Okay. No, 1 hour. So I will just wanted to give you the flavor. What else is there in the rest of the modality therapeutics? These are the one brief discussion. I'm not going to say more anything about that. You can, when you get this slide, please read it. RNA targeted molecule challenges. It's very, very important. These days, RNA is the key now. DNA is old one. RNA is the new kid on the block, I call it. RNA therapeutics, macrocyclic peptides, radiopharmaceutical, and new isotopes are used. Usually it used to be very few, but now there are new ones. Gene therapy and editing, cell therapy, oncolytic, these are highly sophisticated work, which can be done only in very highly sophisticated labs. But I think it is a good idea to think about it now. It can be done here also. That's, I mean, so the sky is the limit, okay. Just artificial intelligence, again, this can be used to improve the drug discovery process, and we are using artificial intelligence in all modalities. So it has a huge effect on us. I will just skip that. You have already seen this before for target validation and other thing. So in summary, I just put this slide that what I talked today is novel chemical modalities. You can see that protein degraders, antibody drug conjugate, gene therapy, protein, oligonucleotide and RNA, and strategic thinking is required in early drug discovery, which one you want to do, and you should know that you can do it. And these modalities are the future of drug discovery and delivery. I will answer any question if you want. It was little heavy, but this is the trend now. So I think you should know and you should learn, and there's a lot of literature already there. Very simple ones are also there. Say it again, please. Yes.
How do we know or analyze that what causing DNA or gene causing the DNA?
So there are tests which are done, okay, and mutations are there, okay, and mutation analysis are done, and in terms of finding how, which DNA, what kind of DNA, where DNA. So you have to do the biopsy of the cancer, and then you will find what is the problem. Yes. Okay.
So we can determine the exact cause for tumor?
It is possible. I think there's a huge mechanism is there for that, and you can determine the cause of the cancer. It may be environmental. Now, how you will say, the air is very bad outside, so that may cause cancer also, right? There are many things. Cancer is not one disease. It is a combination of disease. I always, in high school student, it is a combination of disease. This may be your lifestyle. Smoking cigarette, why people get cancer? There is no cancer in smoke, right? So it is a combination of disease.
So one more question.
Yeah.
So if bacterial resistance develop inside the body.
Mhm.
So what can we do to treat the?
You have to start using, I'm not a physician, so don't take my word as a physician. You have to go to doctor and you ask, he will determine whether it is resistant or not. So they will give you some antibody, it will not work, they will give you another antibody, then combination of antibody, okay? And because mechanism for the treatment of bacteria are already determined, gram negative, gram positive, this antibody. Hmm? Uh-huh. Yeah, yes, that's what I said, that you have to go to, and then they will tell you that which, and they will test a number of antibiotic in culture, and one of them will be working, and they will give you. The most important thing is don't overuse antibiotics. I think one question I'm taking from here. Can I take this one first? He was. That's okay. I'm close by. Don't worry.
He's very soft.
So one antibody drug conjugate is feasible for only one cancer cell, or will it target entire cancer cells? So
No,
how we will know?
Yeah, so I already explained that the cancer cell produces specific antigens, okay, which is not available for other cells. So all other cells then don't express that antigen. Just give you example, CD19 or CD8, these are the antigens, okay. So they are on certain cancer cells. I don't remember which are on breast cancer, which are colon cancer. The expert, colon cancer expert is here, so you can ask him. You'll hear. So these antigens are expressed only on cancer cell, and the antibodies which you are making is going to go to only that cancer cell. But it is not easy. It is not just a way traffic. It has to go through whole circulatory system. So antibody drug conjugate is made in a way that it reaches to target. So it is, you heard about the missiles, right? These are laser guided missiles. It goes to the right there where it is needed.
Okay.
Okay. Yes. Uh-huh. Yeah.
So I wanted to ask is what's the, because antibodies, they have CDR sites, so like antigen specificity is there.
Yes.
So if we are using mouse based antibody, how are we targeting it in humans, like for drug delivery?
So I at the end I said that for development of drug, we start with the mouse antibody. But at the last slide, I said that we have to go for humanized antibody. Okay, so we don't use mouse antibody for human, but in some cases we use
So it's purely for study purposes.
for the development of
Development, yeah.
drug. Yeah, for the development purpose.
Sir,
Okay, where?
sir, I want to ask, what if PROTAC binds to an off-target protein?
It is possible.
So, sir, how will one recover from it?
So in case of PROTAC, how we develop that the protein is known, okay, and there is a binding moiety, what we call it, thalidomide is the most important. I have given the name there, CRBN, VHL, okay. So these are the binding thing which binds to the BAT protein, okay. Now this is, most of the things are initially done in vitro. We find out that, okay, in vitro it binds, what binding moiety we use, okay. So that VHL or CRBN is important. So when we put that CRBN or VHL on the drug, okay, and then it goes to the target protein, and we have given TOI, means target of interest protein, okay. So sometime, or it is our body, different kind of enzyme, the drug can be released before. So it is chemist's job to find the right linker, right bond, so that it can go to the target protein only. And then there is a delivery way also. It is not like that you take only pill. It can be given directly into the cancer. Most of thing is working on cancer. So there are many ways to it. Is like engineering. It depends if you have a handle on the drug, because chemistry has to work, okay? And we need something on a toxic drug, we need something which can bind to the linker to the antibody. So there are drugs, I know one or two, they don't have any handle. Handle means a nitrogen, hydrogen, oxygen, which should be open and which can be chemically bind to a linker to a antibody through maleimide. Okay.
So are there any such cases when we find that drug is very much effective, potent, but we're not able to conjugate with that antibodies?
It is possible. Means this is also engineering. Buildings fail, so antibody the conjugate also fail. Okay. One is here.
We can use a single drug for
Antibody drug conjugate to
yes,
make antibody drug conjugate.
for treating all the, problems. We can use one drug only.
No, I didn't say that. I said there is no. I used the word there is no magic drug.
Yes.
There is no magic bullet.
Yes, sir. But my question is it possible?
And just 3 of my sci paper, we are putting 2 different drugs on.
And then we target a tumor, and then we sensitize that molecule to generate ROS and the further deletion of the cancer. Is it possible?
It is possible, and it is happening also. Photodynamic therapy is well known, and people are doing that. Now, your question is whether we can put that therapy on antibody. Yes, we can do that. See, but you have to think about antibody is a very fragile molecule. It is a protein. You have to know the real chemistry to put the this photodynamic therapy so that it does not affect the function of antibody. Antibody function is only a carrier, drug delivery port. Okay, so how long change? There are many things you have to do that. In some of the drug antibody drug molecule, use a very long linker because the drug was affecting antibody function. So you have to keep away from that, but it is going still. It is bind to antibody. So we have to keep it away from antibody so that it does not affect the antibody function. In some cases, we put a very small linker. That works also. Similarly, not only that, we are putting now radioactive molecule also on the antibody, and that is also working. So there are many, if you look at the clinical trial, there are many things are there. I think I took a really long time.
For the sake of time, we'll move on to the next speaker.
Thank you, sir.
Okay.
Dr.
I'm here, you guys can come and talk.
Yes,
okay?
use the lunch time or the other free times to interact. Dr. Mohammed Zainuddin, we have our industry expert. So he has been working in Jubilant Therapeutics, and he has got a vast and deep expertise in the drug development process. So you'll have a perspective from the industry. We have been seeing things, how it happens in the lab, in a university, in academia, but now you'll see what are the possibilities through the industry. And if he allows, you can interrupt during the lecture. Am I right? Yeah. You can be interrupted during the lecture for the questions. So you don't have to hold up even, wherever you find. Okay, while you are dressing up, we'll... Okay, he's ready. Go ahead. That's fine. I mean, yeah, tomorrow we also have a dinner, so make sure that attendance is full. Okay? So, yes. So we need to make some stimulants. And the location for the dinner is in the Polytechnic Auditorium where we had the inauguration ceremony.
Hello. Yeah, am I audible? Yeah, and so the foundation has been already laid to you guys. You know now the basics. You know how things are in drug. It's one of the complicated things, and I'm really happy to see you guys, especially the questionnaire you guys are asking. See, it's a long way to go ahead, okay? The best part is an idea. And whoever thought, other day we were talking, myself, Azavai and all, that whoever thought that you will have a driverless car, you know. Whoever thought that, I remember in our younger days when we used to sleep in blanket and all, and somebody out, my sibling has to go and jump up and switch off the light. We used to fight who will go outside and do that. We used to think if we can clap or ask them, the lights would be switched. Now it's there. Okay, so it's more of an idea, and I really like your question, Budding. Don't restrict yourself. Whatever you think, write it down. That's the basic thing. Write it down. Then think of operation level. We will talk. I am not academician. I may not be a good teacher. I will try to tell you all that what we do in the industry and what are the opportunities. Similarly, you have seen here, it's a versatile science we are talking. People took 20 years, they perfect themselves in one field. So don't think that you can learn all, but only thing is you pick up the thing which you want to know about it and your expertise lies. That should be the intention. I'm sure it will be very difficult to do the job. That's what, sir is a chemist by training, but you have seen him, he was talking biology on the same plant, he was talking of drug metabolism on the same plant, and that's the key which you can do over the period of time. Okay? So the interdisciplinary topic is basically the foundation of it, and you all have to contact, you have an idea with the chemistry, you have to go to the biology people and say, I have an idea, is it possible you can do this assay? Then you can't do the pharmacology and say, this is what we want to do, is it possible we can do? I have an idea. So that's how we have to do it, okay? So let me give you some perspective from industrial aspect, and this is something which is beyond textbook, and decision, trade-offs and real world stories. That's what I am going to tell you. I want to think of it, I want to tell you all, this will be more of a, I want to make you guys engage, I don't want to be a boring teacher here, but I am not a teacher here. So I just want to tell you some stories which may help you in future to go to the next level. So before that, people can read it. Is it possible you can read it? I remember a photograph of it, and I will also read for you guys. This was a letter written to Railway Department, and it was on 2nd of July 1909. And he wrote, "Respected sir, I am arrived to a passenger train." Don't go for English, okay? And don't go for English, but it was written. It was one of the landmark letters. Still this photo is there at that exhibition, which is the railway exhibition hall, okay? What it does, the washroom he was not having in those days, so he wrote all English and threw it. This was the letter. What it does, this has a historical way, just to think whether my English, so it's but it can click also, and you become a billionaire, no doubt, okay? So I just want to recap of the previous talk which we were having. You know, he already told you about the drug stages, how it is, academic versus industry views also. We will also be touching down to the line this. AI driven drug design, already you have seen some biology about it, how the biology works, case studies and industry challenge, and now you have already field which he was telling, a PROTAC and ADCs are the next gen. This is the demand, although we don't have a drug of a PROTAC. ARV-471 will be coming soon. Probably it's a phase 3 molecule. Let us see how it works. It's an estrogen receptor binder. If it works, then we are done. Then this science will float like this. So whoever is interested, read more of a PROTAC, think of one angle, and industry is looking for those people who have new novel ideas to work on it. Okay? I will show you a story. This is Eli Lilly company. I don't know how many people you know this company. This is the company. This is started with a small room that was 18 by 40 feet. Okay? And this was just a small lab, and you can see this is a historical photo with the two people. One is the founder of this company and his son, both are standing there. And now I'm giving a date of 21st November 2025, not much long, it's around 3 days back. Okay? It became a 1 trillion market cap company. First company. So you all start in one room, people started in garage, they went and became a billionaire. Many stories are there if you read it. So just to give you a recap how industry and an idea can be float. Another from the science perspective, this is a historical photograph. In 1901, Dr. Alan Warner of Leicester has given a lecture, and he's shown two photographs. These two kids were dosed with a vaccine on the same day. Down to the line, one was dosed, another was not dosed. Both were infected with smallpox. You can see the photo. And that photo has really changed the concept of accepting the vaccination. So you all have seen these are the main stages of drug discovery and development. And more importantly, if you see the average cost, we all have heard this, $2.8 billion is spent to bring one drug to the market. And there is a lot of emotions we have not counted, we have not paid for that. Emotions, frustration, okay, and there is a lot of stuff goes on. And it takes around 12 to 15 years from discovery to the market. And then there is around only 12% of a drug entering a clinical trial get an FDA approval. These are the stages. You already seen stages. There is a pre-discovery stage, there is a drug discovery and a preclinical, a clinical trials, review and approval. These are all long process which sir has already covered with these. And if you see, feel free to ask me questions, okay? Shoot the question today, because don't keep the question. Discussion is the part of it. We come from a culture, we stop our CEO and ask the questions, okay? So we don't have that culture. Let's ask the question if you have, don't keep it. So just to bring you the timeline from a discovery stage, when we talk of a discovery, means we are talking of any drug coming to the lead. What is lead? Sir already told you that once you have an idea, you make a molecule. First you decide what the molecule we are going to make. I will be covering some of the portion in that. So at industry what we do, the best part in my office or in my lab where we stay is that we have a one roof. And in all one roof, we start from a lower, there is a structural biology lab, there is an NMR lab, then the next floor which is a biology lab, there is another lab which is the DMPK, third floor is full floor of a chemistry, fourth floor is full floor of a chemistry, and the fifth floor of physicochemical studies. Okay? And the ground floor we have a animal house which is around, where we keep around 10,000 vivarium we have. Okay? So this is how our lab construct. So what is the message I want to tell you? In my lab or in my office, we all work together in one roof. Our coffees machines are there in each floors. We come out for a coffee, and in that coffee, it's only a small teams which work in a program. They all come and say, we have made a molecule. इतने मोलर का मॉलिक्यूल आया, उसमें स्टेबिलिटी नहीं है, देन द अदर गाय विल टेल, ओ यार तुम्हारा मॉलिक्यूल फेल हो गया, दूसरा बनाओ, इसकी एक्टिविटी ठीक नहीं आ रही है. So this is how we discuss, and people go back and do it. What is more important is a cross-functional talks. Okay? So this is the discovery. When you think of a molecule, you make a molecule, then you go to the molecule and see how much is there. You start optimizing it. If the first compound is a hit, which is the micromolar activity usually we take, if you are lucky enough you have a nanomolar, then you go to the next stage, third stage, and that the molecule you want to check it. Then you have a preclinical. That's a 2 to 3 years. It's around $100 million where you go for the animal studies, and then you take of the animal studies, you do the animal studies and see how much. See, there's a huge difference from test tube to animal. So when you have a test tube, when you are doing a study in a test tube, other day there was a question being asked. First day I was writing all those questions, and that was a question which was that how the model you will be doing. See, I can give you one example. Model is the job. Model means exceptions, assumptions. When you make a model, you assume anything. Okay? So similarly, at a lower level, when you do a cell line, you assume that this is the targeted cell line, but you don't know the influence of other things or environmental, structural, everything influence you don't know. Similarly on animal, you assume it will be working, but there will be a subtle change when you go to the human being. Okay, this is what the story is. See, when you saw that lot of money went, okay, but why people will spend so much money? Why don't they do it? This is a 8 billion. There are a lot of failure. There is a success of only 10%. Why should we do it? The only answer is this. See, this is the what? High risk, high reward. Why blockbuster drug drive pharma investment? This is, I will give you example of Keytruda.
For example, in the last week, the statistical data, how much drug passed from the stage 2 in your lab?
See, in my lab, stage 2, I should say, is the lead molecule. Lead molecule is 80%. Yeah. Failure comes once you when you move to the candidate level. And when you go to the phase level, it becomes around 20%.
The number can you give us?
20%, 20% means 100 molecule you put 20% phase 1, you get some MTD, but you don't get efficacy. You go to the phase 10, it's a rub off, you get only 10 molecule out of 100, and then you move forward, commercialization, then there is a issue with the synthesis. He was talking of 10 mg, 20 mg, 30 mg, you have to make that day he was showing you a slide where you have to make a scale up, you are talking of at least a metric ton of molecule you want to make it. So that's scalability is a challenge. So those challenge comes and draw it. So it remains around 10% with all best successful rate.
Sir, my question was more specific. Can you give us the actual data, sir, for last year, how much?
I can give my lab data, but usually it's a 60 to 80. I can give you a range, that's all. 80% is the range, which it's a very successful. That's not a problem. But only thing is, sometimes you take it in around 1-2 months, sometimes it takes around 1 year, sometimes you are struggling with a molecule, you can't do it. You tell computational chemist, oh, you make this molecule, synthetically is not viable. When you make it, then it is not hitting the target. Then you do a biology, you do a testing, then you make another arm. To do it, you add hydroxymethyl, you add some arms, you try to make it, then you send to a molecule, it is not soluble. You go back and try to make it soluble, because until you don't have a solubility, you will not make a solution, and the solution will not do an assay. So you will have a false positive data, then you don't have a stability. The moment it goes to the cell line, it is get unstable, you don't get a data there. You don't have a permeability, then you struggle with the permeability. And solubility, permeability is the different, both arms, solubility you need a hydrophilic compound, and permeability you need a hydrophobic compound, lipid loving compound you want it. So you want that it should remove the cell membrane. How it will go to the cell membrane, that you have to make a balance of it. Until you don't have a solubility, you can't formulate, you can't give to animal. If you don't have a permeability, it will not go to the target and work from that. So this is the chemist game, Chemist struggles a lot. They are the smartest people there. They sit together, they try to do it, but we are the killer. We give a lot of frustration to them. Okay? Yeah. Okay, this is what the reason for investment. This is some of the target which you need a number, and these are the number which you would want to say. These are the per year drug with there. You might have been listening, seeing the word first in class. Okay, I will be talking to you what is first in class, what is best in class, what is how we do it, and why we Me Too drug. All these language you will see, and why the company moved to that level. So first question is how industry strategically chooses the target. Okay, I was struggling actually. That was the reason I was sitting here listening, sir, that I should not make a presentation which should be redundant. So I tried to make something which you guys, which we can add value to the whole process. Okay, so what is the first in class? First in class is nothing. It's a new mechanism which we are talking. New mechanism, new target. No drug is there in a market. Okay, and then you try to do it. It's the toughest one. The driver is the unmet disease. You have seen, have you story of EGFR mutant and all those, even a Keytruda molecule, PD1, PDL1, have you seen a Nobel laureate got this checkpoint inhibitors, they are said now. So read out who the biochemistry people, read out the checkpoint inhibitor. This is something people are talking. There is a small molecule coming, and we have a drug. We have a molecule at the candidate level, which is a small molecule of PD1, PDL1 inhibitor. Okay, so we are proceeding it. So let's see how it works on. So high unmet need, strong biology, and a validated human genetics. Can anybody tell what is the validated human genetics? Probably now you will be knowing about it. Something you know about a target which you heard. His morning also he was telling, he repeatedly told twice to you guys, there is something, a validated genetic target. Means the target which is viable, target which is there. You think of lot of targets. That's what the idea I was telling. You float the idea, but it's your job to see whether the idea can be conceived as a actual. That's your job. Okay, you can't throw an idea and move on. You have to read out, you have to see POCs. I will be using that word POCs, proof of concept. Any human trial, anything you have done with respect to that, any proof which we have done. So that's the reason why we want to. Risk is biology unclear. See, I remember, I don't know how many people are there who are the ferocious reader. I used to be, and not now. That is a fiction reader. There was Isaac Asimov, a writer, is a Russian writer. He used to write science fiction. And what he says the protein is nothing. It's with the warm of the hand, with the shake of a hand, and even with the stare, with the glare, it changes the structure. And if the block is made of that, which is known as the protein, how you expect a human being to be stable? That's one of the beautiful, he wrote pH and all those things, that something subtle change in a protein, it changes, it disrupts, it disrupts structure. And that the whole protein body is made of that building block. How you expect that building block will a human being is, that's why it's such a unstable aspect, you know. Okay. Yeah, I love that word actually. And unpredictable talks, that's the biggest challenge I will be covering in the next lecture. I will be more focusing on DMPK and talks next lecture. Industry example. So, but there is a high risk. If we don't do this first in class, we all will be in a, you can go to Jubilant therapeutic side. We have a molecule which is a LSD HDAC inhibitor. That's a first in class molecule. We are in a phase 2, probably phase 3. We are going to get it very soon. Everything in India also we have started the trial. PRMT5 is also a first in class. We have a target, but there is a lot of people who have failed. Let us see where we will sink or we will swim. Okay, these are the molecules. See Keytruda. Now, how much the life is saved by Keytruda. Merck took early risk in BMS, explode CTL4, and these are the examples. So first in class is nobody has done. You are starting the first thing. And that is what is strategical. We take 5 decisions, 5 program we have to run. 4, we have to take a small risk. We want to do a fast follower program where the people have done. they have a PSC, they have a phase 1 molecule. So we start, chemists are the smartest people in the world, you know. They start doing they do some patent busting and all. I don't want to, they start doing it, they make a small molecule, they read the patent nicely, where they have left, what is there, is the chlorine added or not, if we use a chlorine, how much it is there. So that is, and they are also smart, they try their best to make sure that the dibba is so close, nobody can enter it. So this is how the fast follower program works, and usually fast follower programs succeed like something. Okay, because you know the proof of concept, you know the history, how things are, you are not living in a black hole and your drug is moving out. So that is known as I will go to the next slide. This is the first in class, understood? It's a very high risk but high reward. Once you bring it in a first year, you will take all money what you have spent so far. Depend the patient population you have. Then you have a best in class, or me too, why they still matter. Best in class is something you have a lot of drug. Already 2-3 drugs are there, but there is some issue there. You want to make it and say, see, no Indian company is doing any drug discovery, I am telling you, or development. They are all doing different things. They are doing a copycat, they do a formulation, and there also you can say they formulate in such a way that exposure is nice, or it should not be higher, but their effect is better. So they changed in the formulation. That's more of a generic company. All big companies are doing it. Discovery as such, 3 companies are doing it. All of them is still, I don't see. One Cadila has brought one molecule, and others are still working on it. So that's a phase. Okay. People do in best in class, goal improve to the weakness of FIC. So that's where the chemist comes into play, the biologist comes into play. Biologist say, यार उनमें ये issues है, उनका molecule देखा था, उसकी selectivity नहीं है. एक होता है स्पेसिफिसिटी, एक होता है सेलेक्टिविटी. स्पेसिफिसिटी जिस टारगेट को हिट, जैसे हमने यहाँ से मारना है, जो बता रहे थे आज एडीसी के बारे में, वो स्पेसिफिसिटी का बहुत ध्यान है. जिस सेट से उसने मारा, बगल वाले को ही एक लगेगी, आपको नहीं लगेगी. मगर उस प्रोसेस में जाके वो पीछे वाले को भी लग रही है या कहीं और लग रही है, तो सेलेक्टिविटी भी इशू है. तो ये एक बहुत इंपॉर्टेंट है बायोलॉजी समझना. और बायोलॉजी के बाद उस मॉलिक्यूल को जब बनाते हुए, तो इट्स बॉडी इज मोर ऑफ ए, it's more of a cascade, you know the cascade क्या होता है. हम लोग जब सेल सिग्नलिंग पढ़ते थे तो उसमें यही कहते थे कि सेल सिग्नलिंग जो है इट्स नथिंग, उसकी एक सबसे बड़ी चीज़ है कि ऊपर से कोई चीज़ छोड़ा और उसकी स्ट्रेंथ क्या है, वो है एम्प्लीफिकेशन. तो नीचे से ऊपर से एक पत्थर पढ़ाते थे टीचर, बहुत अच्छे थे, वो कहते थे कि ऊपर जब जाओ एक पत्थर फेंक दो पहाड़ से, और जब नीचे आओगे तो 150 पत्थर नीचे लेके आएगा. दैट्स हाउ द एम्प्लीफिकेशन वर्क्स. ओके, एंड दैट्स हाउ द एसेंस ऑफ cell signaling. This is what we are tagging. When you put a target, there's a lot of kinase target and all those targets. When you put it, it's down to the line. When you go, its effect is amplified many times. And that is what he will be talking more on a downstream, upstream, and that is where the key comes, and that is why the KRAS from a biology side became undruggable for many years. Okay, and this is another is a Me Too, where you come in the similar line. I will give you 2 examples. Ozempic is one of the improved duration of efficacy. That's all came, and there where the Eli Lilly has become such a giant here, became a BIC for the targeting EGFR. This was the drug. So we have out-licensed this drug, EGFR mutant drug, to one of the Blueprint with $250 million upfront, $250 million later. And what they did, they took our molecule and put it on hold, and they are saying it's a strategic reason. They paid $450 million for this molecule, EGFR mutant, which AstraZeneca made a lot of money in it. See, AstraZeneca, with this erlotinib, when they were working, they make a suffered resistant mutation, and AstraZeneca target the resistant, became a $5 billion per year blockbuster. They earned it. And already the EGFR mutant was there in molecule. They went and they thought that it's a mutant molecule which is not working. They went and tweaked the molecule with a mutant, and then they got it, and things fly. And AstraZeneca faced change, There is a fast follower strategy that I was telling, that is the industry playbook, and Nadbhai will laugh on it. once we ask them, give option to our medicinal chemist कि ये 3 टारगेट है, इसका कुछ नहीं पता मुझे, इसका पता है इसके बारे में, तो कहते हैं तो फास्फोवालर दे दो, हम तुम्हें दे देंगे मॉलिक्यूल, तुम इसका टेंशन ना लो, we will give you. Okay, and then there is a challenge there. Then you have a fast followers are done, laser focus, you have SLG tube, FIC, डापा जो है अब देखो यहाँ पे बिक रहा है, डापा सिर्फ 30 रुपए की एक टैबलेट मिलती है, अब मिलने लगी है 11 रुपए की टैबलेट. उससे पहले एक और टैबलेट आती थी, दैट इज स्टिल आईपी उसका नहीं है, BMS का मॉलिक्यूल है, और वो बिक रहा है 57 रुपीज का. So that's how the fast follower help for India and third gen countries. These are something very important. We need a cheap drug and cheap and effective. So we don't have a money to spend a lot of things. We don't have a license. We don't have a much of insurance. So these are the important things which we say in the meeting. Now I will tell you some problem and challenges that what we usually face when we do a drug discovery. Animal model limitation, that's the biggest thing. Other day somebody was telling about dementia, they developed a model, they are working on a model of a dementia. I was really interested to know it. Usually CNS model are tough model, okay, and it's not easy to have the CNS model, and usually 90% program fail due to non-relevant models which we have. Even in a cancer, we can't develop a model, you know. What do you do? How we develop model, I will give you an example, because these are my expertise, I can tell you. We take a cell line, we inject the cell line, and that it was saying a SID molecule or something immunocompromised drug, which we take it, and then you develop, there is a development of a tumor. Why we do it? Why we take a SID or we want take immune compromise? Anybody can tell? I want to check your immunology, please. I given a hint also with using a word immunology. Yeah, you already told. That is important because you don't want anything to be combated by your immunological system, okay? So if you want to give something foreign, there will be antibody response there, so you don't want it. So that's why I'm telling you a small thing. You pick one thing. Do it in your research. When I go for a PhD interview, I have 3 postdoc working under me. They are the manager level, and I'm telling you, we don't want what they do. We just want today that what they learned, how much technique they want, and they know it. And that's the way we take the people, okay? So I want you guys to be perfect with technique. Spend money to get techniques. Go to the good labs and ask for training, ask for something which industry is now becoming restricted for these things. They take 2 years bond and they give you a training and give you a job, but people want to do PhD, they don't usually take this. So these are the real world R&D constraint, timeline, cost, burn rate in biotech, quality, IPR I will be telling, and first in human readiness requirement. The biggest cruel part is our regulatory bodies, okay? They give a lot of, so sir was telling yesterday that there was a truck going on for one of the drug they wanted, and there was a submission in those days. I can salute to the people who were working this day, that how much the handwritten stuff was there they used to put. It's still, a saying goes, I have put one in my slide, a saying goes, you need a village to file an IND, okay? So for filing an IND, you need a village, means everything will be, it's a huge aspect. So don't think, once you take a pill, I can assure you, if you learn anything or you don't learn anything, you understood anything or you don't understood, at least you start respecting the people. When you take a pill, you will know this pill has not come easily. It took 15 years, any pill. And one more thing which somebody has asked for the AMR, stop self-medication, stop going to jhola chap doctors, okay? Go to a doctor who do some rational practice. He should start a stage was, otherwise a time will come, you will be sitting on laying on a bed and you will not having an antibiotic to work on it. They give you a steroid and all, please don't do that. Please don't do that. This is the best thing. Don't सेल्फ मेडिकेशन, गला दुख रहा है, कोई कह रहा है एजिथ्रोमाइसिन ले ले यार ठीक हो जाओगे. Don't do that. You don't need it. It's a viral. Viral doesn't need any antibiotic. Okay? 90% ये हाल है. Please don't do that. And that's why the migraine has come. Your stop sensitivity has stopped. So AMR की सबसे बड़ी चीज़ है, कुछ फिक्र ना करो, दवा खाना बंद कर दो. Until unless, last thing you should do is take medicine. Okay, let me talk. Yesterday there was a valley of death. I am talking of some of the valley of death what we are there. The target to the approval is a valley of death, and you see the basic research 50%. For your question, I think there will be some of the thing. 1 year you take for target identification, 1 to 2 years, and 6 to 7 years for it's all changes sometimes, a lot of things. There is a vicious cycle which comes from target identification to preclinic. It goes like this. Chemists get frustrated sometimes they make a lot of molecule, and then all of the sudden we say we don't want to work on it, and this is how it is. But industrial mandate is, this is what the success. We will discuss crossing this value where the industry comes into play. What are the reasons? Reasons are better genetically validated target. Important is we should have a genetically validated target. If we don't have, we will not do anything. If you want to take any cancer is something which is burning now these days, and sometime you start working, end of the day after some time you come to know, hey yaar, problem. You remember AIDS used to do one of the issues, you know, and then I was taught by one of the guy, he was a top shot, he used to teach us microbiology, and he used to say, then इसको अगर थोड़ा छोड़ दिया जाएगा ना तो ये इतना खुद ही म्यूटेट हो रहा है, ये ख़त्म हो जाएगा इसका विशियस साइकिल. ओके, तो बॉडी हमेशा म्यूटेट करती है, बॉडी स्मार्ट है, अपने को चेंज करती रहती है. तो हम जब चेंज की तरफ जाते हैं तो वो ख़त्म हो जाता है उसकी. कभी कभी लिथैलिटी बढ़ जाती है, कभी लिथैलिटी कम भी हो जाती है. तो वैलिडेटेड टारगेट अपने पास होना चाहिए, डेवलपमेंट पैराडाइम होना चाहिए, रोबस्ट बायोमार्कर. सबसे बड़ा इशू आजकल जो है हम लोग को, हम जो आजकल क्लिनिकल ट्रायल में देख रहे हैं कि रेलेवेंट बायोमार्कर नहीं है हमारे पास. हम आदमी से अंदर तो हर बार पूछ नहीं सकते, हम चाहते हैं कि प्लाज्मा निकला है ब्लड. ये बायोमार्कर की फील्ड भी बहुत अच्छी है. अगर कोई तुम्हारे यहाँ रिसर्च कर रहे हो तुम लोग यहाँ पे, मैं तो चाहता हूँ छोटा छोटा काम करना शुरू कर दो. छोटा काम, बड़े काम. अंग्रेजी मैं, आई एम नॉट ए फैन ऑफ जिसको कहते हैं ना कि बहुत हाई फाई ना, हाई फाई का शौक नहीं है. छोटा काम करो, छोटा बहुत, इसमें से एक हिस्सा ले लो और उसको एक परफेक्ट, एक ट्रांसपोर्टर पे काम कर डालो. देखो कहाँ से कहाँ लोग पूछने आएंगे. हैं, बड़ा काम से कोई नहीं जिस पे जो है आदमी कहे ये लंबा चौड़ा अपने थीसिस हो गई. नो, आई वांट ए स्मॉल टॉपिक. बायोमार्कर, कोई भी बायोमार्कर ले लो. एक बायोमार्कर भी अगर आपने, हम जैसे LSD एचडैक पे कर रहे हैं, अल्फा ट्यूबुलिन एक ऐसे है, उस पे एचडैक की एक्टिविटी मिलती है, अल्फा ट्यूबुलिन ऐसे को डेवलप नहीं कर पा रहे हैं. दुनिया की सब लैब देख ली, $1.5 मिलियन खर्च कर लिया, अभी तक हम लोग डेवलप नहीं कर पाए हैं. मैं इस पे बात करूंगा कि किस तरह से हम लोग और कर सकते. देयर इज अ 3-headed beast, पीपल से, efficacy, safety and development. Sir already told you efficacy. Safety we will be talking, and develop. In industry 95% pure isn't good enough. ये सुन लो, this is how it's important, you know. तुम लैब में केमिस्ट जो होंगे वो सोचते होंगे यार ये पीक आ गया, ये हमने TLC रन कर लिया, ये आ गया थोड़ा सा और हमने इसको दे दिया. नहीं, 99.99 kg की प्योरिटी चाहिए. सर ज़्यादा एक्सप्लेन करेंगे इस पे, और मगर 50 किलो चाहिए होगा. अब सोचो, उसकी स्केलेबिलिटी ही टफ है, और वो आपको उसी दिन प्लान करना है जिस दिन आपने मॉलिक्यूल बनाया कि इसकी सिंथेटिक हम आगे कर पाएंगे कि नहीं. जबकि दैट्स अ डिफरेंट बीस्ट, GMP का काम. हाँ, इसके साथ मैंने काम किया है, सो आई नो दिस फेलो. ही इज ए गाय. कितने लोग जानते होंगे जो एटोरवेस्टेटिन खाते होंगे, लिपिड की जिसको प्रॉब्लम होगी, घरों में लोग खा रहे होंगे, ज़्यादातर लोग खाते हैं. इंडिया में ख़ास तौर से जहाँ हम लोग जो है फ्राइड सामान खाते हैं, तो ओवर द पीरियड कोलेस्ट्रॉल हाई हो गया, ये हो गया, वो हो गया. तो एक ही रामबाण है जिसको लोग कहते हैं, वो है एटोरवेस्टेटिन यूज़ होता है. ये आदमी ने एटोरवेस्टेटिन दिया था. हैं, और इसने फाइजर को दिया था एटॉर्वेस्टेटिन, जो कि एटॉर्वेस्टेटिन बाद में चल के 10 बिलियन की सेल सिर्फ इसकी जो है लॉन्च के वक्त से लेकर 2010 तक हुई, जबकि इसके पास सिर्फ 14 साल का पेटेंट रह गया था। और ये एक मी टू ड्रग था। क्या था? मी टू ड्रग। आई एम लकी आई हैव वर्कड विद दिस गाय विद अ डिफरेंट प्रोग्राम, बट दिस इज हाउ ए रैग बिकम्स अ रिचेस की स्टोरी है। Viagra, you all know well this drug and how serendipitous discovery एक angina pain के लिए लिया गया और उससे erectile dysfunction को उसको हेल्प करता था। उसके बाद उसको एंगल को स्विच किया गया, मेल पेशेंट रिटर्न द पिल, और उसके बाद जब उन्होंने ट्रायल दिया तो देखा कि मेल जो पेशेंट थे उन्होंने वो वापस नहीं करते थे वो पिल्स को, तो उन्होंने उस पर जाकर मालूमात की एंड दे वेंट टू दैट and it became a multi-billion-dollar drug. So this is more of a best example of how serendipity works. How you know guys know serendipity? Accidental discovery, okay? But this was not accidental. There was a whole research went through it. They also changed the formulation. They also changed many of those stuff which were affecting during the whole process. ये KRAS की स्टोरी है। मैं बहुत मेरे पास भी KRAS का मॉलिक्यूल है, G12D का सर मॉलिक्यूल एक है, एंड वी आर ऑलरेडी डेवलपिंग इट, दैट्स ए पैन KRAS। एमजेन ने जब इसको मार्केट में लाया तो समझो एमजेन का स्टॉक, एमजेन ने जिस दिन अनाउंस कर दिया कि हमारे पास पॉजिटिव KRAS G12C का डाटा है, उसने उसी वक्त जो है $12 बिलियन कमा लिया इस स्टॉक से। ये रिवॉल्यूशन था। अनड्रगेबल टारगेट जो आज बात कर रहे थे, ये अनड्रगेबल है, क्योंकि इससे 40% आपका डाउन स्ट्रीम जितने भी 40% जो है वो इसमें इफेक्ट होता है। 40% कैंसर इससे इफेक्ट हो जाता है अगर इसको ब्लॉक कर लिया तो। मगर बाद में देखा गया कि G12C के बारे में जब हमने, अब हम इतना नहीं जाना चाहेंगे तुम लोग को, बहुत अच्छी इंटरेस्टिंग चीज़ है, स्टोरी है, हाउ ए स्विच टू रीजन, सर नोज मोर ऑन दैट। हम केमिस्ट से लड़ते ही रहे हमेशा, DFG इन DFG आउट पे ज़िंदगी भर हैं। तो जितने लोग यहाँ कॉम्पिटिशनल का कोई है, यहाँ पे कॉम्पिटिशनल केमिस्ट्री बायोइन्फॉर्मेटिक्स का कोई है? हाँ, तुम लोग के लिए बहुत ये एक लैंडमार्क स्टोरी है जहाँ उन्होंने हेल्प किया कि हाउ स्विच टू रीजन को उन्होंने इनएक्टिव KRAS को उन्होंने काम किया एंड दे बिकेम ए डेवलपमेंट ड्रग। तो उसके बाद क्लिनिकल माइलस्टोन हुआ, 37% रेस्पॉन्स रेट हुआ, $1.27 बिलियन इन 2024 को उसने कमा लिया। 3.73 उनका यह है कि 2033 तक कमाएंगे। मगर प्रॉब्लम क्या है, जब उन्होंने दूसरी बार रिसर्च की, मालूमात किया, एंड दे व्हाट दे फाउंड, द ड्रग वाज रेजिस्टेंस G12C. Then they started working on G12D. Amgen was having, Mirati was having, this is the way we remember actually drug. Mirati was having, and then AstraZeneca brought a very good compound, and they are doing a phase 3 clinical trial, and that is the G12D pan kinase. Now we need a pan kinase. The life is pan kinase. G12D is the best one which have a lesser resistance so far. People are working and developing it, and if it comes, probably हम कैंसर को बहुत जल्दी कॉम्बैट कर लेंगे unless something new comes up. Okay? That what I said. Cells is, the human is smart, means cells are also smart. Okay, and what it does, it changes itself with respect. They see what is happening. Cancer के लिए best example मैं देता हूँ. Azhar bhai will be mocking you. That it's a retrospective analysis of eukaryotic to prokaryotic. If you really want to know about cancer, read prokaryotic cells. It's nothing. Many of them not affecting your religious thing, but being a science student, we talk of evolution. And when we say that single cell, multi cell, और ये कुछ नहीं है, ये reverse हुआ. यूकैरियोटिक से प्रोकैरियोटिक में चला गया। इसको अगर प्रोकैरियोटिक के सेल को पूरा समझ लोगे, आपने कैंसर सेल को अच्छा खासा समझ लोगे। एंड दैट्स हाउ इट इज। समटाइम्स यू डोंट रिमेंबर प्रोकैरियोट क्या हो रहा है, कैंसर सेल कैसा होता है, आप प्रोकैरियोट के बारे में लिख डालो, अब बहुत कुछ मैच कर जाएगा। दिस इज वन ऑफ द बेस्ट स्टोरी ऑफ PD1। आई वॉज ऑलरेडी टेलिंग यू कि कैट्रूडा ने काम किया, PD1 इनहिबिटर, आज नोबेल लॉरिएट भी है इस पे, जिसने चेक पॉइंट इनहिबिटर्स कहते हैं इनको, और इसने रिवोल्यूट कर दिया है पूरी ज़िंदगी। आज जो सब लोग इंडिया में नहीं है आता है वो, स्टिल वी डोंट हैव एनीथिंग। सो बांग्लादेश पीपल ब्रिंग्स फ्रॉम बांग्लादेश दैट, एंड बांग्लादेश हाँ, और मैं क्लिनिकल ट्रायल कर रहा हूँ, मर्क ने हमें दे दिया है मॉलिक्यूल, हम LSD HDAC के साथ कॉम्बिनेशन स्टडी कर रहे हैं। ना ना ना, कोई नहीं कर पाया अभी तक, कोई किसी के पास नहीं है, असल पेटेंट भी अभी नहीं ख़त्म हुआ है तो दे नहीं पाए वो। This is SGL2 who have the diabetes at home, people who know this, you know well, this was one of the this is the transporter drug which was made, anti-transporter, and that was one of the beyond the glucose provided cardiovascular benefit, and that has really make industrial research confirm the effectiveness. That is what. So a small route I was trying to reach with the, this is a very crowded slide. I wanted to touch base something on your why we inhibit ourselves, why we think academia, what academia should do more. This is more of a quality control. We are talking of more of a stuff where we think why we are IPR protection and all those stuff. So primary goal ही अलग है academia का, उसका काम है publication, हमारा काम है बिल्कुल publication नहीं करना, हमको दिखाना नहीं है कुछ, आपको दिखाना मैंडेट है, हैं? और इसी में एक वे निकाल के कि हम क्या दिखाएं और क्या ना दिखाएं, आधी नॉलेज दिखाएं। पेटेंट भी हम लोग जब पढ़ते हैं तो समझ नहीं पाते हैं उसको मॉलिक्यूल हाउ वी हैव टू मेक इट। सो दिस इज अ वेरी क्राउडेड स्लाइड, यू विल गेट दिस हैंड आउट टू यू गाइज and you read it. That's a very important why we highly research focus, you guys. I salute most of the drugs so far has came from academia. I can tell you 80% drug they budded or seeded in academia, and they all were developed by industry. Okay, so this is something I don't want to get, but more importantly is the take home message. Process is a counterintuitive to the truly innovative scientists. Kill their brains and passion and you kill their science. So this is the right. This was the training was given by Eli Lilly to us. I pulled that slide in 2006, they brought it and they presented it. We were working in CRO, sir. If research and IP process are weak, no solid drivers. So this is just a balance. You take the science curiosity, but put it. That's what the sir told other day. Use of a lab notebook. लैब नोटबुक अगर आज से तुम लोग प्रैक्टिस करते हो अपने लैब नोटबुक की, जिंदगी भर काम आएगी। एक नोटबुक ऐसी होनी चाहिए जो आपकी गर्दन चली जाए वो नोटबुक ना जाए, हैं? अगर आपने वो सीख लिया तो यकीन करो कि आप बहुत ऊपर जाओ। उसमें आपके उड़ते फिरते जो आए वो सब लिख डाल। एक्सपेरिमेंट करते हुए गलत आया तो भी लिखिए, सही आया तो भी लिखिए। Lot of example came where the people failed and they were later realized that was the success. Okay, तो वो आदत डाल दो। लैब में जाओ, हर पॉइंट को लिखो। वो मैंने ऐसे रखा था तो ऐसे घूम गया, वो डाला तो येलोइश कलर हो गया, वो डाला तो ये हो गया। You as a chemist, it's very important. Biologists, they work on SOPs. So sometimes if you get a new thing, it's okay. Otherwise, chemist, मैं समझता हूँ उनके लिए वो। तो neither great science, not great policy, sufficient on its own. Both are necessary and with a success time. I will give you some example. किसनों ने कोडक कैमरा की IPR स्टोरी सुनी है? नहीं सुना है? Oh, come on यार. This is very important story to tell people how IPR can destroy a billion-dollar company. This was a patent war which happened. In 1888 Kodak camera revolutionized, you all know well. आपको पहले भी याद होगा Kodak का लाओ लोग कहते थे. I don't know, you kids and the people who are grown up, they might be knowing about it. Made photography affordable, sparked privacy debate. और उसके बाद क्या हुआ, 76 में पोलारॉइड की एक जो कंपनी थी उसने उसको सू कर दिया. 12 पेटेंट उसने उसके इन्फ्रिंजमेंट में डाल दिए और यूएस कोर्ट ने रूल्ड कर दिया फेवर ऑफ पोलारॉइड. यही पेटेंट की स्टोरी है. कौन करेगा, जो बेचारे ने इतना पैसा इतनी मार्केट खर्च की, ये वो सब तबाह हो गया. 925 मिलियन कोडेक को उसको देना भी पड़ा और उसने वो विड्रॉड इंस्टेंट कैमराज फ्रॉम द मार्केट और कैमरा मार्केट से उसको विड्रॉ भी करना पड़ा. मैं क्यों बोल रहा हूं कि हम इंडस्ट्री के लोग, देखो अभी बात कर रहे थे कि पासवर्ड प्रोटेक्टेड, मैं अपना लैपटॉप लगाता हूं, भेज नहीं पाता हूं कोई चीज़. हम कोशिश रात को बहुत स्ट्रगल कर रहे थे कि हम भेजना चाह रहे थे तो हम नहीं भेज पा रहे थे. तो किसी तरह से मैंने उसमें से बहुत सारी रिड्यूस किया, बहुत सारे स्ट्रक्चर थे जो हटाया हैं. और उसके बाद हमने कहा मैं दिखा लूँगा और उसके बाद हटा, तो कहा there will be something which is going to be, we can't we do it. So there is a confidentiality which is associated, and now the confidentiality in such a way, when I enter in my room or something, if the book is open, I usually put it down. ये अब इनकलकेट हो गया, ये हमारे अंदर हैबिट्स ही आ गई है. अब वो नॉर्मल हो गया मेरे लिए. वो नॉर्मल नहीं कि खुली किताब छोड़ के चले गए, क्यों मुझे लगता है उस पर कुछ लिखा ना हो, कोई टारगेट का नाम ना लिखा हो. तो ये एक और जेनेंटेक जीएचएल की जो जीएचएल जो है वो ताइवान की एक कंपनी थी और इसने भी जो है जेनेंटेक को इन अलीगढ़ वर्ड उसने चूना लगाया बहुत बड़ा. एंड इट हैज़ रियली मेड दैम बहुत. उसके बाद 220-30 उसके लोग चले जाते थे और जूम कॉल करते थे, स्काइप कॉल होती थी और उससे उन्होंने कई बायोसिमिलर्स के बारे में डिटेल्स बता दी. और एक विसिल ब्लोअर एक टर्म होता है इंडस्ट्री में. विसिल ब्लोअर क्या होता है, जैसे आप लोग 4 लोग बैठे हैं एंड आउट ऑफ 4 1 पर्सन कम्स अप एंड सेज they all were mangled when things were going out. One of the person either his उसकी जो है ज़मीर जाग जाता है एंड ही से यार मैं, तो he becomes a whistleblower and the company leave it and the organization doesn't catch him for those instances because he is a whistleblower, and then the things go. Similarly, this story went, and it's one of the beautiful story which happened, and then in 2022 there was a sentencing happened to many of those people. मगर ये टाइम देखो इतना 2013 से टाइम, मतलब 9 साल में, एक साल में 2.4 बिलियन जो कंपनी बना रही हो और उसको इतने साल में इतना नुकसान होने का, पीछे कहाँ चली गई. तो economically we all get that. So that's the reason which we did. So I want to go, it's more of a closing and a key takeaways which we want to take. Complex industrial process, that is one of the thing which in industry, okay. Regulatory compliance portfolio, okay. Regulatory compliance, portfolio strategy, distinguish industrial effort and academic. Future trend, still I am bit skeptical, but I am not closing my door of either my brain or my any ear. Either my brain or my any ear. I just want to know more about AI because still one drug comes, I will be more of a fan of it. But there is a lot of things beyond AI. In my intention, AI can do some process optimization in that, but discovery as such, they can unplug people, it's impossible. Understanding these dynamics helps researchers collaborate effectively and translate science. So that was the take-home message which I told you guys. We live in one building, one roof. We all collaborate together cross-functionally with all those mind. I know the chemist doesn't know the DMPK or another know the biology, but they all come, and over the period of time, I understand chemistry, they understand DMPK, and they all can understand each thing, and they become a full-grown scientist. So just focus a small thing in your area, you can do a successful. Questions are there, if you can ask any question, that's all. But next slide will be more, next topic will be more of a concept oriented. I will be talking. This was more of an industrial perspective. Okay, thank you.
All right, so we have the question session. Anybody?
Yeah.
Sir, as far as AMR is concerned, what are the prospects that lies in the drug repurposing?
A wonderful thing. Wonderful people are doing it, and uh, see, that's also one of the stuff which we can say as a Me Too type. Okay, so people are doing. I know the day I was in IITR, and there was a guy who was doing it, and it was more of a brain work. They were doing it, CNS work they were having, and they did it. So they are asking, still they are on an early stage of a candidate. They are doing a doc PK of that molecule. But I said, why the hell you are doing once you have a human data with that? But I think they want to see how the BBB is crossed. So that's something is very novel for that, but you have to be a very knowledgeable stuff where you are going and seeing the target do have a cross function with that drug and the target you are targeting as a repurposing. That's a beautiful field.
So, but what if it's given to non-targeted population? For example, if a drug is having for a certain comorbidity like diabetes, it is an anti-glycemic drug, and it's also having some anti-infective potential. Then how can we give it to someone who is not having problem of glycemia?
That's what I am saying, that the patient population, when you have to choose smartly. That's how the strategy will be. Why we want to give it, then we have to give a different drug. See, most of the drug now, they have an issue with the, I will be talking more on DDI, that is a drug-drug interaction. Okay, so people are very much focused on that, and there where you can come up your smart idea. But repurposing is something people are started doing it, and hopefully only thing help you is you don't have to cross lot of safety barrier when you are doing a repurposing. Yeah, he has a question. Can you pass up? Yeah, you are audible. Tell me. But maybe not audible for the other people.
So I just wanted to just an unpopular opinion regarding the patenting and IPR. I think that in India, due to the high value market giants that are currently present, like Eli Lilly, Sun Pharma, you could, the academic institutions or private academic individual cannot hold on to a patent and fight if someone infringes his or her patent. It's the current scenario usually tends that we should focus on technology transfer rather than filing a patent, because it's just an unpopular opinion that I think that in India it's hard to battle for a patent for an individual rather than.
I would love for sir to come in, but yeah.
Technology transfer and patent formation or patent giving patent. They are two different things. Technology transfer. This is a cub. I transfer to you. Okay. Are you going to make any money out of it by this transferring? I, as an inventor, I'm not going to make any money out of it. Okay. When I will make money, when I patent it. Okay. So patent is the ownership right. Okay. Technology. So you can transfer after patenting it. You are the owner. You can transfer the technology to Dr. Zain, and then he has to pay you. So this is a triangle now. Technology, you told somebody that, okay, I have this thing, okay? He said, give to me and I will talk to some company. He talked to company, he took money, and in Arabic they say khalas, okay? So that it is done, okay? So there is nothing like technology transfer and patenting. They both goes together. That's what I said in several times in last 3 days. Please don't publish if you think, means for academics you have to publish to get promoted and get the grant. But if you think your invention is good for marketing, patent it and then publish it. and then do the technology transfer. We have every company and every institute has an office of technology transfer. These people have, they don't give anything to Eli Lilly or anything until unless they have a right to disclose. So this is another thing. For example, patenting is very expensive. So I gave you this cup, but I have an agreement with you which is called non-disclosure agreement. I showed that. So do a non-disclosure agreement. It is a standard form on the internet. Print it out. If you're talking about your even idea to next person, ask him to sign the agreement, okay? Because you are telling the idea that may be $1 billion idea. Okay, make sense?
Thank you, sir. Just to add one thing, which type? No, leave it. I have. Which type of a patent we are taking it? There's nothing like that. That is infringement. You can't fight. But depend how broadly you have done. Whether you have filed in US or you have filed in different countries. Other countries can do it. They can do things. So this is not easy. It's a whole field. You know, you word use the patent. It becomes no. In Indian scenario, you can use it. Outside people can do it, okay? So it's all cost money. And one more thing very important, I was hearing other day there was a talk going on and somebody said, I have a molecule, can you take it forward? Okay, so molecule doesn't work like this. So it's very important to know, your sir was telling, you know the importance, don't publish it. You hire a KOL, key opinion leaders somewhere or somebody who has expert, get an NDA of it and try to evaluate your discovery which you have. When I saw something, I said, oh yeah, for me it's nothing, you publish it will be better, that will be late, somebody else can make it. But you think this is drug, no, that's a very early stage of a hit molecule. You made only a molecule, but there's a long way to go, you know. So try to understand that part.
So my question, it's slightly different. Sometimes doctors write some medicines for any disease, and when we go to a shop, medical shop, they said, oh, this particular drug is not available, you can take the another choice. So should we proceed as
Absolutely.
As
but focus on taking a drug from a good company.
Yes.
In India, we make a same drug with many companies. Don't take from Agra based or some people who change formulation. I remember I was taking a drug, a diabetes drug, and I took it from here only. And it's interesting story, you know. I took it and I usually check. I was having my own sensor. So I was seeing, oh, it's shooting like something. What's the reason? So I took the molecule, sent to my lab. They did HPLC, they ran it and said it was a 25 mg, but it was just 10 mg there. So they reported me back and said, then it's a 10 mg. I don't think it will be affecting. If you want to take a double dose of it. So I changed it, and that is how it is, you know.
But sir, for common people, how we decide?
It's a better say. That's what I didn't use that all English with you. I just told you, take the drug, see the best company, use Sun Pharma, because those company, they have a baggage, and if they fall in that trap, their whole stocks will come down and they will spoil. So they don't do it. I'm giving. I don't know much.
Same story I have, and same thing we did. I'm also taking a metformin. When earlier they get metformin from Germany also and from Oman also. So one time from our pharmacy in hospital, they gave me from Oman, and my blood sugar was little bit up, not a whole lot. I really monitor very, very carefully, and I said there's something wrong. So I talked to my friend, so because I lead that group also, and I said one of the scientists that why don't you check now 6 metformin, not one, from Merck, from Hexal in Germany, from JPI, from that company. So they did that work, and they found that the Oman company has the lesser amount of, still it was 95. okay? And the Merck and Hexal has 110%. So we published a paper on that. That's the important part.
Yeah.
So we have a very good paper published, and there was government came also to and talked to us, and they banned the Oman company. So here we are on publication of our paper.
You might have seen
So
some
we
of
have
the
the
plays.
resources, so we did that. This is the person actually, antibiotic doesn't work. Check where you are buying from Glaxo or some other company. Is very right that you have to check sometime. I know you don't have LCMSMS, so how you do that? So we have to do it ourselves.
अपने यहाँ तुम्हारे HPLC होगा ना? हाँ?
No common people.
नहीं नहीं हम तुम्हारी केमिस्ट्री तुमको ये प्रोजेक्ट ले सकते हो आप टेक HPLC would run 5-10 compound market crush करो भेजो वो भेज भी देंगे and publish a data and that will be good for everyone, you know.
So Dr.
That's one project you guys can take.
We published this paper in Helion, which is a self-publication.
मुझे connect कर लेना, I can tell you how because I am more of that LCMS guy, so I can tell you. But this is how. One more thing, you will find some of the drugs which are written SR, sustained release. So that is also a game which goes sometimes. ER also. So देखना. Anything else you have? Yeah.
Sir, we spent around 12 years from schooling from class 1st to 12th, and after that we do BSc 3 years, then MSc 2 years. Then why not to go for industry for training after 12th or after BSc, so that our chances of absorption in the industry get increased? And there also after doing that MSc and doing PhD and coming to industry and getting absorbed here and not so much growth there.
So your question is, if I understood that, why to do PhD and just do masters and go industry?
No sir, after 12th.
After 12th, the basics are not theirs. See, in industry there is, it's not a, what we say, we need person to come and work. If you work for 20,000 rupees, then we will pay you 10,000 rupees.
Yes.
Okay, so this is how industry works. So I would tweak your question and say, people who are doing here PhD, go for, if you are really interested, masters also is a more chance of getting master's job than a PhD. PhD become a group leader, there are the lesser positions, you know. And here the industrial PhD also goes. You take a PhD, you take a supervisor. I have been a supervisor of a couple of people. तो एक यहाँ पे co-guide हो जाता, main guide होता है, उधर co-guide होता है, and you also can do it. So MSc के बाद भी, and then you are more trained. In our lab, there are a lot of, we used to publish paper because we were having a high-end instrument, HRMS and all. So on that 6 or 7 paper, they all get to good postdocs and all, they went to it for the people who are doing. So that's also an option for the GATE who has some, if you don't have JRF, you don't have money there, that's a way you can try it and do it. In the market, we don't have drug. In India, we don't have drug here, but in clinical trial, we have 3 drugs.
3 drugs. So how you manage the budget, sir, in for one drug? You need an investment of $.
No, so that's how we do it. Early on, till candidate stage or a phase 1 molecule, company pays it. It's 100 million or 200 million they pay. We take it. After that, we usually get partner with big pharma, and they take the drug. This is how the out licensing happens, and we don't have a deep pocket to work. Okay, so some companies try to do it, but that's not the way people do it. It's a big, because there is a lot of clinical trials, takes a lot of money. Okay, so next level when they go in our program also, we have a phase 1 molecule or phase 2. After that, we have to go and get some people, and then whatever the rights will be, as the molecule progress, they will get. I told the story of one molecule which we, EGFR mutant molecule, which we gave to the Blueprint. That was a $250 million upfront they paid us, 250 was later, and that 250 was stage wise. Phase 1 if they cross, they will pay you this, phase 2 they will cross it, and they stopped the molecule. We didn't got 250 million, but 250 we got it, what we outlicensed, and that the way they kill the program and kill the competition. I think that was the strategy they made it.
Thanks.
Mm-hmm.
So here we are appreciating the time and the effort it takes for a drug to come to the market. So sir, what is the practicality that personalized medicine would be developed because it would be targeting a smaller group? So obviously, why would the industry be interested in doing that?
No, we are not interested.
I can take that question.
I was going to.
That's what I was putting him. He is the guy with the person. I will tell you.
Before I answer your question about the personalized medicine, there is already an existing population of people who have got rare diseases. So rare diseases are not for everybody. There are very few candidates who are there. But it does not mean that you have to start from scratch the entire drug development process for that person. It's mostly that you are doing a matchmaking. That there are people, there are drugs, you are matching the right drug to the right person. That's personalized medicine. Right? So we are not doing starting from the scratch, $1 billion 10 person. That's not.
Means that molecule will not be developed right from scratch, you know. It's a tweaking which is important, and giving to the right people which you want to, and maybe the dose adjustment and other things you can work on it. And there is one more thing FDA has given, one concept, that is the orphan drug status, and that usually help us a lot. Our both drugs are orphan drugs.
I can add a little bit more what Dr. Aziz said, exactly the same thing. So you, for personalized medicine, you don't have to develop a drug. Okay. For example,
Imitinib.
Imitinib. This drug is marketed for a lot of cancers. One of the cancer. For one of the cancer. But the scientists found out that some population has one mutation and it is more effective in that one. So that became personalized medicine, okay? It is given for other cancers with side effect, but for that population, it was a personalized medicine. The medicine was already on the market. They don't have to develop it. You are right that who is going to develop a drug for few people? True, no company is going to do that. That's the reason that malaria, which is part of the third world country disease, big companies are not developing any malaria medicine, and now there are a lot of resistant malaria is there already. So this is all politics, money, power, and all those things that also goes like anywhere else. But there are the third one is other one, like for example Bill Gates Foundation. They fund these kind of disease medicines which are not profitable. So there are Fox Foundation है, ये है बिल गेट्स फाउंडेशन है, हमारे यहाँ रिवोल्यूशन एक नया फाउंडेशन शुरू हुआ है, किंग सलमान सेंटर फॉर डिसेबिलिटी अभी शुरू किया है उन्होंने, वो भी फंड कर रहे हैं. So there are people are there, and you have to go out and find out who has the deep pocket and ask him to pay you for your research. That's another thing.
Yeah, there is a question.
Good morning, sir.
Good morning.
Sir, I want to know about what is the status of natural product mining in the industry, since I think that there are many natural products, especially who have documented effect, pharmacological action, and if we see there like in the drug discovery process, then we already have the documented effect. So we just have to find out the active pharmaceutical ingredient and just work on the drug discovery. So why not like do this rather than working on the whole new synthetic product? Then just
yeah, absolutely. People are doing it. This is how the money. There is a whole ministry, Ayush ministry is working. They have a lot of money in it.
So what is the scope of this?
Scope is that if you know real, because that's the toughest job, that you don't know the active part of it. That's the biggest challenge. And if you get it, then you have to do, you don't know the toxicity. You have to do some preliminary toxicity to understand that whether that active is okay, or it was given along with many things it worked, and because those were helping to remove the toxicity. So that could be the one of the thing. There is what a new litigation happened with the Baba Ramdev company. That was the reason they all pushed it, and he was answerable to many things. So I was talking to one of the guy who was an Ayush guy about 2 weeks back. I was there, and they are interested for those projects. If you really, I can link you guys with those people, and they can help you. They have a lot of fund also.
Yes, surely.
Sure, I can give you a number. They are wonderful people. You can connect Azabhai also. He write a project, push through it, and they said especially few lakhs is not an issue. That's my signatory. Then we have to go to the next level. Good. Thank you very much. I appreciate.
I think we are done with this session, and I hope the lunch is ready, but we are on time. So we'll reconvene here at 1.
Because that will be a longer one. So.
Yeah, that's the kind of tutorial, so please try to be back on time. I think 1 hour is enough for all the activities. All right, see you then.
दवा लेने से पहले 300 yeah. Yeah, shall we? Okay. If you don't mind, can you come forward? Come forward, come. It will be good. we would have a more interactive session now. This will be a bit of a very different science we will be talking today. We talked of luxury with many of the stuff so far, and we talked of industrial perspective, how drug discovery and development looks like, what we exactly do, and how we select the target, what are the type of a target which we work. Then there are some fascinating stories which we already told. So those stories tells that how people, why we do research, why we do it spend a long time and spend so much money, why industries are interested in doing it. We already told, because if you put 300 million or a 2.8 million dollar, you have a chance to earn 2.8 million per year, and 10 years patent, so you make many fold of money. And when you talk of a 2.8 billion dollar, that's not the dollar which is spent to develop a drug, which includes lot of failures in that process. Okay, so our finance people are a smarter guy, you know, they put all the stuff on the R&D team and say, we gave you, you gave me this, that's why this is the cost of this drug. Fine. So today I am going to, now this time it's more of the bridging the gap, integrating DMPK and toxicology cross translation. I am basically a biochemist by training. I did my PhD in biochemistry. So why I am telling you, this is a field which a DMPK and toxicology, people link more of a people who have done of a pharmacy type of studies, anything, but still the person, there are a lot of people who excel. So biochemistry and these sciences can come and learn a lot, and we contribute more to the people because we know the core of a biology here. Okay. So why I'm telling, because this is a cross-functional team, and here is the understanding of the other subject. You are good in your subject, that we all know well. So understand the other prospect of it and try to choose, work on your strength with the knowledge which you get around it. So first question is, why do promising drug candidates fail despite a strong biology? This is very important. I think I should recap a small bit what Sir has told you from last 2 days. We are all talking, we are all sitting, and we have been involved in lot of discussions, and he told you about the process. He started with what are the stages of discovery. We talked of hit, lead, preclinical, candidate, and then we go to the clinical trial. He talked of what are the targets which are there, and then he told how the chemists work, what are the type of molecules, how they make it, how they make a medicinal chemist, what are the optimization, we all talked, what are the use of it. So most of the thing, there are 3 paradigms of drug discovery, which I will be talking more on it. So most of the stuff have been compared. Then he went to the modalities of drug discovery. Now what we are doing, we are talking of PROTAC, we are talking ADCs, we are talking of molecular glues which are coming now these days. We are talking of some of the advanced work which RNA therapies which we are talking about. These are all Colgate. These are the first half of the drug discovery. Let me talk about the second half of it. Why do promising drug candidates fail? So all these efforts, you have the best brain, guys coming from different big institute with a lot of knowledge, still drug fails, you know. And I tell you, failure is something which we learn and we grab it, okay, and say, fail ho gaya yaar, chal nahi raha wo compound. Tumhara compound chala nahi. Chemists are so quick, their baby are killed, seems, and they fight for that, okay. And there is other field like us who have a job of failing first and failing fast, so that we should be cheap enough. It doesn't work in that way. Come forward, okay. Yeah. Good, thank you very much, I appreciate. So if you see, this is the slide which you will be finding many times coming. Start from first day, people are talking of this slide. And we already know, we already told last slide also why. I want to remind you that there is a lot of money invested, a lot of time invested, and the percentage is very low. So this is the field where people fails, and why this fail, this is the reason I will be telling you. So you have spoken the word attrition in drugs. I was already telling on it, how attrition in drug works, why nearly 90% of a drug candidate fail after the clinical trial. Even before that, due to this is not the only, science is not the only reason for the fail. Sometime you start a program and then you get a target. Today this is a hot target. All of the sudden you move forward, you say that a compound is in a clinical trial in a phase 1, we go and see the data, and sometime you find that efficacy is not coming. There is a lot of toxicity associated with this. तो क्या होता है, कंपनी में फिर एक फीडबैक आता है कि एक कॉम्पिटिटिव इंटेलिजेंस आती है कि कौन कहाँ पहुँचा हुआ है. हम लोग हमेशा स्लाइड बनाते वक्त देखते हैं कि कौन कहाँ पे है, कितने लोग फेज़ 1 में, कितने डिस्कवरी में हैं, कितने फेज़ 2 में हैं, किसके बाद POC है. तो जब देखते हैं यार ये फेल हो रहा है, तो फ़ौरन कंपनी जो है एक स्ट्रेटेजी बनाती है और उसको कहती है इसको आप बैक बर्नर में रख दो, अब हम इसको रोकेंगे, पहले हम इसका डाटा आ जाएगा तब हम आगे बढ़ेंगे. ठीक है, कहीं ऐसा, क्योंकि ह्यूमन का मामला कुछ अलग है, ब्लैक होल है वो हैं, और कुछ चीज़ बहुत अच्छी दिखेगी एनिमल तक, जैसे ही ह्यूमन में जाओगे, दैट इज़ व्हाट द ट्रांसलेशनल साइंस इज. आई एम गोइंग टू टॉक टू यू, दैट्स द नेक्स्ट लेवल. And this Jamia Hamdard do have a department of translational science, and at least on this, where we have a medical college, everything, this is the place where we should also have in AMU. Let me talk of this attrition. Is it everybody can see the slide? Okay, so there are three clinical safety. See, these are the paradigm which you have to read it. Clinical safety, first thing is, when you talk of a clinical safety, how it is, then you have an efficacy. You know efficacy? Efficacy means when you give a drug, whether it is affecting the target. I will give you an example. If you are developing a drug with a cancer, okay, hi-fi, let's talk of anything which is had to reduce the platelet. ET trial, which I am reviewing myself, that is essential thrombocythemia. That's a disease which is known as. What happens? Your platelet goes up. It's so much, people are loaded with millions of platelet in that and they're walking. All of the sudden, the job of platelet is, wow, it clots the blood when it's come down. So when it is inside the body in a higher amount, what happens? There is a thrombosis happens and people get heart attack. This is something which is there. It's around 8 million patients they have found globally, and there are some pockets which are huge. $2.6 billion deal happened with Imago. Merck has taken that compound. That's a very good compound which Imago has taken it. So why I'm telling, so when you give a drug, you have to check whether the platelet is coming down or not. This is the endpoint study. I don't want to go in a cancer's complexity. When you give a specific drug, your tumor shrinkage, your every stuff is going there, then you do a biomarker study, then you check it. I want to simple understanding. You gave a drug, whether it was efficient alag to control the disease. That's a, in that case, this is known as efficacy. There is a formulation, whether the formulation which you made, I think nanotechnology people should keep an eye on it, that the formulation, if you have, do you have a formulation? There are some drugs which could not be made an oral. Chemists tried their best, they were trying there to get the solubility of a compound, but the problem was that when they were working on a solubility, they were losing efficacy, but the drug was so hot, the target, they went for the IV and it remained. I will give you example for the people who are in nanotechnology, really want to work on it. Mirati compound is there with the KRAS G12D inhibitor. That's a hot target. It's an IV. If you can work with your own nanotechnology style and you make it a formulated in an oral, I think it's a bluster. Similar is the compound soratinib which we are talking of, Amgen compound, which is the first KRAS, is an IV compound. Let's bring an oral. Our drug is oral, is still in the early stage. Okay, the second is commercial, then a toxicology. If you see the graph, there is a PK viability, means you give a compound and you see whether the compound remains in a body for a longer time and how much the compound is there in that, that is known as a PK or a pharmacokinetic or exposure of the compound. Then you have a toxicology. You give a compound, sold data, but it still shows, there is a huge amount of a compound, which failed due to the toxicology, which failed due to efficacy, means efficacy is still a concern. And that's the reason we are all talking there is only 10% to 12% of a drug which is succeeding. But the question is why? Then you have to talk of the other phase, that is efficacy is the issue. There is somewhere a PK is somewhere issue, but PK efficacy goes hand by hand, I will be telling you, and then it's a toxicology which is there. Commercial is, I will be telling you, and then it's a toxicology which is there. Commercial is a different thing, strategy is a different thing, but it still doesn't contribute a huge amount and the further things, okay? We already shown this. This is how the structure looks. This is the drug discovery process. This is the arrow characterization till till high throughput screening, and this is a vicious cycle. Sometime you qualify in one month or 15 days, your compound good enough to move forward, but sometime this vicious cycle take years, okay? And then there is a blame game started, your assay is not good, it's not good, your compound is not good, your compound is a rock, you throw this compound in the container, we don't want to work with this. So all these start, failures only bring blames, that's all. Success, there are a lot of takers, okay? Okay, so what I want to bring on the table is successful drug is activity plus druggability and safety. Sir has went length with the activity. He told you how to make a compound with the chemistry, with the biology, with the target and all. It's all come in activity. Now the other arm is druggability. Your compound is very good enough. It will go to the target, it will hit it, fine. But the question is whether it can be taken by a human or not. That is known as a druggability. Means it is can be drugged or not. There are many drugs. Now there is a new science has came, undruggable target has came, and that has revolutionized. Protac is one of the best example. Still we don't have a drug so far. I want to see that drug, but this is how I want to tell you. So I will be more focused on druggability as well as bit of a safety. Okay, I will run through it. From molecule to medicine, this is the story, this is what you guys have to think of. See the effort of how many people are involved in the whole process. One is the chemistry, another is the biology, third is the cell modeling, another DMPK. DMPK is nothing, drug metabolism and pharmacokinetics. Manufacturing, toxicology, regulatory toxicology, manufacturing, and then important IPR. This whole circle is a vicious circle. Once it is stopped, biology goes to cell model. This doesn't go in arrow if you see. You might have seen that's a unilateral process. No, it's an up and down. You start with something, chemistry make a compound, send to biology, biology say it's a micromolar compound, you have an activity but you have to optimize, they start working on synthesis, they say oh यार synthesis बहुत मुश्किल है, 11 स्टेप 15 स्टेप है, मैं नहीं दे सकता, मैं सिर्फ 2 mg कंपाउंड दूंगा तुमको, हम कहेंगे 1 mg दे दो यार हमको दो, तो फिर वो थोड़ा सा कंपाउंड सही हुआ, फिर ऑप्टिमाइज हुआ, जैसे ही ऑप्टिमाइज होने लगा तो उसके बाद सेल मॉडलिंग की हेल्प होती है, बता दो यार कि कहाँ जा रहा है, कि हम इस पॉकेट पे कहीं छूट रहा है कि नहीं, यहाँ पे लाइसिन बाइंडिंग है, यहाँ ये बाइंडिंग है, उसके बाद छूट रहा है, तो वो कहेगा हाँ यार ये छूट रहा है, अगर इसकी सही से हो गई तो तुम्हारी तो ये और तेज हो जाएगा, तो फिर उसके पास वापस जाते हैं विद द नॉलेज केमिस्ट्री मेक्स अ कंपाउंड एंड देन बायोलॉजी वर्क्स ऑन इट से इट गुड. Then you get a something, a hit known compound, a 1 micromolar, people industry standard के हिसाब से 100 or 500 nanomolar का compound मिलता है. Then you go to the DMPK, and DMPK do have some assays to tell whether the drug will stay in the body or not. Once you will throw, it will cleave everything. तो वहाँ जाके उसकी स्टेबिलिटी चेक करते, कहते हैं कूड़ा है तो बेकार है, फेंको इसको, तो वापस फिर आता है वो बेचारा, अब स्टेबिलिटी पे मेहनत करता है, उसकी देखता है। फिर आई विल टॉक समथिंग ऑन मेट आईडी एंड समथिंग स्मॉल पार्ट ऑफ इट सो पीपल कैन अंडरस्टैंड हाउ इट इज। तो फिर पेटेंट भी चेक करते हैं उसी प्रोसेस में कि ऐसा तो नहीं कि मैंने जो मॉलिक्यूल बनाया ऑलरेडी पेटेंटेड है। तो केमिस्ट इज अ चैंपियन ऑफ इट, वो जो है रिंग मास्टर है इस पूरे प्रोसेस का, है। सो उसको ये सब पता रहना चाहिए। वो कभी कभी यही छोटी चीज़ें छोड़ देते हैं और वो बहुत 2 साल की मेहनत सब खराब हो जाती है। तो दिस इज ऑल द मैन्युफैक्चरिंग। फिर कंपाउंड अच्छा होता है, टॉक्स में जाता है, और टॉक्स में जाते ही कंपाउंड फेल हो जाता है, उसमें लीवर टॉक्स दिखती, ये दिखती, फिर वो वापस नहीं आता। या तो आप डिफरेंट स्कैफोल्ड पे काम कीजिए या कुछ कीजिए, देन ओनली यू विल सक्सीड, अदरवाइज इट विल बी थ्रोन इन अ बिन। See, 4 years, 3 years, lot of emotions, lot of people came at night, running system, running assay, and all fail in one direction, you know. This is how the criticality of this experiment, all the whole process it's a lot of emotions are attached. People talk of money, but I talk of emotions. We live with those compounds. Sometimes हमें रात को भी देखता था कि हमारा ये पीक इतना आ रहा है, ऐसे आ रहा है, यार वो खराब हो गया कंपाउंड, दिन भर उसी में आदमी रहता। दिस इज हाउ आई वॉन्ट, सी आई डोंट वांट टू ब्रिंग ए मैसेज टू यू गाइस एंड से यू लर्न ड्रग टेस्ट, नो, inculcate these process in your daily research life when you are doing it. Be with that, live, sleep with that, and sitting and thinking of your own problems, you know. Everybody can bring a problem, but when you do a problem, get a solution of it. Okay? So I will tell you why PK and PD is important. This is a two side of one coin of drug discovery. One is known as a PD, another is a PK. PD is a pharmacodynamics. In other words, what it says that what the drug does to the body. See, what the drug does to the body, when you give a drug, does it changes where the target you are working on? That is known as a PD, that is a pharmacodynamic. And the PK is nothing, it tells what body does to the drug. Okay, so this is a simple experiment. If somebody asks what is PK, what is PD, fine, PD is nothing, is what drug does to the body. It changes the biomarker, and you get the biomarker. Biomarker is nothing, outcome. Any change in the body which is due to the drug or something, any change comes out, how you will read it? You can't read to the cellular level. You read in the plasma, some protein comes, something comes out, you check it and see that it's a PD marker. Word is working on PD marker, and for academia, this is the best thing to work. Get one biomarker strong, I can tell you, you guys will be $1 million lab. People are looking it, they don't have biomarker, they have to run the patient, they have to depend on the physician to understand. Early on they don't get indication, if they get, they will not spend money so that they can stop themselves. Get a good marker and come back. All we have is a money in our lab. Okay, so what we do, we dose the drug. When you take a drug, it is absorbed and then it goes to the plasma, and there you see the circle which I am trying to tell you guys. I don't want to get into protein binding and all, I don't know how many people know, but this is just a concept, simple philosophy you should write, but still there is a lot of controversy going on, lot of papers came, there is the hypocrisy theory which also comes with that. There is a paper which is known as lot of banks were robbed by protein binding, people say, because some 99.9% bound compound has become a drug, some 45% bound drug has never become a drug. So it doesn't mean how much you need, it's a free concentration which matters. I want to go in that. But the protein binding means a free drug which is not bound to the protein. There, in our plasma there is a lot of albumin, okay, 3 proteins are there, they all bind to the protein drug, and they don't allow a drug to go and act on the target. Only free drug will work. Only free drug will work. This is what has been shown, and this is how the philosophy looks also, and this is the fact as of now we should learn. Okay. Then we have an elimination phase where then it comes into the metabolism. and there is a renal excretion. That's the way it gets. And the moment it comes a free plasma, plasma helps to move to the tissues and the target size, and then the effect comes. So what is more important is tissue non-specific binding. I don't want to go in that. There is a lot of stuff. When you don't have answers, you give all these non-specific binding and binding, then, but the science is there still. Target size and the effect. So there are two arms. If you see this arm, I can show. Okay, this arm on the left side of the screen, if you see, this is the pharmacokinetic part where the dose is given, absorbed, protein bound, then it is metabolized, eliminated. This is the PK part. The moment it goes inside the body, it goes to the tissue, from the plasma it goes to the tissue, and then it is distributed at a target site, and there the effect. This is known as the PD part. Is it clear, everyone? Okay, good. So I will go one by one. What is DMPK? DMPK interface to the drug discovery and development. DMPK impact on DDD. DDD means drug discovery and development, and how it has changed to the last 10 years, and future of DMPK view from here. That I will tell you something. This is much more important. What is ADME? You may find when you read it, there are two words which are used. One is the DMPK, another is the ADME. ADME is nothing, it is known as absorption, distribution, metabolism and excretion. That's what we have learnt in our previous slide. Is it okay? Clear? Simple. Make it simple. Don't make a fuss of it. Simple. Means one is ADME is absorption, the drug has to be absorbed, then it will be distributed or metabolized, maybe simultaneously or maybe sequentially, and then after the metabolism it gets excreted or eliminated. This is how it works. Sir was telling yesterday about SIP and all. I will try to cover with that. So what is absorption? The movement of drug into the body. I would have not written all those things. The simple reason I was asked that there will be hand note given to you guys, so it will be easier for you guys to read it. That's why it has been written. Do not consider a drug to be fully absorbed until it reaches to the bloodstream. Okay. So what happened? You might have seen once you take a drug and all of the sudden you vomit. And at home people say बच्चे ने ड्रग ली, ये तो दवा ली नहीं है तो उल्टी हो गई तो परेशानी हो गई तो बहुत. ऐसा होता नहीं ज़्यादातर है. उसका एक छोटा पार्ट तो चला जाता हो बाहर, फॉर्मुलेटेड पार्ट होता है, इसमें कैंटाइटी होती है, मगर वो फिर भी अब्जॉर्ब्ड. मगर मैसेज ये है कि बॉडी एक बार एब्जॉर्ब कर ले ड्रग. एब्जॉर्ब मत समझो, जब तक कि ड्रग ब्लड स्ट्रीम में ना चला जाए वो एब्जॉर्ब नहीं होगा. ठीक है, सिंपल. ड्रग administered early, intramuscular. So anyway, you can do it. You can do orally. The other day he was telling, and then IV, IM. You might have seen during the vaccination process, many of the things subcutaneous. So all these had its own benefit, and the best way is the patient compliance, which is important, and that's why the oral drug is every time loved with everyone. And I want the nanotechnology people to be really, really smart enough to see all the drug which is a blockbuster, still an IV, start working on your oral aspect and do some early work on it and make a drug. Okay, oral feasible. That's a formulation. If you come up, I can tell you there are a lot of takers in India only. Okay, especially they are good in these things. Distribution, the movement of drug around the body. So that's a simple, and elimination is the removal of it. Elimination takes two parts. One is the metabolism, another is the excretion. That's a two part. I will be touching some part in it. When we have a DMPK lab usually have four parts. One is the in vitro. In my lab is the in vitro, in vivo, met ID and pre-formulation. There are four verticals which we have in our lab. So let me talk of DMPK experiment what we have. So DMPK experiment when you do, when the compound come into my lab, I will give you an industrial perspective, that's why I am showing you this slide. When the compound come to my lab, what I do, first thing I do is solubility. I go and run in HPLC and run the solubility and check what is the solubility of compound. How much the solubility of compound should be good enough to run any assay? Anybody can tell? You all guys are doing experiment, you know well. I will tell you. See, you should know the final concentration of that experiment compound which you do. You do 5 micromolar, 2 micromolar, 1 micromolar, 5 mic. So that at least the solubility should be at 5 micromolar. If you are doing a KQ2 assay, I need a 5 micromolar. If I am doing a metabolic stability, I should have a 1 micromolar. If that solubility is not there, then the compound will precipitate in an experiment and you will never get a value which you want. Are you getting what I want to say? It's a very simple thing. Solubility in, and then the question come in solubility. जब तुमसे कोई पूछे ना solubility, तो जवाब देने मत खड़े हो जाओ, कहो solubility किसमें है, किसमें, बफर में, पानी में है, लिपिड में, ऑयल में, ठीक है, तो वो सॉल्युबिलिटी किसमें होगी, जिस मीडियम में आपका एक्सपेरिमेंट हो रहा है। तो ये हमेशा याद रखो जब भी कोई चीज लो कि सॉल्युबल है कि नहीं है उस पर्टिकुलर मीडियम में। तो हम बफर में कर रहे हैं pH 7.4 और देन योर pH कम्स इनटू प्ले। ओके, योर pH and pH should be relevant to the physiological or particular experimental condition which you are looking for. Are you getting? I am trying to be a more tutorial aspect, not the lecture aspect. Okay, trying to make you visualize that you are doing an experiment and how you are doing it. Okay, then you go to the permeability. First thing is solubility, then we run it, we get 20, early on we get 20 micromolar, 100 micromolar, 200 micromolar. How we do that? We do in SPLC. What we do? We make a linearity of it. You know linearity? Linearity, you make a linearity means known standard which you say, and then you run it at 200 micromolar the start, and then you put a unknown of your compound and run along with that particular standard and see where your solubility falls. Okay, and if it is beyond 200, we say it's a more than 200, our linearity was 200. Are you getting? Simple. SPLC देखा है किसी ने? रन किया है किसी ने? SPLC. अच्छा, गुड। खैर, दूसरा चीज जो है फिर हम लोग करते क्या है, द मोमेंट इट गोज वी आस्क फॉर द टियर 1 assay, यूजुअली डिपेंड करता है अर्ली कंपाउंड के बारे में बता रहा है, सॉलिबिलिटी, उसके बाद हम KQ2 कर लेते हैं, परमेबिलिटी, पैम्पा भी कर सकते हैं मगर अब नहीं करते हैं। KQ2 परमेबिलिटी, KQ2 एक सेल लाइन है जिसके अंदर जो है वो अपनी कोलॉन सेल लाइन है बेसिकली, है, उसको हम लेते हैं और उसको लेने के बाद उसके अंदर वो एक मीडिया रहता है, उस मीडिया के अंदर वो सेल ऐसा है, उसकी जो अपने पास प्लेट है, मिलीपोर की प्लेट होती है, और मैं प्लेट अगर वहाँ से आता तो प्लेट लेता, तो प्लेट में उसके दोनों तरफ है, तो प्लेट वो होता क्या, नीचे वाली जो वेल है उसके अंदर जो है अपनी सेल लाइन हम लोग लग जाती है। तो एक अपाइकल पोर्शन होता है, एक बेसल पोर्शन होता है। अपाइकल पोर्शन पे आप उसके ड्रग को अपने डाल देते हैं और डालने के बाद अब 90 मिनट तक उसको वेट करते हैं, और वेट करने के बाद है वो देखते हैं वो नीचे, उसको थोड़ा सा स्टर कर देते हैं, रख देते हैं, और उसके बाद वो नीचे आ जाता है। अब क्या फायदा है KQ2 का? KQ2 का फायदा है, देयर आर टू टाइप ऑफ परमेबिलिटी, आई विल बी टॉकिंग मोर ऑन इट, दैट इज द पैसिव, अनदर इज एन एक्टिव परमेबिलिटी। यू नो वेल व्हाट इज पैसिव एंड एक्टिव? नहीं जानते? पैसिव का मतलब ये हुआ कि ऊपर से कोई चीज नीचे फेंकी, खुद ब खुद आ गई, एक्टिव का मोटर लगा के पानी चढ़ा दिया। ओके, सो दैट इज व्हाट एन एक्टिव परमेबिलिटी, वेयर द ट्रांसपोर्टर्स कम्स इनटू प्ले, एंड देयर इज अ ह्यूज साइंस इन ट्रांसपोर्टर। अगर तुमने ट्रांसपोर्टर में भी काम कर लिया, PGP पे, MDR1 MDCK पे, किसी भी अच्छे सेल लाइन पे लेके तुमने अच्छे PGPs पे काम कर लिया, तो यू आर डन। OATP पे, OTPB1 पे, these are the transporters people are working, and whole departments are made on those. So I am saying just choose one and start working on it. Okay, then you do solubility. I told you how many types of solubility is there. Then you do the distribution. We also run the protein binding, and just to see that the compound should not be very high bound, because it helps to select the compound. If they have sent me 10 compound, I have to qualify and say these are the 2 compound good from ADME perspective, so please deprioritize the other compound and just move forward with the 2 compound. So that's the way we do it, and protein binding plays an important role, because I will be telling you why it is playing a role in it. Efficacy is something which we link with the protein binding, where the free concentration comes into play, and fold over EC50 and other things which we calculate. I will be telling more on it. Tissue distribution in rats and mice, That is what, that's a lateral stage, and then you do excretion, that is a biliary and a urinary, that's a different aspect. Important is metabolism. So chemists want to know whether the compound is soluble, whether it is getting absorbed, and whether it is not getting metabolized. Everybody want our compound to stay for a longer period of time. But if you say it's 100% stable, then also it's a tension, because compound will stay for a longer time and then it will kill or make a toxicity. So it's more of a hit and run situation type of a thing also we want sometimes. I don't want to go in that philosophy, but metabolism is metabolic stability, liver microsome, S9 fraction, hepatocyte. These are the in vitro models which are done. Liver microsomes you all know well. These are the cytosolic is a microsome, you know. In microsome basically SIPs are there, and those molecules, when you do the experiment, this is the easiest method and most predictable one. S9 come with the cytosol. Hepatocyte is the best model, but it's a very costly affair. Now we have a cryopreserved hepatocyte. Somebody is really interested in working on, especially the zoology people or what, they start working on isolation of hepatocyte or something, and if you bring a new technology, Thermo Fisher is looking for people who can come and join them. Okay, new technology, new platform. Hepatocyte is something everybody is looking for, because there is a viability issue, everybody is struggling. Down to the line with the 90 minutes of experiment or 3 hours, they usually lose a lot of viability, and in that form they usually get a false negative experiment, which is the problem, false positive, sorry. So next is SIP. There is a recombinant SIPs also, FMOs, I don't want to go in that. And then we have a DDI SIP inhibition. Sometimes if you see, I don't know doctors, you observe or not, doctor give you a drug, along with that they give another drug, and then they say you have taken this drug, you don't take this. Why? The simple reason, if they understand that's important to the pharmacological aspect, that this drug is, see there are a lot of SIPs, I will be going and I will telling you more about. Then you have in vivo plasma, then you have glutathione tribe, blood partitioning, TDI-MBI, these are all assays happens in my lab. Okay, these are all the studies happens in one more lab, and its a turnaround time is just 3 days. Okay, and that's a game with the turnaround time. So this is what I will, it will help you guys to understand more how the absorption happen. It goes to the stomach, duodenum, intestine, then it is absorbed, and absorbed, that is absorption, through the portal vein it comes, and then it goes to the liver, where the systemic circulation, kidney, and then excretion. What is important is that the part which goes from intestine to the portal vein, that is absorption, and from distribution, from the absorption or a portal vein to the liver or systemic, this all come under the distribution, and the liver is the main place where the metabolism happens, and then you have an excretion which happens. So this is how our drug works. Okay, and this is how you can see in a schematic diagram, which is actually the site of action, clearance, and everything. I think it will be more easier for you guys to understand how what I was telling so far. So this is the whole process, and without giving a drug, you have to understand in vitro system that all these drugs, how it works. See, I can't go to the animal costs us $100 usually, and one animal, you can't have that cost, you need something which is from in vitro setup. And that's what I was telling in a previous thing, that these are the assays which answer you how your drug will move to the next stage, and what will the stages will there will be hindrance. Okay, are you guys with me? Sure? Or food effect? No. Good. In our usually in pharmacology, because I work more on pharmacological aspect, so usually our drug also we don't give with food. Fasting we give 2 hours fasting. Okay, simple reason, there is a food absorption is the one thing which is affected. So you guys are not having a food effect, huh? Get some coffee, run your caffeine. Okay, really you want? Okay, I know this is a new field for you guys, but still try to understand, it will be helpful for you guys. Now the question is, what is the desirable ADME? You need a good absorption, good solubility, you need a good exposure to the target. Of course, if we want to kill someone, if we want to throw a bomb at one place, we need that the best, we should hit to the target, the missile should work nicely. So hitting the target is one of the things, that's why we need a distribution which should have. If we want a cancer cell, if it is working in a colon or if it is working in a kidney or it will working on a pancreas, it should hit there. There is many ways to hit. Whether sir was telling about the ADCs, now you started targeted one. Now you know the war just came, there was a simulation going on, people were knowing on computer what is the target, they were hitting from here and hitting to the zero inhibition contact with a put and shoot everything. So now we are working on that field. So distribution is what we really want, the exposure of a compound should be in the target, minimum elsewhere. We don't want to hit it because that's drug, anything which is outside, xenobiotic, anything outside is a problem, is a toxic. So you want the tox, you have a cancer cell which is in a liver, in a lung or somewhere, you want to hit that. You don't want, because cancer cell will hit there, but if it hits anywhere else, it will destroy the same type of destruction which was happening in lungs. So you have to avoid it. That's the ideal condition. Then it's a metabolism, minimal first pass effect, minimal potential to the inhibit to the induced CYPs, and no toxic. I think you guys have heard of CYPs a lot. I will be talking more on it and try to understand what is CYP. If somebody is interested in biochemistry, run CYP, learn something more on CYPs. This is the enzyme, is still working, new enzymes are coming, new CYPs are coming, and that has destroyed an industry with a big time. Balance between the metabolism and excretion, that is what we are talking of, excretion. Now I will talk you about the absorption. Okay, how does the drug get the bloodstream? First question you understood. How long does it take? Routes of administration, IV, no absorption limitation, and drug directly into the venous. So what happened? Why we give IV and why give oral? Do you know? If you have a problem with ADME, if you have absorption issue, so what we usually do early on, if we stop it. Okay, don't worry, you want to check whether compound is working or not, we will give IV. So absorption route is over. So in absorption only you need, oral only you need absorption, IV doesn't need. Where IV will be hitting it? IV will be hitting systemic circulation directly, okay, and that's why it will affect immediately. Okay, and then oral agent must be soluble, pass across the membrane in the stomach and other thing, gastric pH and everything. Now how you access that was I have told you, and it's written just for your hands out, I have written it, that what absorption, what you do, you run KECO2, PAMPA, and then you do in vivo PK and see how the viability is there, that I will be explaining you. In vitro protein binding, in vivo tissue distribution, that is how you will do a distribution study. So these are the ADME if you want to learn, that what the message I was trying to give. You can't run an in vivo system. There you have to run with the in vitro system. That is what now the science has changed. I will be talking in the subsequent slide. FDA has also changed. There is organ on chip. I will be telling you more on it. Now FDA want that you should use minimal animals, and that's the reason our in vitro ADME people are more important. Only thing is IV, IVC which has to do. IV, IVC. What does it mean? In vitro, in vivo correlation. So you have to establish somebody will tell, oh, I have developed assay here. Fine, but how much the assay is relevant to animal model? The next question comes, how much the in vivo model which is in animal is relevant to human? That is the translational science. So we are talking of three science. One is in vitro to in vivo, that is IV, IVC, that is you go with the animal and then you check whether from animal it is relevant to the human or not. That is known as the translational science. So anything which fascinates you guys, pick up and move, man, you can do it, okay? Okay. Yeah. Yeah, it's absolutely wonderful question. Now the things have, AI has also came up, that the in silico is the base for that. Now we have a lot of data, and sir can bring some more point on him being a wonderful chemist. So they know there is a functional group, there is structure similarity, and these groups are there, they all have a data. So they have data mining is known as the process where you get thousands and thousands of compound and you run and see these type of a compound, heterocyclic or something, you have a compound and this is what the permeability you get. Okay, so they run it. We also ask them to run and give us all this data. Of course they are not, we are not sure 100% they are not right, neither 90%, but at least 50% they are right. So what happens, some of the series it gets a good correlation, some it doesn't get a correlation. Same happened with the in vitro to in vivo. So it's a question is very relevant. These are the things which are AI, what they have done, they have, AI is basically a garbage in and garbage out philosophy. I should not use the word because it hurts people, but the simple reason that you get a lot of data which is the real time. So what you teach as a machine learning, that only throws it. Our field is one of the idiotic field of pharma industry. Why I am saying this, simple reason, we all keep everything to our chest. There is a simple reason we spend a lot of money on it. So lot of failures have never came to the market and you don't have a data. So you have a data which is more of a successful story. From where the data mining is happening, from the way the machine learning, the data which is available in public domain. And then AI put a lot of algorithm which is a finer way of in silico and they have a better data. Still one compound in silico has produced, that is in phase 3. If it comes, I can tell you that life will change with the pharma industry with that. Still I am skeptical. Okay? Anything else? Oh yeah, both are different things. One is the druggability and another is the efficacy. When you talk of an oral thing, means your drug is absorbed or not, that's all. If your drug is good enough to work on a target, if you have a very good exposure, it will work and it should work. देखो एक होता है ड्रग को अंदर ले लेना और वो बॉडी में आ जाना. पहली चीज़ है घर के यहाँ आ जाए कोई, यहाँ किसी को मारना है, अगर किसी को कोई आदमी आता है पहले तो वो अंदर घुस जाए तो उसको एब्जॉर्प्शन कह दिया है, अब फिर वो फैल जाए, वो डिस्ट्रीब्यूट हो गया, फिर उसको तुम्ही को मारना था तो तुम्हें टारगेटेड हिटिंग पे शूट कर दिया है, तो वो आए, अब फिर उसके वेपन में जो शूट करना जिस चीज था उसमें इतनी ताकत होनी चाहिए कि तुम्हें मार दे. तो तुमने पहले सारा काम सजा दिया, लाए बैठाला सजा दिया, आदमी आ गया, एक्सपोजर आ गया, एब्जॉर्ब हो गया, एक्सपोजर आ गया, अब उसमें एबिलिटी मारने की तो मार देगा. अब तुम बच सको तो तुम्हारे में रेजिस्टेंट है, दैट इज व्हाट वी आर टॉकिंग. ओके, क्लियर? ओके, गुड. Once he will die, he will not see whether he got excreted or not. Yeah, of course, you made an endpoint, and then he will have a route to exit also. Okay, and people like that, they make sure that they get exit out of it. So their planning include that excretion part. Okay. So it path is just of 2 minutes, man. 3 hours, 2 hours, depending upon the absorption, how quickly it goes. No, the body is at working at 7.4. All the enzyme work on 7.4. Then we remember Isaac is a movie story. Then it's subtle change in the pH. It doesn't work. So you are more acidic. Your stomach has a more pH. You take a lot of food. That's the food effect I was talking of. When you take a food, your absorption get a problem because the enzyme doesn't work or works nicely. Some of them, because the enzyme work is the range, it doesn't work on 7.4, it works at 6.7 to something. So which is the optimal? That their work is more. That's what the message is for any person who is working on enzymatic studies. Okay, good. So next is metabolism. This is very important. I think how the body changes the drug. See, the intention of body is, if you take a drug, you have to make sure their job is to throw it. Are you getting? That is the job of body. Any xenobiotic, anything outside, from outside if we take, body has the art to throw it. So what it does, the best way to excrete is urine. Okay. So how they do it? They have to see, this is a game, you know. Sorry, I get diverted usually. This is a game, you know. You need a drug which is absorbed. How you want to absorb a drug? A membrane is there where you have to pass. Membrane is made up of phospholipid. Okay. So lipid means like begets like dissolve like, you know. So it has to be lipid soluble. So you need a drug which sir team usually log D and log P. This is something which we do early on. So log D, how much it is? So if it is, so we make it greasy to make it more potent. What we do, because our target is intracellular, we have to go inside. So chemists make it so greasy that molecule, it goes inside and hit. But the problem happens, it goes inside, but the body want it to solubilize it. So it changes with all the enzyme job is to make the protein hydrophilic so that it can move outside. This is the job of an enzyme in the body. Any job, oxidative enzyme you will find, you will be learning that. Is it okay? I am trying to make it simple, excuse me, I am not a teacher, but trying to make sure you understand something, okay? Okay, and then you have this baby which is known as CYP450. This is something which is very, very important in DMPK lab. We do a lot of work with that. We do recombinant, we try to do with microsome, we do with cytosol, we do with hepatocyte, and this is something which has made a life hell with the DMPK and especially the drug development people. So simple as that. You can see that the cytochrome P450, P450 you all know well, the absorption is P450, that's why it is written, and cytochrome is the iron based enzyme, okay. And then 55%, 90% of a phase 1 metabolism is CYP mediated, that's why this plays an important role. Fine, means most of that 90% of phase 1 metabolism, there are two phases rather, phase 1 and phase 2 metabolism. I don't want to go in that, and then you have UGTs and other thing, but the phase 1 metabolism where is 90% CYP is involved. Unchanged drug, non-metabolic 25%, if there is no drug change there will be 95%, 25% यूरिन बाइल की तरह से निकल जाता है. ये है important slide जिसमें बताते हैं कि CYP का डिस्ट्रीब्यूशन कैसे है, रिलेटिव अमाउंट कितना है और कहाँ है. देखो टोटल CYP इतने अभी तक हमारे पास हैं, और भी हैं मगर हम इंपॉर्टेंट जो मेटाबॉलिज्म रिलेवेंट हैं उनके बारे में हम तुम्हें बता दें कि ये देखो इतने हैं, जिसमें से 1A2 13% है, 2A6 4%, 2B6 इतना है इतना है. मगर एक चीज़ याद रखना, CYP3A4 है 30%, ये मेन एंजाइम है जिसको सब पे काम करता है. मैं क्यों ये बताना चाह रहा हूँ कि ये मत समझना कि ये जो ज़्यादा है वो ज़्यादा है क्योंकि नेचर ने बनाया ऐसा है. जो ज़्यादा है इसका मतलब हुआ इसका रोल ज़्यादा है, उसकी तरह के मॉलिक्यूल ज़्यादा आते हैं, मगर ज़रूरी नहीं है. हम लोग ड्रग बनाते हैं वो होता है कि कभी CYP1A2 ही मीडिएटेड मेटाबॉलिज्म है, बहुत सारे फेनासिटीन है जैसे, उसको उसी से मेटाबोलाइज कराया जाता है और बाद में उसको इनिबिशन पे डाला जाता है, डेक्स्ट्रोमेथोर्फिन डाल के उसको बताया जाता है कितना हो रहा है. ह्यूमन साइटोक्रोम P50 द रिलेटिव कंट्रीब्यूशन टू द हेपेटिक मेटाबॉलिज्म, ये देख लो 3A4 जो है वो कम से कम 82% गट में जो मेटाबॉलिज्म है उसमें इन्वॉल्व है, लीवर में 40 to 50% है, और 60% ड्रग इज मेटाबोलाइज बट प्राइमरली बाय CYP. दैट इज अ पेपर पब्लिकेशन. So this is the elimination. I don't want to go much more. I already told the drug is taken oral or IV GI tract which goes to the liver, and from GI tract to liver, A means absorption, then it is metabolized, and then its excretion or distribution to the tissue, and that's how the elimination happens. Now we covered in vitro. Now we have talked much of metabolism here. Now the second portion is PK. That's an IV. Anybody needs a bio break? You want to walk? You just give me 5 minutes, okay? You just walk if you want, so that you can come with a PK. You have a 5 minutes, okay? Yeah, please. Get your walk, get your leg stretched, and see. So, are we there? Okay, good. So, let me start a next topic, that is a pharmacokinetics. So far, we talked of ADME, and this is also the part of a ADME. In the sense, this is in vivo type. We talked of in vitro, we talk of absorption, distribution, metabolism, excretion. This is how these are all coming in one topic, which is known as the druggability. सी ड्रग के दो हिस्से हैं समझ लो, एक हिस्सा है एफिकेसी, दूसरा है ड्रगेबिलिटी, और तीसरा है सेफ्टी. अगर तीनों चीज रहेगी तभी ड्रग सक्सेसफुल होता है, वरना वो डस्टबिन में फेंक दिया जाएगा, चाहे आप अरबों रुपए खर्च कर दो. ठीक है. पहले हिस्सा जो उन्होंने कल बताया कि भाई क्या क्या स्टेजेस होती हैं, हमने डाटा, टारगेट, एफिकेसी के बारे में बात की. आज हम बात कर रहे हैं कि ड्रग क्या हम देंगे और वो यूज. कभी कभी आपको लगता है यार ये बताइए उसने खाया तो उसको आराम मिल गया, हमने नहीं खाया तो हमें आराम नहीं मिला, क्यों? एनीबडी गेसेस? You read all those things, you might have known about it. Yeah, body respond, or we can say हो सकता है हमको एब्जॉर्ब नहीं हुआ, हो सकता है कि हमारे में एक्सप्रेशन उस एंजाइम का ज़्यादा था, तो जैसे ही गया उसने उसको किल कर दिया. तो वो चाहिए था 100 ग्राम काम करने के लिए, और एंजाइम ने ऐसा काटा कि उसके अंदर 1 ग्राम बचा है, तो फिर वो काम नहीं करेगा. बुखार थोड़ा कम हुआ, नहीं हुआ. तो इसलिए हमेशा याद रखो, जो आपको दवा सूट करती वही ले, नए एक्सपेरिमेंट ना करो. अगर ना कर रहा हो तो उससे ये परेशानी, फौरन डॉक्टर के पास जाके चेंज कराओ दवा. आई एम ट्राइंग टू गिव यू विद द एग्जाम्पल्स विच यू डू इन अ डेली लाइफ. दिस इज समथिंग वेरी इंपॉर्टेंट टू अंडरस्टैंड. तो उसके बाद सी ये क्या होता है, जब बॉडी ड्रग जब जाता है तो ये ग्राफ समझ लो. पहले तो इसको समझो, ये लिया हमने, इसके बाद गया, ये जो हम अभी तक बता रहे थे अब्जॉर्ब हुआ, फिर ये लीवर है, लीवर में होते हुए इंटेस्टाइन में गया, इसी लीवर के थ्रू जब निकला तो ये पूरे बॉडी में डिस्ट्रीब्यूट हो गया, और उसके बाद यहाँ लीवर है, यहाँ मेटाबॉलिज्म होता है, फिर ये एक्सक्रीशन हुआ और निकल गया. मैं बार बार रिटेट कर रहा हूँ इस बात को ताकि तुम लोग बात को समझो. ये बहुत ज़बरदस्त साइंस है जो बता रहे हैं हम. आई हैव 62 पेपर्स ऑन दिस साइंस ओनली. ओके, सो आई एम ट्राइंग टू टेल यू गाइस टू अंडरस्टैंड. 14 पेटेंट इसीलिए हैं कि हमने कुछ नया इसमें किया हुआ है. तो ट्राई टू अंडरस्टैंड द बेसिक्स ऑफ इट. दिस इज अ फैसिनेटिंग साइंस, यू लव इट. ओके, नैनो टेक्नोलॉजी ये जो ब्रांच है, दे कैन लुक ऑन दैट पार्ट एंड दे कैन डू वंडर्स, यू नो. सो दिस इज समथिंग विच वी हैव टू डू. ओके, तो क्या होता है जब ड्रग बॉडी की, आप ग्राफ को देखो, ये गया और एक सीमैक्स तक पहुंचता है. होता क्या है, कोई भी आपने देखा होगा, बेस्ट एग्जांपल, आपने पैरासिटामोल खाते हो, डोलो 650 फेमस ड्रग ऑन अ ड्रग स्टोर. ओके, बुखार हुआ, 650 लिया और खा लिया है, और उसके बाद किसी को ऐसा हुआ है कि खाते ही बुखार खत्म हुआ? नहीं. इट टेक्स आवर्स, इट टेक 1 आवर, 45 मिनट, 42 मिनट. क्यों? हम लोग की लैंग्वेज में बोलते हैं घरों में कि दवा का असर नहीं हुआ अभी तक. हैव यू हर्ड दैट वर्ड? ओके, व्हाई इट इज? दिस इज द रीजन व्हाई इट डजंट वर्क क्विकली. ये गया, ये एब्जॉर्ब हुआ, एब्जॉर्ब होने पे जब वो सीमैक्स पे पहुंचा, जब उसका मैक्सिमम कंसंट्रेशन था, तब वो बुखार पे एक्ट करता है, फिर वो धीमे धीमे वो बाहर हो जाता है. फिर आपसे कहता है 4 घंटे बाद लेना. क्या कहता है? क्यों लेना 4 घंटे बाद? क्योंकि 4 घंटे का 2 घंटे उसका हाफ लाइफ जो है वो 1 घंटे का है. उसके बाद वो नीचे चला गया, ग्राफ खत्म हो गया, ये एक्सक्रीट हो गया. अब आपको फिर बुखार आएगा, क्योंकि इन्फेक्शन तो खत्म नहीं हुई, तो बुखार की दवा थी सिर्फ, फिर 4 घंटे बाद दिया गया. आर यू गेटिंग? तो दिस इज नोन एज, इस स्टडी को कि ड्रग लिया, एब्जॉर्ब हुआ, कब सीमैक्स पे पहुंचा, कब निकला. Try to understand, reiterating that word. You take a drug, You take a fever drug, dolo something, it takes 1 hour to act on you. Simple reason, because that's not the Cmax concentration of it. It works on a Cmax concentration, it gets excreted, then doctor says you take after 6 hours. Means your fever will come, you have not killed the infection, the cause is still there, effect has been reduced, now the cause will induce one other effect and that effect will be controlled by your drug. Are you getting? एक होता है कॉज और एक होता है इफेक्ट. कॉज है इन्फेक्शन, इफेक्ट है फीवर. फाइन. इफेक्ट की दवा हमने दे दी फीवर की, वो थोड़े. तो इस स्टडी को कि ड्रग लिया, कब सीमैक्स पे पहुंचा, कब निकला, उसका टी हाफ क्या था, कब इसकी, इसको कहते हैं फार्माकोकाइनेटिक्स. क्या कहते हैं? ए काइनेटिक ऑफ ए ड्रग इज नोन एज फार्माकोकाइनेटिक. आर यू गेटिंग इट? रियली? कंपनी pays a lot of money to understand that part, huh? How do we learn about the pharmacokinetic drug? We already talked physical chemical property, absorption, distribution. और ये सब क्या होता है कि हम लोग पहले जब ये देते हैं ड्रग केमिस्ट हमको, तो हम इसको पहले इन विट्रो में टेस्ट करते हैं. उन्होंने 10 कंपाउंड दिया, मैंने कहा इसमें से 2 अच्छे हैं, अच्छा इसमें से 2 में ये बेहतर है, इसको हम एनिमल में डाल के ये फार्माकोकाइनेटिक स्टडी करते हैं पीछे वाली हैं. अब इससे बहुत कुछ निकाल, मैं इस ग्राफ से कम 4 पेज लिख सकता हूं कि इस ग्राफ में क्या क्या है. ठीक है, और इसी से स्टोरी एक ड्रग की बनती है. ओके, नाउ यू सी ओरल वर्सेस इंट्रावेनस. ओरल जब लिया तब क्या हुआ कि हमने दिया ट्रांसफर टू द गट लीवर टू गेट इनटू ब्लड स्ट्रीम. Intravenous drug go to the directly to the blood stream, there is no absorption. That what I was telling, I am trying to repeat that word which I was telling earlier, but this will give you a message how it works. अब ये बताओ कि फार्माकोकाइनेटिक काम क्या करता है? Why are the pharmacokinetic important? If a drug is going to work, it needs to be in a right place, right concentration and right amount of a time. Simple reason. You need a migraine. You have a migraine. You know that migraine कैसे हो रहा है. अब जब माइग्रेन जहाँ हो रहा है उसकी टारगेट पे या तो ड्रग जाए, पहले तो पहुँचे, फिर पहुँच के उस पर काम करे और फिर सही वक्त तक रहे, तभी कामयाब होगा ना. फुटबॉल देखने वाले देखते होंगे, उसमें लेता है कि सेंटर फॉरवर्ड इज अ गुड फॉरवर्ड हु गोज एट द राइट प्लेस एट राइट टाइम. लोग कहते हैं वहां पहुंच गया, उसने शूट मार दिया गोल में. नहीं, ही स्टेज एट द राइट टाइम. ही नोज कि बॉल ऐसे आएगी, वो अंदर मारेगा, सेंटर करेगा, फिर हम शूट करेंगे अंदर. तो दैट इज हाउ द ड्रग वर्क्स. यू नीड दिस पैरामीटर टू फुलफिल टू हैव ए ड्रग वर्किंग ऑन इट. तो ये जो ग्राफ है ये बताता है. ये अब इसमें देखो कितने वैरायटी के ग्राफ हैं. एक ग्राफ बता रहा है कि ऊपर गया और बहुत जल्दी वो प्लेट्यू हो गया और नीचे आ गया. समझे? ये ग्राफ बता रहा है ये एब्जॉर्ब हुआ, थोड़ा सा टाइम लिया, फिर एक उसने सीमैक्स मारा, उसके बाद फिर ऊपर मारा, फिर एब्जॉर्प्शन हुआ. एब्जॉर्प्शन होने के बाद धीमे धीमे इसका, तो जितना बड़ा ये सर्किल होगा, ये जितनी बड़ी सर्किल होगा उतना उसका टी हाफ ज्यादा है. टी हाफ क्या होता है? Yeah, it's not usually half, it's along with a lot of things. 0 point, if you remember the whole story, people say half time जितना half elimination में जितना लग जाए, ऐसा होता नहीं है, हैं? उसके पीछे पूरा कंसेप्ट है. ठीक है, तो अगर किसी ने केमिस्ट्री अच्छी पढ़ी होगी तो उसको ये याद होगा कि टी हाफ इज नॉट द हाफ, इट्स a lot of things, lot of constant act on it. But for the sake of understanding, it's a drug. Half of the concentration happens at the respective time is known as a T half of a drug. यूजुअली हम लोग कहते हैं 3 घंटे का है, मगर कंपाउंड 2.5 घंटे में ख़त्म हो जाता है, एलिमिनेशन गिरा जाता है. मगर वो एक कैलकुलेशन है, ट्रिपीजियम कैलकुलेशन, जिस पे एक्ट करता है. अब देखो, अब क्या है कि अब इसी से ये मालूम हो जाएगा कि कौन सा ओरल है, कौन सा आईवी है. अब आईवी में क्या हुआ कि हमने प्लाज्मा में दिया. क्योंकि क्या करते हैं जब हम ओरल ड्रग देते हैं, पहले हम ड्रग को लेते हैं. ये ट्यूटोरियल है इसलिए हम तुम्हें बता रहे हैं, वरना आई वुड हैव थ्रू. हम लेते हैं, हम ड्रग को लेते हैं, 1 mg लेते हैं, उसके एक पर्टिकुलर वॉल्यूम को हमें डोज करना होता है. और डोज करने के लिए हम जो है एक बीकर लेते हैं, उसके अंदर फॉर्मुलेट करते हैं. आईवी हमेशा सॉल्यूशन फॉर्मुलेशन होता है. क्या होता है? क्योंकि उसके अंदर हम कोई और सस्पेंशन नहीं डाल सकते हैं, वो सॉल्यूशन फॉर्मुलेशन विद अ पर्टिकुलर पीएच पे रहता है, ताकि वो सीधे आईवी में जाता है सीधे और वेनस में जाता है, और ओरल को हम अक्सर सस्पेंशन करते हैं, अगर नहीं होता है तो हमको सॉल्यूशन करते हैं। उसकी कई रीज़न है क्यों करते हैं, वो हम आपको हो सकता बताएं आगे स्लाइड में होगा तो। मगर क्या हुआ, जब हमने आईवी दिया, वेनस में दिया, तो क्या हुआ, और फिर क्या करते हैं, एक पर्टिकुलर टाइम पे, डिफरेंट टाइम है, देखो 2 घंटे, 4 घंटे, 6 घंटे, 8 घंटे है टाइम है, इसी तरह 0 आवर, 8 आवर, 16 और 24 घंटे, इन टाइमों पे हम सैंपलिंग करते हैं। क्या करते हैं, सैंपल मतलब प्लाज्मा लेते हैं, ब्लड लेते हैं, अब ब्लड को स्पिन ऑफ करके हार्वेस्ट करके प्लाज्मा बनाते हैं। कैसे बनता है, कोई जानता है? ब्लड से प्लाज्मा कैसे बनता है, कोई बता सकते? कितने पे? हाँ, 3100 RPM। हाउ मच टाइम? रियली? बहुत जल्दी हो रहा है। कौन सा है तुम्हारे पास भाई? It's depend upon the rotor, it's depend upon the seed, you may be right, but it takes around 30 minutes or 20 minutes, something which we run, and make sure that you should not overrun it. Why you should not overrun it? Because it will hemolyze, you will get a lot of blood out of it, and it will be useless for doing an experiment. Okay? तो क्या हुआ, फिर हमने उसके प्लाज्मा लेते हैं, और फिर हम प्लाज्मा लेके उसको एनालाइज करते हैं। एनालिसिस का पार्ट मैं तुम्हें बाद में बताऊंगा। उस एनालिसिस से हम उस कंपाउंड को देते हैं जो दिया है उस प्लाज्मा में। अब ये जो कंसंट्रेशन है, ये प्लाज्मा का कंसंट्रेशन है, प्लाज्मा के अंदर ये जो पॉइंट दिख रहे हैं, ये नैनोग्राम पर ml में हम कैलकुलेट करते हैं। ये वाली साइड में देखो नैनोग्राम पर ml है, ये टाइम है। क्योंकि हमने आईवी दिया, तो आईवी देने की वजह से हमने डायरेक्ट ब्लड में दिया, तो उसको एब्जॉर्प्शन की जरूरत नहीं हुई, तो ये सीधे ऊपर से ग्राफ आ रहा है। और यहाँ पे हमने उसको ओरल दिया, तो ओरल एब्जॉर्ब हुआ, फिर डिस्ट्रीब्यूट हुआ, इसके लिए वो प्लाज्मा में जब आया तो ऐसे कर्व बना है। कभी कभी लोग गलत डोज कर देते हैं, तो पता चल जाता है कि आईवी दिया या एससी दिया। आईवी देने की जगह एससी दे देते हैं, तो उसके ग्राफ में ही दिख जाता है कि उसने आईवी नहीं दिया, क्योंकि एससी में क्या होता है, वो स्लो एब्जॉर्प्शन होता है, वो यूं जाता है, कहता है आईवी दिया, गलत होता है वो है। तो ये ग्राफ बहुत कुछ कहानी बताता है, ये ग्राफ इतनी कहानी बताता है, लोग ये ग्राफ लेके फोटो खींच लेते हैं बड़ी बड़ी कॉन्फ्रेंस में और जाके बताते हैं कि इनका ड्रग इन्फीरियर है कि बहुत अच्छा है। मिराटी का ड्रग आज काम, इतनी अच्छी एक्टिविटी है, इतना अच्छा उसका क्लिनिकल स्कोर है, वो सिर्फ मात खाएगा एस्ट्राजेनिका से ओरल की वजह से। मैं बार बार कह रहा हूं कि कोई फॉर्मुलेशन डेवलप कर ले, हमने तो कई तरीके से कोशिश की हुई है, हम अपने यहाँ नहीं कर सकते हैं, बाहर एक कंसेप्ट है मेरे पास, प्रोवाइडेड विल डू इंशाल्लाह। प्रोटोकॉल्स ऑफ पीकेज, पीके का प्रोटोकॉल कैसे बनाते हैं, वो ये है कि ट्यूब, व्हाट ट्यूब शुड बी यूज, व्हाट सैंपल विल बी कलेक्टेड, हाउ लॉन्ग इट विल टेक, इंटरिम सैंपल कितनी देर का लेना है, वो डिपेंड करता है, आपको ड्रग का मालूम हो कि कितनी देर रुकेगा बदन में, तो ये सब लेके हम लोग फिर फ्रीज़र टेम्परेचर पे स्टोर कर देते हैं, फिर उसको सेंट्रीफ्यूज करते हैं, उसको किसी लैब से किसी जगह शिप करना है तो वो भी टेम्परेचर कंट्रोल्ड होना चाहिए। एक और बहुत अच्छा काम है कि आजकल टेम्परेचर कंट्रोल्ड सिस्टम नहीं है, बहुत अच्छी साइंस है, किसी के पास अगर कंसेप्ट आ जाए कि हमको किसी तरह एम्बिएंट टेम्परेचर पे सैंपल को हम कोई चीज़ केमिकल डाल दें और उसकी वजह से हमको टेम्परेचर कहीं चेंज ना करना पड़े या टेम्परेचर स्टोर ना करना पड़े, तो समझ लो तुम जो हो उस दिन के एटलीस्ट नोबेल लॉरिएट हो चाहे ना हो, एंटरप्रेन्योर तो हो जाओगे और एक ज़बरदस्त चीज़। तो ये सब सोचो, जो आदमी इंजीनियरिंग बैकग्राउंड का है, इधर है, वो सोचे कि ऐसा कुछ बना सकते हैं क्या, हम ड्रॉप करते हैं, उससे इफेक्ट भी ना जाए उसका। आई एम ट्राइंग टू गिव यू आइडियाज, ट्राई टू थिंक ऑफ दोज आइडियाज फ्रॉम योर नेबरहुड और ट्राई टू अंडरस्टैंड फ्रॉम अक्रॉस द प्लेस। ओके ओके, दिस इज हाउ द बायोबिलिटी, दिस इज वेरी इंपॉर्टेंट कि आपकी बायोबिलिटी कितनी है। बायोबिलिटी क्या होता है, बायोबिलिटी कंपाउंड बॉडी के अंदर, हमने आईवी दिया, आईवी को हम प्रिज्यूम करते हैं कि 100% हम उसमें ड्रग चला गया। क्या चला गया, क्योंकि हम सीधे प्लाज्मा में दे रहे हैं, वेनस में दे रहे हैं, और ओरल की वजह से जो इतने बैरियर हैं इनकी वजह से कितना कम हुआ, उस रेशियो को बायोबिलिटी कहते हैं। जितनी अच्छी बायोबिलिटी, ओरल बायोबिलिटी, उतना अच्छा ड्रग है। अरबों रुपए लोग खर्च कर देते हैं, अरबों रुपए। सिप्ला जैसी कंपनी, जाइडस जैसी कंपनी, इंडिया की सारी कंपनी इसी पे काम करी, इसी पे जी रही है, इसी पे अरबों कमा रही है, सिर्फ बायोबिलिटी बढ़ाती है, फॉर्मुलेशन बढ़ाती है और कुछ नहीं, इनोवेशन उनके पास नहीं, यही काम है उनका। ठीक है, तो ये है कि हाउ यू वांट टू डू इट एंड हाउ यू वांट टू टेक इट। ये हम अपना एक प्रोटोकॉल तुम लोग को शेयर कर लेते हैं कि क्या है। देखो ये फॉर्मुलेशन 1 है, ये फॉर ए नैनो टेक्नोलॉजी एंड स्पेशली द टीम हु आर रियली इंटरेस्टेड इन फॉर्मुलेशन। ये देखो ये एक ब्यूटीफुल एग्जांपल है। इसके अंदर हमने क्या किया कि हमने एक पुअर बायोबल कंपाउंड था हैविंग ए गुड परमेबिलिटी एंड मेटाबॉलिक स्टेबिलिटी। हम उसको क्या किया, हमने कंपाउंड की सॉल्युबिलिटी को टेस्ट किया और उससे पता चला कि वो 10 माइक्रोमोलर सॉल्युबलेट है और उसकी जो परमेबिलिटी थी वो भी जो है उतनी अच्छी नहीं थी। तो हमने क्या किया कि हमने फॉर्मुलेशन ग्रुप को बताया और ख़ुद फॉर्मुलेशन डेवलप किया। पहला फॉर्मुलेशन है 280 मिथाइल सेल्यूलोज, वो सस्पेंशन फॉर्मुलेशन दिया, दूसरा सॉल्यूशन दिया, तीसरा पैक दिया। 3 टाइप के फॉर्मुलेशन दिए और देखो फ़र्क क्या पड़ा। पहले फॉर्मुलेशन में 6% बायोबिलिटी थी, फिर 84 हो गई, फिर 91 हो गया है। और यहीं पर अगर जो फॉर्मुलेशन पे काम करना चाहे नैनो टेक्नोलॉजी में, ख़ास तौर से पीपल जो वांट टू वर्क ऑन समथिंग गुड विद रिस्पेक्ट टू द formulation tube, they come up with a concept, they want to work some nanoparticle which can improve it, like sky is a limit. Okay? ये है प्री फॉर्मुलेशन की एक पूरी बड़ी टीम होती है, पूरा काम होता है। ये हम लोग की लैब है जिसके अंदर हम लोग काम करते हैं। दूसरी चीज़ है कि परमेबिलिटी। अब हम बता रहे थे इतनी बात कि परमेबिलिटी क्या होता है, व्हाट इज़ परमेबिलिटी। तो दिस इज़ हाउ इट इज़, दीज आर द स्ट्रक्चर जिसके अंदर हम दिखाते हैं कि ये छोटा मोटा स्ट्रक्चर है, इसमें ये आर है, ये एन है, इसको चेंज करो तो ये मिल जाता है, मिथिल ग्रुप है। तो पहली देखो सॉल्युबिलिटी, परमेबिलिटी इसकी सिर्फ 0.2 है, पैप 0.2 है, मगर बाद में 5.6 जैसे ही हुई तो देखो बायोबिलिटी कहाँ पहुँच गई, 24 से 84। ट्राई टू अंडरस्टैंड, 24 to 84 it has went. So this is an incredible change, but improving the permeability of a compound. कंपाउंड की परमेबिलिटी बढ़ी और ये सब, तो ये प्रूफ करता है कि जो इन विट्रो, दिस इज नोन एज IVIVC, क्या कहते हैं, इन विट्रो टू इन विवो कंपेयर। तो आप जब इन विट्रो, जैसे किसी कोई बच्चा हमको इन सिलिको काम दिखा रहा था पीछे, उसने जो काम इन सिलिको किया है, अगर थोड़ा सा उसको अगर सपोर्ट इन वीवो का मिल जाए या उसको सपोर्ट मिल जाए कहीं, उसका क्या नाम है, इंडस्ट्री का सपोर्ट मिल जाए, और इवन कोई डाटा अगर उसको रिसर्च पब्लिकेशन में मिल जाए कि ये काम हमने पहले किया है कि नहीं किया, किसी ने इसकी बायोबिलिटी चेक की, तो उसको प्रूव कर लेगा कि मैं इन सिलिको में जो दिखा रहा हूँ ये हैज़ प्रूव्ड, और फिर पेपर फ्लाई कर जाएगा। अभी तो कोई पढ़ेगा कहेगा एनी प्रूफ यू है, अगर मेरे जैसा आदमी के पास, रिव्यूअर के पास आएगा, मैं पहले ही क्वेश्चन पूछ लूँगा कि सब कुछ सही है, ये बताओ तुमने इसको कंफर्म कैसे करोगे कि सही है, सही है। This is how it looks when you talk of cell-parent dose, and this is important, this is very important. Somebody has asked this question. See, अगर ये ड्रग है, ये ड्रग कंसंट्रेशन अगर कम है तो इफेक्ट नहीं आएगा, अगर ये हरा वाला है, ऊपर जा रहा है और नीचे जा रहा है तो इफेक्टिव है, और ज़्यादा चला गया तो टॉक्सिक है। तो गेम इज बैलेंसिंग द कंसंट्रेशन। अगर आपका कंसंट्रेशन कम है तो आप घर जाइए, अगर कंसंट्रेशन ज़्यादा है तो पेशेंट मर गया। और आपको एक पर्टिकुलर एफिकेशियस डोज पे अगर मिल जाए, दैट इज नोन एज एफिकेशियस डोज, एंड इफ इट इज देयर मीन्स योर लाइफ इज सेट। So it's more of a game of a juggler, you know, but there is not 2 ball, 3 ball, there is 1000 ball, you have to balance it. Okay? When I take a ball, it means lot of data you have to do. This is what I play with that. This is known as LCMS, and this is instrument we have, and you all know well what is LCMS. Is the mass spectrophotometer, and this is mass to charge ratio which we talk. और इसी के अंदर जब हम इंजेक्ट करते हैं कंपाउंड को, इसका एक फ्रंट एंड होता है जिसको HPLC कहते हैं। ये पार्ट HPLC है, ये पार्ट HPLC है, ये LCMS है। एक होता है LCMS जो केमिस्ट्री वाले यूज़ करते हैं, और एक होता है LCMS-MS। तो ये 2 अलग अलग पार्ट है। फिजिक्स पीपल हु आर रियली इंटरेस्टेड, फोकस ऑन something in your science which with the instrumentation people, physics and those instrument, there is a lot of job people are creating there, and this is one of the science which is moving like something. money and something is huge. Okay, so focus on that part with these knowledge, because they usually hire product expert. So if somebody knowing a physics and if he knows these background of this biology, they are the champions for them. Okay, this is inside the mass spec. ट्रिपल कॉट के अंदर क्या होता है, ये सब 2 सिलेंडर Q1 Q2 होता है, फिर कलिजन एनर्जी कैसे निकलती है, कैसे हम ड्रग को इंस्टॉल करते हैं, और उसके बाद ये जो आयन फिल्टर, ये सब उसके पार्ट है। और मेरे हिसाब से कोशिश करना चाहिए ये इंस्ट्रूमेंट सीख लेना चाहिए। अगर कोई बायोकेमिस्ट्री या केमिस्ट्री का कोई है जान, तो उसको ये ट्रेनिंग ले लेनी चाहिए। ये अच्छे इंस्ट्रूमेंट हैं, ये बहुत काम देंगे, आजकल के स्टेट ऑफ आर्ट की ज़रूरत है। ये सब डिटेल्स है, आई विल नॉट गो। तो करते क्या है, पहले हम कंपाउंड को लेते हैं, उसको ट्यून करते हैं। ओके, ये ट्यून करके 174 उसका Q1 आ गया मेरा। अब Q1 480 इसका मॉलिकुलर वेट था, ठीक है, वो जो टूटा तो 170 पे आ गया है, और 132 दूसरे Q1, ये हमारा मेन 174 इज ए Q1। उसके बाद हमने क्या किया, उसकी क्रोमेटोग्राफी सेट की, और उस क्रोमेटोग्राफी HPLC से सेट करते हैं, और ये दिखाते हैं 1.2 मिनट पे हमारा रन टाइम आ रहा है, और हम उसको 1.2 मिनट का रन टाइम रन कर देते हैं। चाहते हैं ऊपर ले लें, नीचे ले लें, हम जल्दी से जल्दी चाहते हैं ताकि हमारे उतनी जल्दी सैंपल रन हो जाए, ठीक है। और उसके बाद ये हमारा असली जब क्रोमेटोग्राफी रन होती है, तो ये हमारे दीज आर द chromatograms which we get and see the peak. This is an ideal peak. Okay, जब पीक देखने लगते हैं तो ये पीक होना चाहिए। हम लोग बिल्कुल कहते थे बिल्कुल शार्प लाइक ए निडल। ओके, और इसके अलावा होते तो हम रिजेक्ट कर देते थे, लोग दिखाने आते थे। तो ये रन आता है, कंसंट्रेशन का रन होता है, इस तरह का एरिया आता है, एरिया रेशियो आता है, स्टैंडर्ड आता है, और क्रोमेटोग्राफी रन, ये स्टैंडर्ड चल रहे हैं, नीचे अननोन सैंपल्स हैं, और यहाँ पे हमने गोला इसलिए किया है कि इससे दिखा रहे हैं कि इंटरनल स्टैंडर्ड वेरी कर रहा है, तो ये हम रन को रिजेक्ट कर देंगे। उसके बाद फिर ये, तो ये इतने इंस्ट्रूमेंट मेरी लैब में हैं जो हम लोग काम करते हैं। ये अभी अब और अपडेटेड हो गए हैं, इतने इंस्ट्रूमेंट हैं हमारे पास, इतने LC-MS हैं, HPLCs हैं, और ये सारे हम लोग अपने एक्सपेरिमेंट के लिए रन करते हैं। ये सॉफ्टवेयर्स हैं, किसी को अगर शौक हो मैथमेटिक्स का कोई है या कोई इंटरेस्टेड हो, तो यू इनॉलिन, ग्राफपैड, गैलीलियो, दीज आर द गुड प्लेसेस वेयर यू कैन लुक फॉर ए good options they are looking for. अब एडवांस हर चीज़ होती जा रही है, तो these are the ways. Now AI has also done what, you know, these ChatGPT, even Capitol, they have done. They have used these Winolin and GraphPad and they have attached to their own system. Now you put the question there, they will attach and give the answer from them also. So future is there, technology, you know. Don't sit and think of it. Now tomorrow once you go after this, you should think novel, you should think new something. We can do it. और उसी में पैसा है, ये नहीं है कि हम चले जाए ये कर ले, ना ना ना, कुछ ऐसा करो अलग सा। Now there is a meta ID. This is a beautiful science. मैं बहुत सोचता रहा दूं कि ना दूं, फिर बाद में मेरे I was having something which I wanted but I removed it due to some. This was a paper I published around 12 years back or 13 years back. I published this paper, and this is a meta ID paper, and these are the all the structure elucidation and how the number which we get, what are the peak which we get, how much the masses we are getting. What is meta ID? You know, meta ID is nothing. When you put a compound, it is get metabolized. When it metabolize in a form of a small, one of the metabolites. Okay, and compound do have lot of metabolites. Now our job is to understand what are the metabolites coming, because they will remain in the body and they can be toxic. Your parent is not toxic, but after metabolism it is toxic. Your CYPs metabolize the compound and it makes metabolite. So your job is metabolite identification. A beautiful science, a fascinating story of MetID, and if you get an expertise, there are very lesser expertise in the world. So if you climb on this expertise, you guys will be new to this field and people will love to hire those guys. But that needs a knowledge of chemistry, needs a knowledge of biology, needs a knowledge of instrumentation. Now the last part is safety. Okay, we talked of activity, we talked of drugability. Now the last topic is toxicology. All things are poison, you all know well, and nothing is without a poison. Only the dose permit something not to be poisonous. तो कितना खाते हो, शुगर भी 1 चम्मच खाओ तो ठीक है, पूरा खा लो तो प्रॉब्लम है, ठीक है. तो हर चीज टॉक्सिक है, डोज डिसाइड्स. दिस इज हाउ द पाथवे लुक्स फॉर इन ड्रग डिस्कवरी डेवलपमेंट. पहले हम इन सिलिको, आज कोई बात कर रहे थे कि इन सिलिको एनालिसिस कर सकते हैं. Still it's too nascent, people are trying, they have some data, because the टॉक्स के साथ अच्छी क्या है, उस पर डायनेमिसिटी कम है. जो टॉक्स होना है वो होना है. लीवर में टॉक्स है तो ALP बढ़ेगा और दूसरे लीवर एंजाइम्स बढ़ेंगे, कुछ ना कुछ ये आगे चल के अगर कोई बहुत ज़्यादा हिस्टोपैथ में जाके देखोगे तो कैलेंजियोफाइब्रोसिस हो जाएगा, ये सब होगा, ये होना है. तो अब वो टॉक्स वाले क्या करते हैं कि उनके पास डाटा कम है, इफेक्ट का डाटा कम है, कॉज का ज़्यादा हो तो अलग चीज़ है. तो व्हाट दे डू, दे मेक एन इफेक्ट एंड दे कोरिलेट विद द मॉलिक्यूल स्ट्रक्चर एंड सी कि ये जो हाइड्रोक्सिल ग्रुप था या ऑक्सीमिथिल ग्रुप था या नाइट्रोजन ग्रुप था या डाइमिथिल ग्रुप था, ये कैसे ये ग्रुप जो है ये यहाँ पे एक्ट कर रहा है. तो उनका जॉब हो गया है आसान और उसकी प्रेडिक्टिबिलिटी बढ़ गई है. देखो प्रेडिक्टिबिलिटी की प्रॉब्लम क्या होती है, जहाँ डायनेमिसिटी ज़्यादा हो. टॉक्स एक चीज़ पे डिपेंड नहीं करता है, इसलिए इट्स नॉट ए केक वॉक. उसके बाद डिस्कवरी में हम लोग नॉन GLP पहले ही चेक कर लेते हैं कंपाउंड है कि नहीं. फिर IND में इम्पॉर्टेंट है, उसके लिए हमको GLP सेटअप चाहिए होता है. उसमें जो है हमें जीनोटॉक्स चाहिए होता है, इनविट्रो टॉक्स चाहिए होता है. फेज 2 में इन विवो करना, क्रोमोसोम टेस्ट, डैमेज टेस्ट, MTD, इट इज़ नोन एज अ सेफ्टी मार्जिन. हम NOL निकाल लेते हैं फर्स्ट इन ह्यूमन डोज में. अलाउ नहीं करेगा रेगुलेटरी अगर हमने उसको बताया नहीं कि आपका टॉक्स कैसा काम कर रहा है. स्ट्रेटेजिकल टॉक्सिक प्लानिंग, ये सब डुअल स्पीशीज, 2 स्पीशीज में टॉक्स करना पड़ता है, एक रोडेंट और एक नॉन रोडेंट. रोडेंट में हमें करना पड़ता है या तो हम रैट में कर लें, माइस भी लोग नहीं पूछ रहे हैं, रैट ज़्यादा क्योंकि उसके डाटा बहुत है मार्केट में. और दूसरा या तो डॉग में कीजिए या मंकी में कीजिए. 2 टॉक्स अगर नहीं करेंगे तो आपको क्लिनिकल ट्रायल कोई करने नहीं देगा. ये टाइमलाइन होती है, शुरू में 102 दिन सिंगल टॉक्स, 7 to 28 डेज में क्रोनिक टॉक्स, और उसके बाद 90+ डेज की हमें क्रोनिक टॉक्स स्टडीज़ की लॉन्ग टर्म करनी पड़ती है. की एंड पॉइंट मेजरमेंट, एथिकल, ये सब इम्पॉर्टेंट है. ये मेन पैकेज है कि इतना पैकेज हमको देना पड़ता है ये सारी स्टडीज़ करके. जब सारी स्टडीज़ हो जाए, टॉक्स अच्छा हो, उसके बाद भी ये डाटा हमें कैलकुलेट करके उनको देना पड़ता है. तब ये ऊपर जो है वो रेगुलेटरी एजेंसीज़ हैं, US, EU एंड जापान, इनमें कहीं भी दो तो ये सब रिक्वायरमेंट है. और I don't want to go in that. This is a good news for everyone sitting here that now FDA announces to phase out the animals testing requirement, monoclonal antibodies and the other drug. Subsequently, it will come back to other drug also. अभी मोनोक्लोनल में है. तो ये रोड मैप पे वो ये कहना चाह रहे हैं कि आप ऑर्गन ऑन चिप टेक्नोलॉजी, अगर कोई बहुत फैसिनेटिंग है, ऑर्गन चिप आसान है, सस्ता भी है ऑर्गन लेना, तो उसके बाद टेक्नोलॉजी आपके पास इतने अच्छे यहाँ पे इलेक्ट्रॉनिक्स के और इन सब के लोग हैं, इनको साथ आप काम करो और कुछ नया सोचो कि हम कैसे सिग्नलिंग उसकी ले लें, किस तरह कर लें. और एक क्रॉस फंक्शनल डिपार्टमेंट अगर कर लेगा ना, आई थिंक वी कैन ब्रेक द आइस. लॉट ऑफ पीपल आर डूइंग, देयर इज अ लॉट ऑफ साइंस गोइंग ऑन. फिर उसकी फिजियोलॉजी एनहांस हो जाए. लिमिटेशन ऑफ फोकस क्या है कि हम एनिमल टेस्टिंग को कम कर लें, और FDA अब सपोर्ट कर रहा है. ये डाटा अगर उसको दे दो तो आपकी टॉक्स स्टडीज़ कम कर देगा, ज़्यादा नहीं कम कर देगा, रोकेगा नहीं मगर है. उसके बाद ये ह्यूमन डोज जो हम लोग करते हैं, तो ये मेरा काम है कंपनी में. अब तो हम जिस लेवल पे हम यही काम करते हैं, हम ये बताते हैं कि आप कितना पहली बार ह्यूमन को डोज दो तो बचेगा, मरेगा नहीं. दिस इज नोन एज फर्स्ट इन ह्यूमन डोज कैलकुलेशन. ये हमको रैट से मिल जाता है कि NOL कितना है, डॉग से मिल जाता है कि कितना उसकी STD है, उससे हम कैलकुलेट कर लेते हैं. ये हमारा कोई ड्रग होगा, हमने तो नंबर सब बदल दिए हैं, तो इसलिए जो है ये कि आप 20 mg दे दो तो वो सही रहेगा. 20 mg मरेगा नहीं. अब उसके बाद 20 mg जब 3 पेशेंट दे देते हो तो 3 पेशेंट नेक्स्ट उसके 28 डे की साइकिल होती है, किसी की 21 डेज की, डिपेंड कि आप क्लिनिक में कैसे यूज़ करोगे। फिर आप उसको कोहोर्ट, इनका कोहोर्ट देते हैं, उसके बाद देने के बाद एक नेक्स्ट कोहोर्ट आता है, फिर आप 20 से 40 में चले जाते हैं, 40 से अब आप अपने डाटा से देखते हैं कि 40 जाना है, 60 जाना है कि 80 जाना है, तो आप नेक्स्ट पे चले जाते हो 80 में, जब तक कि आपको एडवर्स इफेक्ट नहीं दिखते आप जाते रहते हो, और एक स्टेज पे जब एडवर्स इफेक्ट दिखते हैं तो आप रुक जाते हो, उसके भी 3 पेशेंट और करते हो, करने के बाद उसको रोक देते हो, फिर उसके बाद डोज एक्सपेंशन स्टडी होती है, वहाँ फेज़ 2 शुरू हो जाते हैं, और फेज़ 2 के अंदर जब MTD RP2D कहते हैं वो जब रुक जाता है तो फेज़ 2 में आप एफिकेसी देखते हो, अगर एफिकेसी अच्छी है तो फेज़ 3 में एक बड़ा सा ग्रुप लेके किया जाता है, डिफरेंट प्लेसेस का ग्रुप, वो अच्छा है तो आपका ड्रग समझो सक्सेसफुल हो गया, और आप फिर फेज़ 4 में जाते हो, फिर एक नई स्टोरी है। Important message, it takes a village to create a file an IND to FDA on any regulatory। तो अगर एक ड्रग बनता है तो उसके लिए समझो एक शहर बसाना हो गया, ठीक है ना, तो एक ये शहर बसता है, इतने सब इंफॉर्मेशन आपके पास होनी चाहिए। ये रिक्वायरमेंट है GLP टॉक्स की, वो हम अब नहीं करेंगे, so we already spoken much more on it। So the thing is, we reach to the conclusion, first and foremost we know how the molecule are made, how the chemistry works, that already been explained, what are the stages of drug discovery, why there is attrition in drug discovery, what are the reasons for that, there is another arm to it, one is the activity, other is the drugability, and third is the safety. We talked of activity morning session and yesterday also, and it will be continued sometimes, and others will also be working on it now with some specific target like a cancer and something he will be working more, talking on to you guys. And then is the drugability, where you absorb the drug, excreted, it get distributed, then you are asked of ADME, and the third portion which we talked of, it was safety, which we talk of a tox, how the tox is done, what are the reasons for that, what are the coming signs which people are advocating these days with the regulatory stuff. And then we talked of a regulatory, how it is easy to. So most importantly, it's a cumbersome process with a lot of science, with a lot of cross activities involved in it, and lot of interdisciplinary focus on it. And I think we can keep to the rest today with the next lecture.
So what can we do to increase the absorption in the body?
Yeah, actually, first you should know the problem. Okay, problem is whether it's an absorption, and absorption can be sorted out why it is absorption. There can be two reasons. One is it's not permeable, compound is. So what we usually do, where I usually analogy and other, we make a micelles, we make something in such a way that the compound is encapsulated inside and it goes and get opened. Okay, that's a one way, easy way. Second is whether it is due to the transporter effect, means your drug has been thrown out of the system. There is a PGP, you know, PGP, have you heard? Have you heard of PGP transporters? There are two types of transporters, I told you, one is the active, another is the passive transporter. There are some active transporter which is the motor transporter which throws the compound outside, and there is a lot of things. So important is where the problem is absorption, what is the problem, then you understand, you can formulate in such a way so that it can be given, or is going inside the body and getting precipitated out also. So that is dissolution issue. So you have to understand. So this all is story, millions of dollars people spend just to make sure that know the problem, and there are the solution which are there, where the new field comes and they do it and you sort out that issue. No problem. Yeah, go ahead.
You are developing or trying to modify different functional groups and trying to figure out that which structure will be suitable for drugability. But at that aspect, how we are taking help from plants, that different components, we know medicinal plants are there. So
that is what the question which I answered earlier, somebody was asking from that you have to get an active constituent from that, and most of the best cancer drug example, which was Glivec, if I remember, or something, which was done from a plant extract only. So doxorubicin was one of the example which is there. So important is get the the right chemical entity which is acting on it, and you have to do the same studies. Okay, any questions? Yeah.
So nowadays focus is being given to environmental health. So is there any such aspect being covered during the drug development besides the toxicity of human health?
Yeah, we are all succumb to that pressure from government also. And I think we make sure that our chemicals, especially radiochemicals and all, are put in a. We follow the rule from the Bach society they give. So that's a given thing. You know, you can't live in India. That's the biggest problem than anything else. So you have to do it, and we try to maintain as such as possible.
All right, so
Thank you, sir.
thanks to you. लेकिन अगर आपने ये तस्वुर किया था कि आप एक अच्छे टीचर नहीं है तो I would disagree with you. How many of you agree with me? So let's give him a big
compliment from you.
Yes, take it. All right, so we are moving towards the last session, the most difficult session. You are tired, sleepy. People have come from Varanasi, and you came today morning, right? Okay, so, but I can assure you, you will all be able to connect to this lecture. So we have been talking about the drug discovery and drug delivery in general, right? Today I'll try to give you a flavor of specifically a disease called cancer. How many of you are familiar with cancer? I think all hands should raise up, right? Is there somebody who doesn't know CAN, CER? Everybody knows CAN, CER. Cancer is a disease, and unfortunately, some of you would have experienced it through somebody whom you know, right? So I took up my career in cancer research because of these reasons. Otherwise, I did vaccine development in my PhD. I had developed a vaccine during my PhD thesis. But then I moved to doing the cancer research, and for the past 22 years, I'm doing cancer research. That kind of focus. So I'll try to bring some cancer story from the drug development perspective, and here's an outline that I'll be talking about. So within this outline, because cancer is a big field, it's a very diverse field. So I'm trying to, while I was looking at my presentation, it was hard to skip क्या रखूं, क्या छोड़ूं, क्या चीज़ बताऊं, क्या चीज़ न बताऊं, because there's a lot of things that look interesting and I want to transfer it to you. So introduction, I think we can have a quick overview that from you guys. What is cancer? Can anybody help me with that? Uncontrolled growth of cells. Anybody else has any other definition? Everybody says the same, right? So uncontrolled growth of cells. But this uncontrolled growth of cell, if it just forms a tumor and it stays in its place, it's not cancer, right? It's just like warts. कभी कभी मसे हो जाते हैं ना, मसे भी यही हुए, skin cells जो है uncontrolled growth हुई, but then they did not spread. So if they don't spread, they are benign and they are not cancer. So the definition is incomplete when you say uncontrolled growth of cells. It's uncontrolled growth of cells with if it is spread to other locations, then it is called metastasis. If it is even confined within the same place, but it is spreading, it continues to spread without any kind of intervention, then it's cancer. Then it is qualifying as cancer. So there was some, this definition, as Dr. Imad, was not just one disease, it's a kind of a syndrome. There are multiple diseases. So it was hard to find out how to say what causes cancer or how can we define a cancer cell. So then there was a seminal paper that I like to explain the slides fully. See here, nothing. Okay. Let me see. So in cancer cells, it's like a vehicle oncogenes, and we also have the tumor suppressor genes. Oncogenes are like accelerators, and tumor suppressor genes are like brakes. So, and then we have got the growth suppressors. So there are things in the body that suppress the growth. They say, okay, you have got enough, now you slow down. But cancer cells are able to evade these growth suppressors. They also evade the immune destruction, because at a point they become non-self. They are not like the normal cells. So immune system tries to filter out these cells, but they are smart enough to avoid those immune destruction as well. They have replicative immortality. That means they are continuously multiplying. Can we tell me replicating? Exactly. So telomeres are the indicators that how many times a cell can divide, because every time it is getting shortened, and then ultimately cell is doomed to die. But in case of cancer cells, they have got a mechanism by which they keep elongating the telomere, inflammation that helps these cells to grow. This creates an environment which is helping them to grow. And it also helps them to invade and metastasize. Invasion means going to different territories and landing there and sprouting up new tumors. They also need, when they reach to new places, they need more supply. Right? If somebody is going to inhabit in a desert, they will need the supply also. How does the supply come from? Increased vasculature. The new blood supply is coming in. New blood vessels are forming in. I think it will look like more of a cancer biology class for you. But no, I'll move to the drug development soon. Then you also have the genome instability and mutation, because a normal DNA would function normally. So it has to be something different that cancer cells have to develop in their DNA, by which they can have these capabilities. Then also they resist the cell death. So again, the markers like apoptosis, autophagy, senescence, necrosis, all these are being avoided by the cancer cells. And the cellular metabolism, they deregulate the cellular metabolism. The normal process of energy generation is compromised. Warburg effect. They adopt a mechanism of energy production which is the least smart method. That's called Warburg effect, by which they are generating energy, and the reasons are still unknown. Right? So they have been summarized into this picture where there are senescent cells as well, and polymorphic microbiomes, which is the gut microbiota that we are talking about. Changes which are known as epigenetic changes that are also happening there, and plasticity. Plasticity means they are able to change their shape, their format, and this helps them to grow, survive, and evade the immune system. This is the plasticity. Again, I'll be happy to stop and answer anything that you have. Plus, you can also have talked about these tomorrow as well. So once we know what defines, started to take them one by one and hit them with the targets. So if you remember my yesterday's lecture, the first thing in the drug discovery was target identification. So first thing is to know where to strike. ड्रग्स तो तब आएंगी जबकि पता होगा कि कहाँ पे हमें उनको हिट करना है. So for target identification, we have to know what the cell is looking like, and for all of these hallmarks, now you have got the EGFR inhibitors for sustainable proliferative signaling. So they are inhibiting each of the hallmarks here. So what all we discussed, now there are different classes of drugs which are acting on these hallmarks to stop the growth of the cancer cell. Clear? Now this is a slide which helps to tell you about what are the major risk factors for cancer. So I'm sure all of you are aware of these, that tobacco is one single agent that is causing a lot of cancer, and this has been established. Then you have got different lifestyle stuff as well as some of the chemicals and radiations, etc. I will not go much into the epidemiology, as all of you acknowledge that this is a big problem. And even in countries like ours, this is emerging. The recent report from Apollo is saying that India is picking up at pace with the developed countries. So we are developing, and we are also taking the statistics of those countries. Because usually cancer was supposed to be the disease of the developed countries, but now India is also picking up and going there. And most of these cancers, so this is called incidence, that how many cancers are developing, and then mortality, how many people are dying because of those cancers. So here is some data here, and most of them you can see is upper digestive tract, lung is supposed to be the most biggest killer, breast cancer, colorectum, liver, prostate, and others. This is the global scenario, but we have got the similar data from India as well, right? I don't know why this is not right. So we have got here introduction and challenges of treating cancer, and now we'll move to the next one, which is what are the scope of doing research in cancer. So one of the biggest problem in cancer is the heterogeneity, right? Heterogeneity means that cancer is not one, heterogeneity means there is not one target. We think that we found a target, but no. The cancer cells are all different. Even in one tumor in one place, the cells are different. And now this is called intratumoral heterogeneity. Now you talk about tumor in one place versus tumor in another place. They are also different. This is called intertumoral heterogeneity. Then you have got one patient is different from the other patient. This is interpatient heterogeneity. So there are several levels of heterogeneity which is making cancer so complex that even after so many years we don't have a proper cure. So tumor heterogeneity is one aspect. Then we have the tumor microenvironment. The environment along with the cells that are different to begin with, it's adding to the heterogeneity of these cancer cells as well. So the environment is helping them to grow, and environment is making them survive even in the presence of the immune system. So it supports the tumor growth, invasion, immune evasion, and metastasis, which is the spread of the cancer to other organs. This is a snapshot of what are the different cells along with the cancer cells that make up the tumor environment. So you have got here the immune cells, dendritic cells, fat cell, apoptotic cells, which are the normal dying cells. Also you have got the natural killer cells. So there are many cells that are coming together to help the cancer to grow. Right? So this is another way of looking at it, is the same. These are the components of the tumor microenvironment. It also involves fibroblast, extracellular matrix, vasculature, which is the new blood vessels, cytokines, growth factors. All of them are helping the cancer to grow and helping in the heterogeneity of the cancer, which makes it difficult to target. But cancer cells, we develop drugs, and cancer cells are really smart. So even if we are with all these complexities, we are able to find a drug and target them, shoot them. They develop new ways to, they learn, and they develop new ways to evade that in the next time. And that is what is known as relapse or drug resistance. So there are a lot of patients who are known to have first cycle, they show very good results, second cycle, third cycle, now resistant. The drug is no more working. Same drug which was effective earlier is no more effective. That's another problem with the cancer cells. So it is due to the drug resistance, and there are several mechanisms to it, because the tumor is evolving in terms of its own hallmarks as well as the environment around it. It keeps changing. So earlier there was a thought कि अगर हम ट्यूमर की ब्लड सप्लाई काट दें तो ट्यूमर मर जाएगा, right? This was their idea. But it was observed that if you cut down the blood supply by using some inhibitors, it become more aggressive. Now they're not there, they are spreading to other places. जब तक blood supply थी तब तक तो वहाँ रह रहे थे, अब वहाँ से blood supply काट दी तो they are not spreading. So it was more disastrous. So that's why this drug resistance and tumor evolution is another big problem that needs to be tackled. And this drug resistance is coming through different methods. It could be due to mutations in the DNA. It could be because the drug is pumped out. It could be due to the inactivation of the cell death pathways, or it could be because of the better repair mechanism that these tumor cells have. So it could be many mechanisms by which this cancer cells are developing their resistance towards the drugs. So drug बनने के बाद भी काम नहीं कर रही है. अभी आपने जितना भी सुबह से शाम तक सुना, बन गई सारी toxicity studies हो गई, सब हो गई, but cancer cells are smart, they develop resistance. So we have to keep in mind that we have to treat in such a way that we are not making the cancer resistant, rather we are completely killing it. Yes, go ahead. Yes, so what happens when you are cutting down the cell supply, these cells, these tumor cells, they evolve, and the one that survive, there's one cancer stem cell theory here, that they survive, and the ones that are surviving, they spread, and now it's more aggressive. So I'm telling you the clinical story, because as soon as you cut down the supply, it's not that everything is, all the cells are killed. I mean, it's like a ball. So all of them, they don't kill at once. The ones that survive, they are more dangerous. Yeah, they learn how to utilize less energy. They go dormant. Yes, and this time they move to other places and find a new destination. Right. Nevertheless, we cannot sit down and say we can't do anything. We have to go for finding out new ways to combat this heterogeneity, this drug resistance, and that's where the drug development process continues. Is it the same for cancer drugs? What do you think? Finding a target, doing the preclinical phase 0, phase 1, phase 2, phase 3, phase 4, is going to be the same for cancer drugs? I mean, the stages are the same. The process is the same, but everything is different when you are talking about the cancer drugs. Starting from the target identification to the final phase 4 studies, they're all different. Right? So for the target identification, we can have multiple ways to do the target identification, and most of my work is here for the target identification. We try to find out the genes that are responsible for these cancers and how can we target that. But beyond that, it's the work of the chemists to synthesize the molecules which can hit the target, or the biologists who can make the antibodies to kill that target. But most of my research, that's for identification of these targets. So it involves different omics. That's another thing that I would like to bring up, that once we say that a cell is transforming from a normal to a tumor cell, it is not just a function of DNA. Mutation ho gaya, disease ho gayi, right? Rather, it's a function of many molecules that are present inside the cell, which could be DNA, RNA, protein, metabolites, and when you study them all together, it's known as omics. Genomics is about the DNA, transcriptomics is about the RNA, proteomics is about the proteins, metabolomics is about the metabolites, and there are many more omics that needs to be combined together to understand one single cell. And then only we can come up to the right target. I always tell the story of this elephant. I'm sure some of you might have heard about it. कि एक अंधों का एक गांव था, उसमें एक हाथी आया. अब वो रात में आया तो आवाज़ हुई, तो सबने जाकर के उसको देखना चाहिए है क्या ये चीज़, कहाँ से ये आवाज़ आ रही है. तो सबने उसको जो है, किसी ने उसको छू के देख के समझने की कोशिश करी। फिर सुबह सब बैठे कि भाई क्या है, तो हर एक ने अपना अलग डिस्क्रिप्शन दिया। किसी ने कहा कि ये एक चट्टान की तरह है बिकॉज दे टच द ट्रंक। कह नहीं तुम गलत कह रहे हो, मैं बता रहा हूँ ये पंखे की तरह है। उसने उसके जो है कान छुए थे, तो क्या तुम लोगों ने लगता है कोई और चीज़ देखी है। मैंने देखा वो तो एक सांप की तरह था और रस्सी की तरह था। हालांकि द फाइनल पिक्चर वॉज, इन सब का एक मजमुआ था, इन सब का इट वॉज अ कॉम्बिनेशन ऑफ ऑल दीज फीचर्स एंड ऑल ऑफ देम वर थिंकिंग दैट वी फाउंड, वी अंडरस्टैंड द animal or the thing that has entered into our village, but all of them were wrong. This has been happening in cancer research as well. People who found the mutations 20 years ago, they said, we found it. P53 mutation, it is mutated in the cancer cells, we are going to fix it, cancer done. Wrong. Then they said, no, this gene is overexpressed, we'll bring it down, we have found the gene that is causing cancer, we found it. They were wrong. Proteins, phosphorylations, glycosylations. So I'm trying to sell this idea that it's a combination of all these things that is known as multi-omics, which can define the cancer cells, and then only you can find a good target that can defeat cancer as we know it. And for that, we need to have the full idea of all these omics, and then there's something called biomarkers. I'll be coming up in the later slides, and that leads to personalized medicine. Because as I told you, interpatient heterogeneity is a lot there, so we cannot apply one-size-fits-all approach in cancer at least. But unfortunately, we are not yet there. So target validation, I think you have heard about it a lot. We have got animal models, expression profiles. There are different ways of doing the target validation. Target validation means पता करना कि क्या ये वाकई जो हमने target मालूम किया है ये सही है भी कि नहीं है। इसके लिए मुख्तलिफ protocols हैं, and we do that. I think you've heard a lot about the target validation. Then it has to be an iterative process. So we learn every time we take something to the clinic, to the bench side, we see the response of the patient and come back and feed it into the clinical protocols as well, so that experimentation and therapy, they keep on feeding each other. And that's where the AI is helping a lot, and continuous learning models. And this is also, these multi-omics approach is also possible because of the AI stuff. Otherwise, how many bases are there in our DNA? My students should tell at least. No, bases, ATGC, I'm not talking about ATGC. How many are there? There are 4 types. Somebody said a number. 3 billion bases are there. So just imagine now we can sequence this. We can say ATGC 3 billion, and this is one set of DNA, how they are arranged. And then you have got the RNA, then you have got the proteins. So all these data points, they become millions. So it's beyond human comprehension to identify a target in all this. So machine has to come in and help us in finding out. Right? So next the pattern of clinical trials, once we have got the target, we have got the lead molecules, we have got the target validation, we go into the clinical trial. I'm skipping those chemistry and the formation of the drugs and stuff. I'm going straight into the clinical trials. That suppose these chemists and everybody we found the target, they synthesized the molecule, and they found that preclinical was there good, then how do we move on from here? And again, you have heard about these phase 1, 2, 3, 4 trials multiple times. I hope now you memorized it. There was one more phase that I talked about day before yesterday. 8-0, very good. So in cancer, there are other type of trials. As I said, process same, but story is different. There's something called umbrella trials. Umbrella trials में करते हैं कि एक disease कैंसर है, उसके अंदर सब ग्रुप्स बनाए जाते हैं. सब ग्रुप्स मतलब किसी को अगर कोलन कैंसर है, उस कोलन कैंसर के अंदर कुछ बायोमार्कर्स निकाले जाते हैं और उन बायोमार्कर की बेसिस पे पेशेंट्स को क्लासिफाई किया जाता है. जैसे कोलोरेक्टल कैंसर में इस वक्त जो क्लासिफिकेशन चल रहा है वो है कंसेंसस मॉलिक्यूलर सबटाइप्स, CMS 1, 2, 3, 4. There are 4 types. And then you drug them accordingly. You don't drug all colon cancer patients with one type. No. You do subgrouping. And after subgrouping, you give them the dosage and drugs separately. So they are given different types of drugs for the same disease. This is called umbrella trial. And the reverse of it is called basket trial. So one example of this umbrella trial is lung map trial. This is an example that has been done. They are trying to find out which subgroup of the lung cancer patients will be benefited by which drug. So now lung cancer is separated into subgroups. So and it's basically the genetic makeup that is being considered, and then there are different sub-studies here. And this trial has been going on for now more than a decade to collect data and finally come up with a protocol for different type of patients. And the opposite of this is the basket trial. Basket trial is any person who has having a particular mutation, for example, BRAF mutation. BRAF is the gene. It has got a mutation, valine converting into glutamic acid at position number 600, V600E mutation. So anybody having this mutation will be treated by using a particular drug, whether it is lung cancer, whether it is breast cancer, whether it is colorectal cancer, it doesn't matter. So it is tissue agnostic. Tissue does not matter, rather that particular mutation matters. So I've given here the example for the basket trial, the BRAF inhibitor in patients with BRAF V600 mutation. Now this mutation is present in melanoma, the skin cancer, colorectal, thyroid, non-small cell lung cancer, and even some other diseases, Langerhans cell histiocytosis, and Erdheim-Chester disease. So if anybody has got this particular mutation, they will be given this drug, right? So it's tissue agnostic. Then there's another type of trial. So umbrella trial, basket trial, then N of 1 trial. Why? Every patient is different. N of 1 का क्या मतलब है कि एक ही पेशेंट, वरना यूजुअली ट्रायल्स होते हैं, आपने कल नंबर्स क्या देखे थे? फेज 1 में कितने होते हैं? फेज 1 10, yes, 10 to 100, yes, I heard 10,000. And phase 2? 100. Phase 3? और इसमें यूजुअली क्या होता है, स्टैटिस्टिक्स चलती है भाई 10 पेशेंट थे, 10 में से 6 में अगर अच्छा रिस्पांस आ गया, ड्रग इज गुड. बट ये सोचना है कि जिन चार लोगों में नहीं आया था, व्हाट वाज द रीजन देयर. या लुकिंग ऑन द ऑपोजिट साइड, सपोज 10 लोगों में से 2 में रिस्पांस आया, स्टैटिस्टिकली तो वो ड्रग फेल हो गई, 2 out of 10. बट उन 2 में रिस्पांस क्यों आया? व्हाट वाज देयर? What was with those patients? So this is being captured in N of 1 trials. So every patient is a trial in itself. इसके लिए करते हैं कि they take 2 drugs. पहले drug A दी, response देखा, drug B दी, response देखा, दोनों में से जो बेहतर लगी उसको continue किया. So this is how they use the same patient as its own control. Otherwise randomized control trials वगैरह जो होते हैं उसमें बड़े बड़े कोहोर्ट्स लिए जाते हैं और फिर स्टैटिस्टिकली अगर वो सही निकला तो माना जाता है, नहीं सही निकला तो द ड्रग इज किल्ड. So this is the traditional approach: target validation, compound screening, lead optimization, preclinical testing, phase 1, phase 2, phase 3, phase 4, and then post-marketing stage. Whereas in N of 1, there is already pre-existing platform knowledge that what are the mutations in the patient, what is the protein profile of this patient. Then the design is, and this is pretty much expedited. If you can see, the timeline here is much lower. So you start testing 2 drugs, and then these 2 drugs, you found that this drug is better. Or there, in some cases, they also do 3 drugs, and then you say, okay, out of these 3, this one is good. We'll start with this for this patient, right? This is N of 1 trials. And then I told you this story yesterday. even for the screening of the drugs, there's a fly avatar that I had. My friend, he's now relocated to Ireland. He used to work in Mount Sinai in New York. He developed these Drosophila. So we used to sequence the patient का जीनोम पूरा सीक्वेंस किया, उसके देखिए कौन कौन से म्यूटेशंस थे, और उसी म्यूटेशन के साथ फ्लाई एवाटार्स बनाए ड्रोसोफिला के। तब क्या हुआ, हमारे पास सोचे छोटे छोटे ह्यूमन बीइंग के जीनोटाइप हैं। देन वी टेस्ट द एंटायर FDA अप्रूव्ड ड्रग्स। उसमें से पता लगा 5 ड्रग्स जो अच्छा काम कर रही हैं। वी हैंड इट ओवर टू द डॉक्टर कि दीज आर द 5 बेस्ट प्रिस्क्रिप्शन फॉर दिस पेशेंट, हाईली पर्सनलाइज्ड। Now this company is Vivan Therapeutics के नाम से एक कंपनी अब लंदन में है। Laura, she has bought this technology and now she is doing it. But the major hurdle here is money. I wanted to get it for some of my friends, patient here, but I couldn't get it. It was very expensive. It starts from $100,000 starting. So this is another aspect of drug development which we haven't covered in this. The economics, that many of the drugs that are available, they are not accessible due to money issues. So then here there's an example of this fly avatar models where they have utilized a patient from acute myeloid leukemia, and उसके पास usually ये उस तरह के patients में करने की इजाज़त मिलती है जिनके ऊपर सारे other options ख़त्म हो चुके हैं, phase 4 वगैरह में पहुँच गए। तो एक AML पेशेंट था जिसके सारी थेरेपेटिक अप्रोचेस के ऑप्शन ख़त्म हो चुके थे। इसके पास ETV6 and TREC2 ये जीन फ्यूजन है जो कि इस पेशेंट में था। सो देन दे प्रिपेयर्ड द फ्लाई एवाटार्स एंड दे फाउंड आउट दैट दिस पर्टिकुलर ड्रग लारोट्रेक्टिनिब वाज द बेस्ट ड्रग दैट इज ऑलरेडी अप्रूव्ड। सो यहाँ पे अब अप्रूवल का चक्कर भी ख़त्म। सो बिकॉज दीज ड्रग्स आर ऑलरेडी अप्रूव्ड बाय द FDA। तो टॉक्सिसिटी स्टडीज इनकी हो चुकी हैं, फार्माकोकाइनेटिक्स इनकी हो चुकी है, DMPK इनकी हो चुकी है, तो दे आर रेडी टू यूज इन द पेशेंट्स। ऑल यू आर डूइंग इज knowing which patient is going to get the best response from which drug, and the patient achieved the partial remission also. So it was a success story. For doing this, there is a very big market, very big area of biomarkers. बायोमार्कर्स हर उस चीज़ को कहते हैं जो कि हमें किसी हेल्थ कंडीशन के बारे में बता रहा हो। ब्लड प्रेशर इज़ अ बायोमार्कर, जो कि आपकी कार्डियक हेल्थ के बारे में बता रहा है। ब्लड शुगर, दैट्स ऑल्सो अ बायोमार्कर। तो इस तरह के बायोमार्कर डिस्कवरी जो कि स्पेसिफिक हेल्थ स्टेटस को बता सके, दिस इज़ वेरी मच नीडेड। पर्सनलाइज्ड मेडिसिन बगैर बायोमार्कर के हो ही नहीं सकती। कैसे पता करेंगे कि इस पेशेंट की क्या खास चीज़ है जिससे कि हम इसको टारगेट करें? So biomarkers are therefore, biomarker discovery is an entire career. And fortunately, we have done some of these studies, and we have these 2 genes, MUC4 and ADAM12. These were the genes which we found as biomarkers for metastasis. We found out that जो colorectal cancer cells हैं, वो जब primary से metastasis में जाते हैं, तो ये दोनों genes हैं, अगर आप इनको measure कर लें, तो you will get to know कि whether they will be metastasizing or not. तो ये चीज़ें biomarker discovery is very important and it goes hand in hand with the drug discovery कि किसको सही drug देनी है. Next we come on the regulatory and ethical considerations which is always there क्योंकि कैंसर में this is again a big problem as I just told you कि फेज़ 1 ट्रायल्स भी किसको दिए जा रहे हैं? N-of-1 ट्रायल्स जो किए जा रहे हैं जिनके ऊपर सारे ऑप्शन ख़त्म हो चुके हैं। तो I had a team of surgeons, pathologists, then we have the molecular biologist, endoscopist, and it's very hard to convince them कि इस पेशेंट के लिए ये ड्रग ज़्यादा बेहतर रहेगी। कहता ना, अगर 5-fluorouracil चल रही तो वही चलाएंगे हम। हाँ, जब सारे ऑप्शन ख़त्म हो जाएंगे तब आप जो कह रहे हैं तब सुनेंगे। Because there is a lot of regulatory and ethical problems that they have to consider, and it's valid. I would not say that they are wrong, but yes, it creates a lot of problems. Like it's written here, patients are the greatest sufferers, being denied the chance, hope of research, therapy, and paving way for future. So, इसीलिए mostly it's voluntary. So beside economics, regulatory or ethical considerations they are big impediment. But still, there are a lot of scope. There are therapy designations for different targets, like repurposing of the targets. And if we have got substantial evidence that this particular therapy is going to work, then it can also be accelerated by the FDA. All we need to do is to show some good evidence. Orphan drugs के बारे में बताया गया था, कुछ drugs ऐसी हैं जिनका target पता ही नहीं है. Drugs तो बन गई, active हैं, selective हैं, बन भी गई, soluble भी हैं water में, लेकिन उनका target क्या नहीं पता है, काम करती हैं ये पता है. तो इस तरह की drugs भी use की जा सकती हैं. तो एक तरह के there is a treasure that is being already made where you keep these molecules ready. If they can be used for human intervention, they don't have a toxicity, they have got good renal clearance, all those DMPK and PK studies वगैरह करके रखी जाती हैं, so that they can be utilized immediately. So then the review process could be faster. So there are different types of trial designs where standard is this one, which takes a long time. But if you have got an orphan drug, or it could be fast-tracked, accelerated approval, priority review, breakthrough therapies. This is all from US FDA, but there would be similar mechanism by CDSCO in India as well. And this is a very important slide that most of the time, anybody has any idea कि clinical trials होते कैसे हैं? कुछ है idea किसी को? जी बताइए. हाँ मतलब अगर हमने कोई ट्रायल करना है तो यूजुअली होता ये कि देयर इज एन अनाउंस इट्स रजिस्टर्ड ऑन द वेबसाइट. फिर इसके बाद अगर किसी को चूज़ करना है, किसी को एनरोल करना है, तो अपने प्रैक्टिशनर के थ्रू जाकर के फिर वो एनरोल करवाता है. राइट, दिस इज द प्रोसीजर. बट मेनी टाइम्स देयर आर sections of the society which get missed, right? जो बेचारा कहीं दूर देहात में गांव में रह रहा है, उसको क्या पता कि वेबसाइट पे कौन से ट्रायल्स हैं, मेरे डॉक्टर जो है मुझे उससे कनेक्ट कर पाएंगे नहीं कर पाएंगे, किस हॉस्पिटल में चल रहा है, कौन सी कंपनी करवा रही है, दे हैव गॉट नो आइडिया. सो दिस इज an ongoing challenge that we should be including all sections of society in making these clinical trials more successful and more inclusive. So eligibility criteria इसमें restrictive बहुत ज़्यादा होता है कि इसमें comorbidities ना हो, ये ना हो, वो ना हो. So we should be working on these aspects as well for making oncology trials even better. Regulatory barriers, access to clinical trial sites, which means you are living in far off places, and that's why you can't even reach a hospital, let alone a site for clinical trial. Language, cultural barriers. I've seen that. People not knowing the language, they will not sign your form. And low health literacy, mistrust, fear of exploitation. We want them to get help to the clinical trial. They said, "Aapka kya fayda hai isme?" So they think that probably if somebody is enrolling you in the clinical trials, inka kuch fayda hoga. So these all things also need to be taken into consideration while dealing with the cancer patients. So some of the drugs that are already being used in the cancer patients, these are classes of anti-cancer drugs. Most of them are systemic drugs. Systemic means, you would know that as you defined initially, कि cancer cells होते हैं जो rapidly proliferating होते हैं, right? तो ये drugs उन सारे cells को hit करेंगी जो कि rapidly proliferating है. कहाँ पे होते हैं rapidly proliferating cells? Blood. इसीलिए सबसे पहला इफेक्ट ब्लड पे आता है, गट लाइनिंग वहाँ पे आता है, हेयर ग्रोथ, तो इसलिए बॉल्डिंग शुरू हो जाती है, स्किन के ऊपर आता है. तो वेरेवर जहाँ भी सेल्स ग्रो हो रहे हैं सब इफेक्ट होते हैं. तो दीज आर सिस्टेमिक थेरेपीज, जो सबको क्लस्टर बॉम्बिंग. एंड देन देयर आर टारगेटेड थेरेपीज ऑल्सो, जिसमें जैसे एंटीबॉडी ड्रग कॉन्जुगेट्स आप लोगों ने देखा, इसमें स्मॉल मॉलिक्यूल्स भी हैं और एंटीबॉडीज़ भी हैं, जैसे सेटुक्सीमैब है, पैनिट्यूमैब है. दीज आर द एंटीबॉडीज़ that are specifically administered for colorectal cancer patients. And then you have got different mechanisms which are being targeted here. So RNA, DNA, both of them are here, and there are different drugs, pentostatin, 6 mercaptopurine. These are again, but most of them are systemic. DNA जहाँ DNA को वो replication से block कर दिया. 5-fluorouracil is a pyrimidine analog. तो pyrimidine analog का मतलब वो जाकर के decoy की तरह DNA में लग गए, and then they stop the DNA from replicating and the cell goes to die. So again, they are all systemic, means they are not targeted. They are going to kill all the cells that are rapidly proliferating. And then you have the biologics or monoclonal antibodies. They are more targeted, more specific, and that's why अभी जो checkpoint inhibitors जो immunotherapy के नाम से कैंसर में hope नज़र आई है, जो successful stories आ रही हैं, वो इन antibodies की वजह से आ रही हैं कि कैंसर में कुछ खास receptors होते हैं, पहले उसको आइडेंटिफाई किया गया, वो receptors पता लग गए, तो अब उनको टारगेट किया जा रहा है एंटीबॉडीज के थ्रू. राइट शर्जील? ओके, गुड. You already took the nap, huh? Right. So, I wish we can. So these are different T cell receptors as well as APCs, antigen presenting cells or tumor cell. So these are the different receptors, CD19 and PD-L1, PD-1. These are all different types of receptors that are found. And if you block these receptors, that means you are blocking the functioning of the cancer cells and you are specifically killing the cancer cells. And that is the basis of immunotherapy and CAR T cells. Anybody familiar with CAR T? अभी recently India में भी approve हो गई है. Again, very expensive therapy. Chimeric antigen receptor, T cells. Chimera, anybody knows chimera? Yes, combination of two. Chimera is combination of two different things. So now here also there are different generations. तो chimeric antigen receptors में जो receptors होते हैं cells के ऊपर, उसको दो अलग-अलग format में बनाया जाता है. And this is engineered within the T cells. So इसलिए ये CAR T cell therapy कहलाती है. और ये फिर T cells वापस डाले जाते हैं patients में ताकि वो फिर अपने ही cancer cells को मार सकें. This is how a simple one looks like. So this is a CAR chimeric antigen receptor, and these are the transmembrane domain, and then you have got the co-stimulatory domain and CD3 domain. So these are different domains which are going to function, and this is for the recognition for the tumor cells. Once they bind to the tumor cells, these domains help in releasing the cytokines and killing of the tumor cells. Then in case of, this was all about the drug discovery. Now we also have the drug delivery innovations. You have a drug, 5-fluorouracil, and I know a guy, Patrick Chong, he has got his company Nantomics, and he's also doing multi-omics. And he was the one who developed the drug called Abraxane. Abraxane was Taxol in liposome, and he's the wealthiest surgeon in US. So Abraxane was just a liposome surrounding the Taxol. Taxol is very toxic chemical that was used to treat cancer patients. But then he made it better. He reduced the dosages through this drug delivery innovations. So there are different types of formulations. So this comes during the formulation stage. अब drug discovery हो चुकी है, सारे studies हो चुकी हैं, अब उसको बेहतर से बेहतर बनाना है. तो ये सुबह जो lecture हो रहा था ना, first in class, best in class और me too, ये किस में आएगा? Best in class. कि आपको फर्स्ट इन क्लास तो नहीं कहेंगे क्योंकि ये ड्रग ऑलरेडी हो चुकी है. अब बेस्ट इन क्लास कई सारे मॉलिक्यूल्स हैं, आपको उनकी पैकेजिंग करनी है. पैकेजिंग के लिए डिफरेंट तरीके हैं, उसमें ड्रग डिलीवरी और नैनो टेक्नोलॉजी कम्स इनटू प्ले हियर. And it could be passive targeting or active targeting, or it could be triggered release. Triggered release is like release due to pH change, release due to change in the particular enzyme. जैसे हमारे गट के अंदर जगह जगह पे अलग अलग एंजाइम्स एक्ट कर रहे हैं, राइट. हमारे गट में जगह जगह पे पीएच अलग अलग हैं, लंग्स का मामला अलग है. सो बेस्ड ऑन दीज फिजियोलॉजिकल चेंजेस वी कैन हैव टारगेटेड रिलीज. रिलीज मीन्स पहुंच गया वहाँ पे, बट इट इज गेटिंग रिलीज ड्यू टू सर्टन स्टिमुलाई, कंट्रोल रिलीज. So one of the examples is hydrogel. Hydrogel is a substance made, right? So this is machine learning omics. They have done different cancer types here, and machine learning based simulations, predictions. So the Cancer Nanomedicine Consortium that I have made here, we have got a surgeon who collects the sample from the OR, operation theater. One of my students, she's already collecting samples. That sample goes to pathology, where the pathologist tells you this sample is cancer, this sample is normal. He gives us a green signal, okay. Comes to my lab, I do the DNA extraction, RNA extraction, and protein extraction. Then it goes to the sequencing facilities, where we will be generating all the sequence data. And all this data will go to the AI guys. AI guys build a prediction model, and they say, okay, this patient has got this much life, this patient can be treated with these drugs, and then it goes to the oncologists in the oncology department, and it's up to them to decide whether this drug should be given to the patients or not. So this is my dream team. Alhamdulillah, I've made the team in AMU, and we are hoping that we'll have a spin-off company by providing these reports to the clinicians if everything goes well. So in conclusion, I would love to hear the conclusion from you guys rather than telling myself. If one of you, few of you can help me with the conclusion. Yes, please. What do you get from the slides? That will be the conclusion. My conclusion is more important than your comprehension. Anybody? As soon as you conclude, we are ready to go. Come on, quick. Yes. From slide 1 to slide 55. Any one thing that you remember? This will also help you for your exams.
Cancer is a continuous evolving syndrome, if you would like to call it that. And it's the one thing that I learned that a series of compiled omics that you mentioned would eventually lead to finding a specific treatment for cancer itself.
Good, that's very satisfactory conclusion for me. That he understood that multi-omics is defining a cancer cell. Yes, anybody else? Okay, good. Anybody else? Come on, quick. Hmm. Hmm. Yes. Okay.
Cancer is not due to the change in the DNA only. It is because of change in protein and some other markers which are present in the cell and outside the cell. So it is not only because of the mutation in the DNA.
Good. Wonderful. Thank you. Yeah. So it's a combination. It's the elephant. It's not just the tail. It's not just the ear. It's not just the trunk. It's a combination. Right. And with that, I would like to acknowledge and sincerely acknowledge my two PhD students. Swati, she is not here. She's on vacation, enjoying a wedding. And the other one, she is tirelessly working at the back, Sayyida Shabi Zafar. They even helped me in preparing this presentation. So this presentation is not entirely my creation. They have helped me in preparing this presentation. Any questions or we wind up? Yes, Bashir. Okay, the sequencing is of several types. One is whole genome sequencing, and people rarely do it. It's expensive. And first of all, we don't do sequencing. We don't have any sequencer, even an entire oligo. Next is exome sequencing. Exome sequencing is that portion of the genome which is expressed in the form of genes. So this is known as exome sequencing. So mostly we go for exome sequencing. And the third type is targeted sequencing, where we know that we have to sequence these 50 genes, 100 genes, 200 genes, 500 genes. This is known as targeted sequencing. But we go mostly for exome sequencing.
Exome sequencing.
Not specifically which has the mutations only.
No, no. There is no such sequencing where you can do only mutation. That is called targeted sequencing, where you have got the panel of genes that are carrying mutations. So you can sequence those, say 50 genes, 25 genes. That is targeted sequencing.
About the life expectancy of the patient, that on the basis of the pathological finding also we can tell which stage of cancer is that, stage 1, stage 2, stage 3.
Yes.
Then what's the role of the AI team here?
This question is about the use of AI in predicting the lifespan. I think nobody can define the lifespan.
Okay.
To be very honest, there is no such model which can tell you, because so far we have not been capable of capturing all the factors that define a lifespan, and that's a distant possibility. But yes, it can tell us, the biomarkers tell us that if suppose CEA levels are very high in case of colorectal cancer, the disease is going to progress very fast. Now even the young patients who are having colorectal cancer, they have got a prognosis of going very fast disease and very worst prognosis. Most of them die.
Okay.
So these are the factors which can define, yes. Right.
So what's your take on metagenomics and gut microbiome specifically, like as a predictive biomarkers for colorectal cancer and other cancers as well?
Yes. So I'm not answering your question directly, but I usually say, give me more money, I'll do that as well.
Is there a potential in gut microbiome?
As I said, more we are able to capture, more closer we are going to the target. In fact, Patrick Chong, I met him in San Francisco and I asked him why you are not doing metabolomics. He said, we are doing it, but you will see it soon. Check his company, Nantomics. And they have got a tool called Cancer GPS, which is giving to the doctors for doing all this analysis. And this is happening in US. They are already treating the patient based on their genetic data.
Sir, when we do biopsy, as you said, if you disrupt the vasculature of a cancer cell, it would spread. It would like go into a shock. It would become aggressive and spread throughout.
Yeah, this has been observed.
Okay.
That it had happened in some patients. If you disrupt the blood supply, they became more aggressive.
So in biopsy, if a tumor cell is cancerous, we do biopsy to test whether it's cancerous or not. So that won't disrupt its vasculature and lead to.
It could. In fact, in prostate cancer, the guidelines are that don't do anything. Just wait and watch. Prostate cancer patients are advised, don't go for any surgery, don't disturb this tumor. It might hang over for several years without any symptoms, without any change of lives. So it's a possibility. Yes, please.
Or other lung cancer, they are advised that go for surgery.
Yeah, surgery still remains the best option to see and remove, rather than relying on drugs which will go there and do the job that you can do by your knife. So, but there are also limitations. If you leave 1 or 2 cells, even that will be coming back. Yeah, the metagenomics, I haven't got the money to do that yet.
Not by you, by anyone else.
I am not sure. I didn't get the question.
How do we evaluate drug resistance mechanism in cancer cells?
So there are different mechanisms by which we can assess whether this drug is resistant or not. So one of them is like drug efflux. So drug is going inside the cell, but it's pumped out. So if we find out that which are the ion channels which are doing this job, or the pumps that are effluxing it. In fact, one of the molecules that we discovered, ATP8B1, it's also a flippase molecule. And maybe it's also helping in drug resistance. We don't know yet, but it is also an ion channel Felipe-based protein. All right, so I think we're done for the day. Let's see, and we'll see you tomorrow. And I apologize for today's delay in lunch. It was a huge delay, and it should not have happened. But let's hope we do it better tomorrow, inshallah. Okay. So there's some tea outside if you want to have some more caffeine. Otherwise, we'll see you tomorrow. So we start at 9:30 again tomorrow, right? Yes.